Okay, everybody, we are back, and we're about 30 meters off the bottom, so it should be coming into view very shortly.
Sonar.
This is Chris Kelly from the University of Hawaii. I'm serving as the biology science lead, and next to me on my right is...
I think it is.
It is.
Jasper Conter, who had to flip his mute button off in a nice way. We are indeed just waiting to see what we'll find. We're showing about 30 meters and closing in from slightly to the north of the ridge line.
We're on the geote that's tentatively called Delilah geote. We didn't know it had a name until Jasper looked it up yesterday. And this is about 30 miles to the northeast from where we were diving yesterday. We're also diving on a ridge. And you're seeing a live broadcast from the NOAA ship Okeanos Explorer, and it's a Deep Discoverer remotely operated vehicle.
Starting to see you in the fisheye. Yeah, copy.
See me in the fisheye.
I've got you solidly on the fisheye.
So this is sort of the
back right.
The moment of truth here.
Starboard.
I always enjoy getting the first glimpse of a place that no one's ever been to before.
Yeah, real exploring, right?
Yep.
15-meter delta.
Bring my heading around.
Do you want me to turn towards the ship as well?
No, just keep your heading.
Okay.
Because you're pointing up slope too. I see ground.
And there's the bottom.
I see ground as well.
All right.
Yeah, you can bring your heading to
starboard
070.
Sorry, port. Yeah.
And it looks like sort of a similar look as what we had on our first dive. Lots of loose rocks with some sediment in between. Not quite as much like yesterday's dive where we had these more solid manganese encrusted layers.
You can see my light pool right there.
There's more rounded shapes here, so probably more billow lava-like.
Bit of current down here.
Yeah.
And we're just waiting for everybody to get settled here.
Yep, and I
That
believe
is opposite of what we observed on the
There's
surface.
definitely more sediment here, and as you can see below, there's some bed forms, which is sort of suggestive of some current
ripples. Yeah.
Going to bring sonars to 30 and 50 meters and adjust the gains a little bit.
I actually like where my gain is.
You do? Okay.
Yeah.
So I can see a few things growing on the rocks. It's not a huge number of animals so far. I can see them sort of spaced out a bit.
You're okay.
We're
So
still
almost
pretty high up, so it's hard
down,
to see things.
slightly to the
and
left.
yesterday we didn't see a lot of stuff until we got quite close because there were very, very small Octa Corals that were all over the bottom.
You probably need to turn more to port as well, though.
Yeah, there I am.
Yep. Gotcha.
Lost my pen.
Yeah, there's a bunch of stuff there.
There you are.
There's a couple branched Octa Corals. Looks like a big sponge, a couple big sponges.
Okay.
Yeah, so we're going to see some stuff.
Yeah, it looks like it could be even 90 degrees.
this is upslope here.
Yep, I'm going to pull up slightly.
Oh, there's a nice size fan right there.
Yep.
I think that's going to be turn out to be a bamboo, but
Yeah, I think once
it's
you get
a
on top of that saddle,
unbranched bamboo
you
right
head
behind
to,
it as well. So
port, we'll start going up.
got a bunch of stuff to image right
So
off the
going
bat.
to high pack there.
Hey, how you doing, co-pilot? You good?
Good. Looking good.
Yeah, you can probably come down just a little bit, but
Okay.
you're good. So you want to look at this watch sleeve?
Yes, please.
Okay, coming in.
Very happy with this.
So Scott France has already ID'd from this distance that this first coral is a bamboo, and it's lyrate in clad D. Excuse me, clad E. And what that means is that the genetics of this family are in the process of being worked out right now. So a lot of
Emily,
the
now,
bamboo
if there's
corals
pen over there, could you
are
grab
only
one?
sort of known as which particular genetic clade they belong to. So it's a very complex group, and it's got a lot of different
Should have
species
lasers on at some point.
in the family.
I can turn them on right now.
Maxim.
You can sort of see the lasers shining through the water.
Oh yeah.
That means there's a lot of
A lot of stuff in the water.
stuff in the water,
Yeah.
a lot of particles.
So Asako Matsumoto from the Chiba Institute in Japan has joined us. She noted a sponge. We will take a look at it in a minute after we finish the beautiful zooms on this animal.
Chris, I probably should have typed in clade I4. We have another code that we don't usually use that is the E code. It just makes things more confusing. But I think if we're looking at the historical records from last year and this year in the event logs, probably I4 is better.
Okay, thank you, Scott. And that was the voice of Scott France again from the University of Louisiana, Lafayette. So he is one of two people that are very actively working on this very difficult family of Octa Corals. The other person is Les Watling with the University of Hawaii, and they're collaborating, trying to figure out what's going on in this family.
Is there anything else you would like to see on this?
I think we're good, pilot. Thank you.
Hey, when you're good video, you can come out.
Should get a full screen with lasers or full species.
We got a dead sponge behind there. Dead sponges aren't that interesting. We kind of like live animals. And we've got another bamboo behind it, an unbranched one.
What else would you like to see here? Watch this.
Let's see if we zoom out a little bit. I think Asako Matsumoto was pointing out a sponge. It might be a little off to the left. Yeah, that
Maybe-
could be it in the back there.
Maybe behind a rock formation.
Yeah. And I'm not sure what we got here on the left coming into center right now. It's
The
kind
lower
of
part or the upper?
the
Chris,
upper.
did the
Can you
He's looking
partially
up
pull that
above and to the left of the lasers. Well, the others.
Oh, I see. Yeah, let's take a look at what's right here to the right of the lasers. I didn't even see that.
Hey, come on in video.
Maxim.
So we are looking, Scott has identified this as a Chrysogorgia in the family Chrysogorgiidae, genus Chrysogorgia.
Bridge,
and
are we now?
Asako concurs.
Go ahead, Nav.
That's the
We
Octa
have
Coral.
reached the bottom,
And
and
then
I'd like to change our
inside of
bailout
the
plan
Octa
to
Coral is
315
a squat
degrees.
lobster, and we believe this
Copy
the name
bailout
of this
plan
guy
change to
is
315.
well, not the name, but
Copy,
the
thank you very
genus
Slightly
is
out if you like it more,
Europtychus.
just let me know.
His name's probably just Bob.
Right.
Yeah.
Actually, I would guess
Pulling out a
the
little bit.
name is Mary,
Oh.
other female.
Ah, there we go.
Okay.
Okay, give me a good video.
Okay, pilot, we have just about reached our dive start waypoint, so whenever you find the peak of the ridge, you probably want to turn to port and follow it up.
Yeah, Roger.
Possible tourniquet on the bottom left.
Oh, take a look at this. Sure, let's take a peek at this.
Okay, come on in.
So we will wait for the chat room to weigh in on this. I have an idea what it could be. I'm going to see Scott France has confirmed it's a tunicate, and so is Asako, and I would agree. And there's only one family I know of carnivorous tunicates, which I think this is, and that's Octonemidae, and I assume that that's still a correct name for it. So tunicates are perhaps more known for, yes, Scott France has confirmed it's an Octonemidae, and so this actually eats things. It's not just filtering
Is it like an opening?
particles. Yeah. So we see these octonemids about every place we dive, at least one of them, and who knows if it's the same species? Probably isn't. You know, there could be very subtle differences that we're unable to differentiate. But this type of tunicate seems to be fairly common down at these depths. And Scott was sort of looking at a fuzzy thing to the left of the base of the tunicate, wondering what that was.
Go ahead, video.
And yes, I'll go ahead and take a stab at an ID here. Fuzzy stuff.
Fuzzy stuff.
Yeah, I concur with Scott. It's fuzzy stuff.
Yeah.
Could be a hydrozoan, bryozoan.
Actually, it was Bruce that asked if it was what the small fuzzy
Oh,
thing
so
was.
I
you're
concur with
concurring
Bruce.
yeah.
Okay, got it.
Yeah. I think I'm going to go with fuzzy stuff too.
Okay. Asago's probably right here. She thought it could be a bryozoan. That's a little more accurate than perhaps fuzzy stuff.
If you could video. Anything else here?
No, maybe that sponge over in the upper left corner looks like a live one. I think the other sponge behind this little tree is dead.
Yeah, copy.
It looks like a dead Lefroyella in that genus. Too bad it's dead because they're really cool sponges to look at closely. But this one right up ahead, white, is definitely alive, and it's a glass sponge of some kind. And it's got a nice little feather star cap on its head.
It found a nice hat.
Right.
Partial.
So this particular type of sponge might get a little bit confused. It could be one of two genera here. It's either a Feria or it's an Aspidoscopula. I think it's more likely to be Aspidoscopula. Both of those genera are in a family called Feriidae. So it's a very brittle sponge and not soft.
Co-pilot.
And then this, whoa, there goes
Pilot
an eel
actually
right
kill
through the
lasers.
Oh,
scene.
lasers.
And he's swimming off
Yeah.
quickly.
Let's come in on the sponge.
On the replay that I'm seeing here next door, it looks like that the fin was set back quite far.
Okay, so it's an eel. It's going to be a synaptobranchid. What has Bruce got down?
Bruce.
What's the fish eel? I guess ELO. I should remember the abbreviations myself.
Yeah.
I forgot.
He hasn't seen it yet, though. He's still waiting for it to shoot by.
Oh, he hasn't. Oh, okay.
Okay, I've just seen it. It did look like a Synaphobranchus in either Brevidorsalis or Oregoni, the one with the shorter dorsal fin. It did swim by too fast to really see. I do have screen lag here, so it takes me about half a minute before I see things that you see.
Yeah, thanks. That's the voice of Bruce Mundy. He's one of the world authorities on fishes here in the Pacific, and he's at the Inouye Regional Center's Exploration Command Center on Ford Island in Pearl Harbor. So thank you, Bruce, for that ID.
Looks like there's something on the other side of this.
Yes, you're welcome. And I actually work for the NOAA Pacific Islands Fisheries Science Center. So thank you.
Yeah, thank you. So the other thing is dead. We could skip it. It's just an older one of these that's in the background. There is, I don't know if you can take a look at that coral that's dead ahead now in the center. It's got the long branches, sort of a bushy thing. Yeah, that one.
It's a little bit to the left of the middle.
Right.
This guy in the center?
I said a center, up center. So I think the lower center is another smaller version of what we imaged when we first came down.
Or something to the lower right.
Yeah, maybe I will get a look at this longer coral for the moment. It's probably, well, let's take a look at the anemone if you can, and maybe a Serianthid, which would be a good to document since it's in the sediment here. That's a tube anemone, a common name for Serianthides or Seriantharians. These are types of cnidarians that live in the sediment.
It's partial.
Sort of hard to say there. I think it's going to wind up, I think it's just an anemone. Just he's probably on a rock that's just barely under the surface there.
I agree with you, Chris. The tentacles aren't right for a Serianthid.
No, when he was just going over, when we were higher, I thought I saw a darker center that could be some of those little oral tentacles around it, but it clearly doesn't have them with this view. So, yeah, I was mistaken.
Yeah, that looks like just the reflections of the mesenteries
Going for a full
underneath.
specimen.
I mean, certainly in the spot that you would expect a tube anemone, they're typically burrowing into the bottom, but yeah, there must be rock or something hard not too far below the sand grains here.
Pulling out for fish.
It looks like there might be a
I
prognoid
guess
just
so,
to
yeah.
the left.
Yeah, that's where we're going for right now.
So we want the long coral in the back.
Yeah, that's correct. Thanks, pilot.
Fish is off to your right-hand side. See it in serious.
We want to try to get that fish. He's off to my starboard. I can come back for the coral.
Yeah, if we, you know where he is? Is he showing on Sirius or?
He should be.
It's another crinoid down below.
I think I lost him. Do you see him, Merlin?
Last time I saw him, you're on the back side of that rock to the bottom
Chris,
right
do
of
you
your
recognize that other one that's in the background? that was in the background on the left there that was branching very low and had 6 or 7 long branches?
Yeah, I think it's going to come in now, Scott. He just swung briefly to catch the fish, and he's coming back down to it now. I think it's going to be a Narella.
Yeah, no, I realize, and I think we're going to look at this planer one. Oh, maybe not. Okay, you're going to look at the other one. There's another planer one that you've just passed over, but yeah, I was asking about this one in the background.
Yeah, the little planer one, I think, is another one of these I4 clade. It's just a small one. I got a pretty good look at it.
Oh, really?
Yeah.
No, I don't think so. They shouldn't be that. They shouldn't start their lyrate branching that small.
Well, we could probably take a look at it if you wanted to check it out.
Yeah. That was the one I was referring to as I thought it was a primnoid, that earlier one.
Right. So this is another type of Octa Coral, and it's in the family Primnoidae. And my guess would be it's in the genus Narella. I'm just waiting to see if anyone else has got a different opinion in the chat room. Now Scott suggested it might be a Narella as well.
I think our best view is down at the bottom where the joints are.
I think there's a number of these long branched Narellas, if that's what it is. So I'm not sure we can nail it down a little further than that. There's something called Narella.
The view I'm looking at, I've got a nice view of the topmost whorl, and you can count very nicely that there's 5 polyps kind of arranged in a ring around that main axis. And so you have a series of these rings, each with the polyps, and they're somewhat facing down, and when the tentacles contract and the polyps pull themselves in, they would curl downwards. And that's one of the hallmarks, as you mentioned, the genus Narella. So that's probably a very good guess. You can also see nicely on these this imagery that the sclerites in the bamboo corals, we point them out, they look like little needles and rods, whereas here there are more like fish scales, highly ornamented fish scales that overlap one another like a knight's suit of armor.
Pulling out for full specimen.
Go ahead.
You may hear some tapping in the background. Somebody's doing maintenance work on the ship, I think right on the wall next to us. So that's what happens when you're out at sea. You gotta take care of things, you know. So they're doing a little bit of work. Hopefully they'll finish that up pretty soon. Got a little ophiurite on them.
Pulling full wide.
So I don't know how easy it would be to take a quick peek at this coral that was off screen below us now. Scott was wondering what this might be.
He would be down looking a little too.
Yeah, it should show up any second here. There he is, right there.
Yeah, you don't need to spend a lot of time, Chris. I think it's the second.
It is. It's a different. It's definitely not what I said it was. So good correction there, Scott. I took a snap ID, I guess.
Yeah, it reminded me a lot of a sort of typical
Lobster in the bottom right, underneath the coral.
So I think this is potentially a different species of Narella, maybe? It's definitely in the same family, Primnoidae.
Yeah, it has kind of a dichotoma branching, doesn't it?
Yes, And then after this, I think we could probably go ahead and start moving up slope. I think we got the main things here. That we're right around the landing site. Oh, we've got a lobster, a Polychaete. That's a nice, a very small one. These are, I don't know if they're called lobsters. I'd have to take a look at them, but they're considered to be basically living fossils. It's a very early or primitive member of the lobster group. So they're always very interesting to see them. They all tend to be one here and one there. They don't seem to aggregate. So if you're lucky, you might see one per dive, sometimes two. A lot of manganese crusting on the surface there, Jasper.
Yeah, it's pretty thick here. I've been looking around to see if there's any rocks to pick up. Most of them look kind of stuck.
So to intercept the slope, maybe more like 090. And then once we reach it, we'll turn to port.
So we can, if you bring your heading to 090, co-pilot, then I can push out and we can get it moving.
On the way.
Co-pilot sonar looks good.
Yes.
So we can get it moving.
So looking up the hillside, it looks like there's a fair bit less sediment.
Do you agree we're a bit
so
north
I
of
think
the
if I'm looking on the map over there, we really landed in a bit of a
us
level spot
I
between the
see.
ridges.
30 meters at 0.2.
Now we're getting away from that. We get on more solid ground again.
Yeah, so we might actually see a change. It almost looks like we're starting to see a few more things already as we get onto this consolidated
so
rock.
If you keep your heading this way,
Yeah, there's
I'll
a
adjust to deal with the current.
Okay.
a couple
this way,
different
which
things that are
09.
showing up right
Yeah.
now. This looks
The
like it's a
direction
black
of
coral
movement.
Yeah.
here on the
Bridge,
left-hand
RV nav.
side.
The
I wonder
sort
if
of
we could take a quick look at
tan-colored
this.
one.
Yeah, this
We'd like a
tan-colored
ship
one.
move, please. 30 meters, bearing 090 at 0.2
One
knots.
behind it looks like it could be another Primnoid, but
30
this is
meters
quite
bearing
different. This one here,
090 due east at
Pretty
0
sure
to
it's
2
an
knots.
Anipitherian,
Good
but
copy, thank you.
Scott also happens to specialize in Anipitherians, so we'll wait to get him to weigh in on
Come
this.
on in, Mitty.
This looks similar to what we sampled yesterday.
Yeah, more branches though.
Yeah.
Those ones yesterday had only just a few branches or none.
There are several of these black corals that have this similar morphology where there's the main axis and then these very small, fine little branches. And the polyps are almost those little dots on them. They're very, very tiny. So it's kind of confusing sometimes to try to identify them, even to genus, unless you're a specialist. They're branching, but more often it's those small pinnules, the structure of the pinnules, the number. And then these are also called, in addition to black corals, they're called thorny corals, and that's because the skeletons have a series of spines. And so the size, shape, and distribution of the spines on the branches are what are largely used to identify the species and so on. In fact, very rarely is the soft tissue itself used, and so often you can identify a black coral in your hands if you just have the skeleton and not necessarily the soft tissue.
So Scott, do you have any candidate genera here, like maybe narrowing it down to 2 or 3 genera?
Hey, Nav, did
Yeah,
you say ship was moving?
Heptapathes,
Yeah, bridge
Heptapathes, those are the ones that are going through
Thank
my
you,
mind.
Rich.
What,
Yes,
What was
just
the
move
guy
just
in the back?
started.
Thank you.
Distance?
Co-pilot
30 meters.
video.
090
Sorry, repeat.
at decimal
Let's
2
get
knots.
this guy in the dead center.
Yeah, and this is another Narella, it looks like. It's got some ophiurids on it. These are brittle stars. And it's also got a crinoid, this white thing right in the center. All right, well, whenever we're done on this close-up, then maybe we can start heading up slope and see if we can get into a little denser community here. I would view this community as kind of typical in that there's a smattering of animals here and there, but we're not seeing really dense numbers like we were seeing yesterday, for example. So again, as Jasper pointed out, maybe we'll see something as we get in a little further and slope in a little steeper terrain. We're going to hang out just for a moment. There's a shift change going on right now. And while the shift change is going on, I'll go ahead and give you a little bit of an update on environmentals. We're at a depth of 1,981 meters, and our dissolved oxygen is 3.2. Our temperature is about 2 degrees Celsius. We are on, I guess we'll call it from now on, Delilah Seamount, Delilah Geode. It's a flat seamount. And you are watching a live broadcast from the ship, the NOAA ship Okeanos Explorer.
Pilot.
Now we're getting into a nice little area of loose
good.
rocks.
Yeah.
Video.
I'll have to see what sizes these guys actually are.
Welcome aboard, pilot.
Now this seamount,
Nice
I didn't
to see
even
you.
tell you where the seamount is. The seamount is very close to 200 miles northwest
We're headed
of Wake
090. I'm
Island.
lined up on 092,
And it's within
so
the
pretty much on track.
Wake Monument, or the Wake Unit of the Pacific Remote Islands Marine National Monument. So we're doing some of the first surveys ever in the deeper waters of the Wake Monument.
Swing arms out at all or anything?
So you're seeing things for the first time just as we are.
Okay.
I'm hoping that
wanna
we've got a
drop
couple
down?
of aquariums displaying the
Come
video.
down?
I know that the Waikiki Aquarium in Waikiki Beach, Honolulu, has got a display center, and they're probably showing this
Center
video.
up HD2.
Welcome to
Yeah, of course.
any of the
do
visitors
it
to
for
the
you, pilot?
Waikiki Aquarium who happen to be watching. And
you want me to
also
center up
the
your
Maui
videos?
Ocean Center on
Videos
the
asking.
island of Maui
yeah, sure.
also has a viewing center, and they're probably watching this video as well. It's another
Swimming
beautiful
fish.
aquarium on Maui.
Left half.
So welcome
screen.
to the folks who are over there who may be watching the broadcast this morning. On our chat room, currently we've got Sako Matsumoto from
Slightly
Chiba
lower than
Institute in
center.
Japan, and Bruce Mundy from the NOAA Fisheries Science Center
Slightly
on
right. You see
Ford
it?
Island. Charles
Yeah, it's
Wall,
out there. See 'em?
he's with the NOAA Marine Protected
forward.
Areas
Yes.
Division.
You got room to push out there.
And
if
we've
you
got
want it.
Michael Vecchione from the Smithsonian, and we've got Scott Frantz from the University of Louisiana Lafayette.
And the
We've got a
screensaver.
fish stray down. I wonder if we could get a look at this fish. Seems to be cooperating, at least for the moment. Aha. So, Bruce, you're going to be happy in a few minutes. We're seeing something different. Bruce has a slight delay to the video, and some of you also may have a slight delay to your video, and so keep watching, because the fish I'm pointing out will show up any second.
It's getting close to you
It's
now.
right and close with you, yeah.
Pulling out so you can see where he's
Now
at.
this is
full line.
Bruce. I see the fish clearly enough to tell that it is one of the deep water cusk eels, family Ophidiidae, which is one of the families that we see most often, along with these cutthroat eels on these dives at this
You can
depth.
see it in your HD2.
My guess is that this is in the genus Basozetus, which is one that we see fairly often. It has moderately small eyes, a fairly robust body for the Ophidiids, kind of distinctive,
Center it up, I can
rounded,
zoom
very
back in.
bulbous head. So there are a number of species, and the species are very difficult to identify unless you have a specimen in hand. So a cusk eel in the genus Basozetus.
Great, thank you very much,
Ship
Bruce.
is in position, pilots.
I think that's all we have video.
It's like he's had
That's
enough of
right.
Yeah,
us.
he said that's it for now.
Yeah.
I'm gonna
Go find someone else to light up.
You know, corals tend to cooperate a little
Yeah,
bit better.
Yeah, they don't run away so much.
No, they don't. Not quite as shy. Although some fish will actually stay there and pose for a while. I sometimes think some of the
Yeah.
Go ahead, Bruce.
Oh, I was going to say, if the fish were more cooperative, zooming in on the head, with particularly the gill cover, can be helpful. Some of these have spines on the gills. This one's not going to be that cooperative, though. It's not going to let us do that.
Lasers are now on.
It's trying to size up some of these rocks. The lasers.
Oh, yeah.
You can actually see the lasers propagating through the water column.
So I'm forgetting myself here. Usually I like to try to grab a rock right off the bat.
Yeah.
Do you see anything that might work?
Oh, there. Now I actually see the dots.
Yeah.
These are relatively
lasers
large.
Looks like loose rocks.
Yeah, Washington.
I lost it. Oh, there they are.
Shout out.
That's a good bit bigger than what we picked up yesterday, I think.
Yeah, and then those ones up above to the upper left there, they look pretty substantial too, a lot of them. Some of them are smaller though. Yeah, because if we get into a consolidated substrate as we get on the actual ridge
we're not
crest,
picking
we're
up
not going to
anything.
be right. So I don't know, do any of these fit the bill?
Pilot, would you like me to turn left to keep you in view?
Maybe like, yeah.
Okay.
As long as they don't exit, yeah.
Coming left.
How about that one to the left of the lasers right now? Lasers are on it.
Yeah.
Facing.
No?
I don't know.
How about this one up here to the
further up, maybe? Yeah.
Yeah, a little bit over to the left, maybe, and slightly above the
co-pilot.
lasers. Anything
Heading
there
to
that might
dive end is 67.
Slightly above this one?
Or actually 71.
We're looking a little further up still.
I have uphill starboard of that, but we can scooch over towards him if he needs more room for this. Seems okay at the moment. But
So the
I think
reason
they want
we're
to, acquire
looking
the
at
ridge
collecting
and
the
then go up.
rock here, it's not just going to be a paperweight for Jasper's office. We're actually, everywhere we go, we try to collect 1 to 2 rocks, and the reason for that is many of these seamounts have not been, their ages have not been determined, and their chemistry has not been determined, and just by grabbing a couple really good rocks, we have a chance of actually figuring it out. But, you know, I don't know if you want to mention anything more than that, but the rocks are most definitely as important to collect as some of the species that could be new species.
Yeah, so we use the volcanic rocks that make up the main volcano structure that makes up the seamount to see if we can get something out, and to see if we can use some lab techniques to come up with an age.
How about this one right here to the directly to the left, and directly, yeah, he's swinging over. The laser's a little bit above it. Keep going and down. That one there with that, well, it's a little small, I guess.
That's a bit, yeah.
that's actually
Maybe
20 centimeters.
let's look a little bit
It's a
further
good
over
size.
to the left,
I think that rock is bigger
past the
than you
really
think.
big one.
So some of you might be wondering, well, it's a rock, just grab it. They're all the same, and they are not all the same. As it turns out, there's a huge amount of crusting on these really old seamounts. It's manganese and other types of metals and minerals that are precipitating out of the seawater. And some of these rocks are nothing but pure sediment or manganese crust. Other rocks are
I think everything
manganese
to the
crust
left there
over
is pretty
sediment.
sedimented.
And what we're looking for here is a rock that actually might
That one
have
to the
some
right, the top
volcanic
shot
material
before might be
inside of it,
loose.
some basalt,
Maybe
because
come
that's what you
above it,
need
some
to
above
age
it
the
might be
seamounts.
loose.
How about,
I think
you think we could go up a little higher to the left and up? There's a couple of slightly more angular looking rocks sitting there.
Copy that. Just, yeah, let me know.
Yeah, that area that we're getting to with the lasers right now, maybe one of those. How big do those look?
A lot of these look stuck in place. They don't look loose.
They do. If you see a nice one, I'd be happy to poke it, but they do look stuck.
They look pretty stuck, huh?
Yeah.
Some of the ones to the right were maybe a little looser, but this might just be an area.
So this is the other challenge, obviously, with this process, is finding a sample that not only looks like it might have something else inside besides manganese, but also finding one that is loose enough that we can pick it up.
Let's see.
Yeah, maybe that
Washington,
one.
do you want to keep looking in this area,
Or how about this bigger one right here? You think that could work?
not
It's
sure
right
if
above the laser, slightly to the right.
Just slide up.
Yeah, that one there.
To the right?
To the right, that
there
one.
There you go. Does that look... Can we zoom in on it a little bit?
Yeah, it's pretty big. Video quick zoom.
May or may not be loose. I don't know. How about the one to the right that's a little bit more to the right? Keep going. There's one that looked like it even had some angles to it and maybe up just a little. Oh, no, there it is.
There it is.
That's it. What about that one?
Right there.
Yeah.
Looks a little more reasonable.
Think that's free?
Can we push that around a little bit?
Yeah, I'd be happy
Maybe
to.
squeeze it?
Yeah, absolutely. All right, Dave, if you want to come out.
Let's hold here for
So
now.
for those wondering
now,
why I asked
Yeah.
for squeezing, sometimes we're able to squeeze it in half, and if we are able to squeeze it in half, that usually means it doesn't have a volcanic rock inside. The reason we're interested in the volcanic ones is that gives us a better look at these volcanic structures. Sometimes these samples have only manganese, sometimes they have sediment inside, and the sediment is much harder to work with in order to find an age for these rocks. It's usually not really possible to use the sediments unless you start looking for microfossils and things like
stow
that.
port lower swing arm.
I think it's better be safe. Yeah, you can stow it.
Can leave it if you want it. I just.
So in the
It's
meantime,
just
we're
being blocked by that rock right now anyway.
trying to reposition the ROV to see if we can actually
Push it.
push this
See
thing
if-
around.
The reason you got to push it is because a lot of times the manganese crust just glues it into the other rocks around it, so it's impossible to get off the seafloor. But we will see about this one. And if you're joining these dives for the first time, what we're talking about, this manganese crust, you can see a really good example of it on the rocks right above the ones where the lasers are located. See a very sort of bubbly, bumpy type of surface, then that's what this manganese crust looks like after it's had millions and millions of years to form a thick layer over the rock.
Let's give this rock a poke.
Manganese crusts and the communities that live on them is one of the project's priorities, and the actual project we're working on here, it's a 3-year effort by the Office of Ocean Exploration and Research. That's one of the NOAA offices, and
What would you like?
it's a 3-year
I'm
effort in
happy,
the
yeah. What
Pacific
would you like
called
up
the
there?
Capstone Campaign to address
I'm fine like
monument
this now.
technology,
Okay.
science,
Get
and
to it.
ocean needs, or something
Yeah.
like that. I always screw up what the acronym means. But
Right
we're focusing on monuments in the Pacific
give you a
because
little
there's
more than
quite a lot of them here. And then aside from monuments, we're very interested in manganese crust communities because we're trying to get a head start on surveying the communities in advance of deep sea mining really taking off. And that industry is going to be active in the future. And so we're trying to survey these and provide baseline data before that happens. And we've got some other priorities too. We're very interested in deep water coral and sponge communities. Corals and sponges are the dominant benthic fauna that you see at depths below 1,000 meters.
Looks pretty cemented either way.
Nice.
If you can get in that, I guess.
Yeah.
Yeah, that's kind of a tough one there.
Yeah.
That looks like a good rock. Otherwise, if the thing would, if the darn thing would just move.
Yeah, that's in there.
Sorry, Washley, it's that stack. That's not budging. Is there anything else in the immediate area, or should you want a quick poke?
Well, I'm not sure we're gonna come home with that one there, Jasper.
No, that's a shame. I think that one looked nice.
What about the one right above the right laser there? Is that too?
Hold on, co-pilot, I can't hear you.
Oh, sorry.
Go ahead.
There's one right above the right laser that might be. You could try it.
Yeah, you can see if you can push that one to the left, maybe.
Absolutely.
looks like that one's going to be a...
Yeah, that's glazed.
Sorry, Washley. Those are both stuck.
They are glued in manganese.
Yeah.
How about the one above it on the surface? Can we zoom in on that a little bit?
That's stuck. That's stuck. That's going to be glued harder than the other ones are, I think.
Then I think we're...
well, we'll keep our eyes open as we continue on, and
I think we're done in this corner.
Yeah.
Right, sorry, Washley. We'll get you another one.
We appreciate the efforts. Thanks, guys.
You know, I kind of feel that way on certain days, too. I just don't want to budge. I get up, and I find a spot on the couch, and that's it. I'm done. Get a manipulator next to me, and I'm not moving.
I was going to say, there's a name for that. It's called Saturday.
Yeah. There's another name for it. It's called couch potato, I think. You know, right around the football season.
That seems to happen more. It occurs more often, and then it's usually today, Sunday.
Yeah, it is. It's kind of strange the way that works out, but.
Washley, I'm just going to get push out a little quick and recenter on a Sirius.
And Scott France is making an alternate ID on the black coral, suggesting it could have been a lilipathes.
6-8 here. So if you see anything loose on your way over, then you can stop. But otherwise, get in the middle of this. We'll head up. I guess.
Bruce Mundy has also added a little bit of information in the chat room as well about that cusk eel that we saw earlier.
Yeah, right in this area that might be.
And he's noting that there's two species in the genus Basozetus that are known from the vicinity of Wake Atoll. So there are some possibilities. This may be a known fish that we were imaging earlier.
Yeah, that's where I've got them.
Loose. Where do you see this?
On the Sirius cam.
Yeah, this is Bruce. So as I said in the chat room, unfortunately, a lot of the characters that you use to identify the species
it's hard to tell on G2's cam, let alone
are internal or very difficult to see.
And the shadows play tricks.
The number of fin rays, the numbers and sizes of scales, and then some of the internal characters. So unless a specimen's collected, we'll never really know what we're seeing, except for the genus.
I don't know what that was.
Off left.
It went off left.
Yeah, that's the tricky part, trying to collect
some pink
fish from 2,000 meters depth. Well, it's usually not done with submersibles. I think it's usually done with some type of fishing gear.
Yeah, and you don't think the little beach net, a little scooper from the local big box store is going to cut it here.
No, you're probably not going to be able to fly cast for it either.
Yeah, next to the dead sponge.
That's a dead sponge.
Is that angular enough?
The one in the sand?
Yeah. Oh, if it's in sand in this flat, then it's likely fallen from somewhere, or it could have fallen. Or if you find it on a wall, then it's likely cemented.
So we're talking about this one right here?
Yeah, some rounder ones. So a little bit.
You know, Pilot, maybe there's a rounder one that's up ahead over the other side of this boulder that's in front of us. This one might be a little bit flat.
Flat is okay if they have depth to them. If they're really flat, it's probably just a slab of just the manganese crust.
Right.
Copy that. Yeah, if anything.
Yeah, so you're saying in this area.
Yeah, there's a couple round ones coming close to the laser, just to the right of the laser that might work.
Yeah, do you want to poke these two right here?
Yeah, right in there. One of those might work.
Copy that. So make sure I don't hit the sponge with my toe.
Yeah, you can see your manip got a ways.
So we're trying to grab something here because we're hoping as we get upslope after this, we're going to get into more consolidated substrate, and our chances of getting a rock are diminished. But
just like what it looks like in the background of this area. Yeah, that's kind of what we saw yesterday for most of the dive.
Yeah, we're sort of in a little bit of a flats area here where some things might have tumbled down, so we have a chance. Yeah, that one's worth a shot with the right laser. I think we're at an angle. It's a sort of a more elongate piece, and we're looking at one side of it.
Yeah,
But
I'm worried it might be stuck to the thing behind it.
Yeah, it might be. But
all of these things are about the same in that sense.
Yeah.
All right, Washleads, I'll give it a quick poke nonetheless.
You're still spun up.
Still spun up, great. Coming on.
It's moving.
It is moving.
It's nice size one. Some of it was buried too.
Yeah.
All right.
What do you think?
Phew, I was getting worried there.
Can you pick it up and squeeze it?
Absolutely.
Do you want to get your rock box open yet first, or?
Yeah, I guess we can open it real quick in case we sample. Do you want to bring wing inboard? Washleads, just in case we collect this, I'm gonna get my rock box ready.
Okay.
Ready for inboard?
Yeah, ready for inboard.
Here it comes. Let me know if you want any other view, or if you want me to tilt you up on this view or anything like that.
Oh, I'm not hearing the pilot. I gather you are, but yeah, I had it on earlier and I didn't hear it.
Washleads, do you hear me?
Yeah, I hear you now.
Okay. Sorry, I might have just been talking quietly.
The left-hand side of the screen is one of the boxes that are used to bring the rocks to the surface, and what he's doing right now is opening up the lid.
And this is actually quite interesting. If you think about this, he's opening up a box 2 kilometers from here.
Yeah.
Force.
That's right.
Yeah.
Yeah, without the cable, we'd be on a much longer delay too, probably.
I might have got a dead sponge.
It's not crumbling. That's a good sign.
That is.
Yeah, the group force 7, so that's pretty strong.
I mean, tilt up for the zoom and spin.
Yeah, I got it from here.
Okay.
Washleads, what do you think? Oh yeah, I got it.
Oh, you got a dead sponge there with it.
Yeah.
Oh.
Good job.
Do you want to zoom on
This
that
is
box?
not counting as a biological sample. It is
really?
not counting. No, it is not.
Are you going to call it a commensal
He's
again?
asking if you want any zoom.
I don't know what you would call this.
Yeah.
Not sure it was
I
actually.
think we'll just take it.
Take it?
Yeah.
All right. Video, do you want to do your zooms?
Yep.
All right, we'll use center.
Okay. That spiral pattern on the top right.
Not too many commensals on it, so looks like
Center
it'll
that?
save us a little work. Oh, by the way, I forgot to mention, particularly to Sako Matsumoto and Scott France, when we got the rock to the surface yesterday, we actually discovered that we had collected a couple of those little white things that Asako was interested in particular. And one of them was in really great shape. It was perfectly intact. And so we have preserved that, and I took a look at it under the scope, and I'm not really good at stylasterids, but it looked like a stylasterid to me, and I think that's what Sako thought it could be. It looked like it had small holes coming up the side of it. It was definitely hard. It was not soft.
Okay.
And
so
we've gotten one of those. We got one of those surreptitiously.
That's great news, Chris. And your description there does sound like a stylasterin. Just now when we were looking at the rock close up there, I saw a group of concentric circles, and I wonder if that's evidence of where a coral had previously been attached to the rock, possibly, and if the way the skeletons grow, they're put down in concentric layers, especially the black corals, so that's a possibility.
Could it have been from the dead sponge we grabbed? Given that it was sitting right next to it? No?
I don't think so. I think that sponge had fallen off from something else further upstairs and just came to rest near the rock.
Yeah, and I'm not sure a sponge would create concentric circles like that.
Okay,
I'm not
so
either. That's why I asked.
You want to get the...
and it's in. Or it's out. Stowed.
That one went into the port box?
Yes.
Okay, so maybe since we haven't really gone that far, maybe we can make a little distance here on our trek. And
copy that, wash sheets.
Great. But we've had a nice productive session here. What?
You guys thinking something like 070?
What's the matter? Oh, you got the wrong dive.
I put the wrong dive number in. We'll have to fix that.
Okay, got it.
070. I think I'd be fine with that.
I'm lined up at 06.
Are you saying distance or you're heading?
065.
Heading.
I think 065 is good. That matches all right with your bathy.
Yeah, you're looking at...
yeah, I don't know. That's when I point generally uphill. That's when I get. His sonar is frozen, so we'll restart him here.
Thank you.
It's unfortunate that's happening again.
You guys okay with that? 30 meters?
The sponge looks like it's a
looks
Tritoflora.
I'll call it.
I'm going to go ahead and record
Yeah,
that.
at least we've got a lot of room.
Actually being here.
Pretty
That's a
sure
glass
over now.
sponge that just went off
Closest
the
thing I've got is at least 40
Go ahead.
away.
Ready for a ship move, 30 meters,
40.
bearing 065, 0.2 knots.
We've got ship move range 30 meters, bearing 065 at 0.2 knots.
It's a good copy.
Lots of dead sponge skeletons here. So sometimes we sort of speculate there may have been some event that occurred that killed off a whole bunch of these sponges, but I'm starting to think that that's not the case, that really you're just seeing cycles of life that have occurred over a very, very long period of time, and they're just accumulating. They just persist down here in these little pockets for quite a while.
Wash sheets, any interest of what's coming ahead?
No, no interest.
Copy that. Just going to move around it.
It's sort of a dead sponge with some ophiuroids on it. It's pretty common when these sponges die, and if they haven't fallen over yet, then brittle stars seem to like to climb up on them and get off the bottom a bit.
It seems like the right water column for that, given all the stuff that's floating around.
little Chrysogorgid.
Nav
another
bridge,
dead sponge. So
movement is
again,
complete.
fair number of dead sponges at this site, just like we saw yesterday.
Ship move is initiated.
Move initiated. Thank you, Bridge.
Washleads, any interest on this right here?
No, again, I think if we can make some distance, that would be good, and then we can stop for some close-ups there. I'm a little concerned we're going to wind up burning our dime here before
Copy
really
that.
getting into the good stuff.
Ship is moving, so.
Great, thank you.
Port lower. I'm gonna
yeah,
stick it out a little more again.
Burning your dime? That doesn't really make sense, does it?
It's going to take some heat.
Yeah, it's going to take a lot of heat, no?
Okay. Probably not worth the dime.
No.
Set another one of those little.
Yeah, there's so
Maybe
many
a
sayings that if you think about them,
Chrysogorgid or
make no
something.
sense.
Some more of the, there's an enemy down there like we saw before. Yeah, I'm really interested to see if we'll get into another big thicket of big bamboo corals.
Yeah. Which way do you want to go?
Straight up, right on the...
Sure.
Straight up right along the ridge line. I think that's our best chance of spotting some things we can...
Hey, Washleads, as we're waiting for the ship to catch up, would this be a good time for us to do introductions?
Yeah, but before you do that, can you zoom this coral? This is something we have not seen on this cruise yet.
Yes.
So, and it just popped into view, and then we can do the
video.
intros.
Might be
probably one of those you want to set up for a great shot.
Small amount.
Yeah. Washleads, is this something you want really good shots of?
Got the time for it.
If we've got time, yeah. This is one of the. It's in the precious coral family, so it actually is worth, since we're here, and if the boat's.
Yeah, we're out on our leash, so we're waiting for the boat to catch up. So yeah.
Yeah, I was kind of hoping we would see one of these yesterday, and we did not. So I'm pretty happy to suddenly see this crop up out of nowhere. So Scott thinks it's a Paragorgia. I was sort of thinking Corallium, but Scott may be right on this. Now I'm getting another look at it.
I suggested that, Chris, because of that large astroschemid ophiuroid. They're pretty typical of the bubblegum corals, so that was my guess as we were coming in. I haven't had a close look at it yet.
We are getting a close look, and it is a hemicorallium, so it is in the corallidae. You'll see this in a second. But yeah, they're hard to tell until you really get a closer look. It's got one of these astroschematid.
Excellent.
So when we call it a precious coral, these are the types of corals that are actually commercially harvested, and they use these to make jewelry out of.
I'm going to wait till I get you a little more in fisheye, and then I'll spin around and get you. If I spin right now, I'm going to tug on you. I'm already tugging on you a little. That's why you're a little bouncy.
But it's related to the ones that are up around
giving you a little bit of leash
meters, which are harvested in some places of the world.
Maybe. Swing a little closer.
The issue that some of us biologists have with that is that
could move in a little.
A colony like this can be hundreds of years old.
Are we in a ship move right now?
We are.
Oh, okay, good.
5 meters left in the move.
All right.
They can be used for a variety of surface.
Tack on another 30.
A variety of analyses, including some of the corals can be used to analyze past climates conditions.
He's going to take a while on the shot.
So they're very valuable when they're alive, and perhaps
tail cam there, and I'm getting a little tethered, getting a little closer, so I'm going to swing in.
Yeah.
Fortunately, I fear I'm going by you because you're off.
Thank
to
you
the
very
side
much.
There. All right,
so yeah, maybe we could do some introductions to the front row.
065 was that move.
Yeah, so piloting right now is Sean Kennison, and then my co-pilot to my right.
You're not an XRV. Or OKX.
OKX.
Yeah.
So piloting right now is Sean Kennison. To my right is Jeff Lanning as co-pilot. And to my left is Levi Unema as my navigator. And in video, we have Roland Bryan. You can't quite see him. And I'm not quite sure who's on the back row. We have a Nick Palenko.
And Emily, you want to? Yep.
Ah, yeah.
Yep. I don't think she's...
You're being introduced. Sorry. Right. Emily Narrows, so she's doing the clipping in the back row.
Did I...
Oh,
Is that
we can
everyone?
actually wave at him.
Emily, and then... Okay. Yeah. I think that's everyone.
Emily's the only one that waved so far.
So
well, it goes without saying to make this all work.
Trying to get lined up on track again here. Yeah, bouncing around a little.
6'5", is that right?
But yeah.
So we're just responsible for the talking.
Yeah, we're just babble on as we go.
And it looks like the next move might be
we have to do besides
a little bit north of the last
people
one.
See and
right.
in the footage.
Yeah, like there, 060.
I
don't know if that's okay with Bathy or not, but that's what I got for
all these things that happen.
Sonar return.
Yeah.
Something along the 060 line.
Yeah, that's fine.
Okay. I'm ready when pilot is.
For the next move?
Yeah.
Yeah.
Okay.
Put it in there.
Yeah, just more dead sponges on the bottom.
There's a fish. Any interest on this?
Bridge, are we now?
Nice.
Go ahead. Ready for a ship move. 30 meters, bearing 060 degrees, 0.2 knots.
Oh, there he is.
Ship move bearing 30 meters, bearing 060 at 2/10 of a knot.
Oh, he's a pretty one. Look at this guy. A beautiful blue shrimp. I don't think I've seen anything like this. Whoa. Hello, who are you?
I'm not going to say Bob again because then Scott's just going to call in and tell me that it can't be a him.
Oh, I'm hoping that Mary Wickstein at Texas A&M, she's been one of our arthropod specialists who's helped us with identifying shrimps and crabs and things. Hopefully she will get an image of this, because honestly I have never seen a blue shrimp this deep, or even 1,000 meters above this.
Average.
It's
Ship move
very
initiated.
Unusual.
Move initiated. Thank you, Bridge.
Okay, and it looks like Scott doesn't know either whether it's a him or a her, so we can go with Bob for now.
Yeah.
Blue Bob.
Yeah, so... yeah, we're all...
It does have the same...
Go ahead, Scott.
Ship is
I
starting
was just going to say that it has the same very long pleopods, those swimming legs that you see that kick out to the side. I believe it's Plesiopenaeus that we see often, the red ones swimming like this. May have that genus name wrong, or they may have changed that genus, but we often see these
lasers clear.
Very well-developed swimming legs, and they have this kind of rowing pattern, much like some of the worms that sort of kick out and fold back. And that's a little bit unusual. Very often you see these shrimp swim, the pleopod swimming legs are held directly below the body and not out to the side like that. So these are very well adapted for swimming. As you can see, it's doing a really good job. But I've never seen one blue, so this is a first.
Yeah, and I would agree, Scott. It does look like it's in that family, but they're generally either sort of a brownish color or a red color. Blue, I've never even seen any pictures of one of these things that's blue. It's a really gorgeous animal.
Getting up into the back
I know there are some, unusual blue lobsters occasionally, and I don't remember what the physiology is that turns the carapace blue.
He's right between
but it would otherwise be, sort of a typical New England lobster. But
he's not right between
something has occurred such that the carapace turns blue. I don't know if that's the same situation here.
A light bar.
This is gone full wide.
Yeah, and another option is that the carapace is, I don't know. That's not very good. Did they? If I'm not mistaken, the shrimp could have hemocyanin for blood, which would have a blue color. So if the carapace was completely clear, you might have a blue tint to it. But it's that's sort of a uniform blue color. So I don't know.
Yeah, that's an interesting idea, except that the pleopods and the legs and so on were not blue. Maybe
Right.
just because they're so packed with muscle that it would obscure the hemocyanin, but that's certainly a good possibility.
Yeah.
Or something worth considering.
Right, and just as I said that, I'm not sure who's
Ship
at
is
the
about
ISA
halfway
Watch
through the
2.
move.
They were also asking whether the blue color could be from the blood.
Shrimp's in your aft cam. Hanging out behind you.
So you got something a little different sticking out here on the left.
Left.
Yeah, or in center now, excuse me. It's well, now it's a little bit more to the right. Yeah, that kind of thing on a stem. I wonder if we could take a quick look at that. It's right on, coming off the side of this little boulder here. Up a little bit.
Oh, I see it.
Yeah, there it is.
I will get over there.
So one thing that we haven't seen so far is a chrysogorgid called Metallogorgia, and this may or may not be one of these. And I don't think it is. Scott, I think it's the same thing we saw yesterday.
It's got that same wiggly stem again.
I agree with you, Chris. And Jasper, it's a Chrysogorgia, family Chrysogorgiidae, which translates to the gold corals. You can see the base of the main stem there had a kind of golden sheen to it. Jasper, it's a Chrysogorgia, family Chrysogorgiidae, which translates to the gold corals. You can see the base of the main stem there had a kind of golden sheen to it. That's the color of the skeleton below, the more pinkish purple tissue.
Do you want to come out? I can
has one of these chirostylid squat lobsters in it. The chirostylid refers to those very long arms, the pincers that they have. Lots of different species of those squat lobsters that live associated with corals, and you find very host-specific situations where certain species of that crab will live only with a certain type of coral.
We've seen this before. I wonder if we can go ahead and move on and still keep going up slope unless we need to stop. Thank you.
There's something different behind this next rock. Maybe just a quick zoom as you're passing it by.
Which one, Scott, are you referring to? I see about three things here.
It's just off the top right of the frame.
Top right?
Top right.
I think that was the direction you were going.
The more pinkish or the white
behind the boulder?
Not the lyrite one. It's almost on top. Yeah, I think it's pink. Maybe it's another hemicorallian. You can probably see better than I can.
Yeah, I think
just right of the middle.
Yeah, I think it is. I think it's the same coral.
Right. Okay.
Looks like ships in position, pilots.
You've got another one of these branched black corals that could be. You were suggesting it could be a lilipathes, I think, and that's off to the left a little bit.
Say it again, Capone.
I said I'm happy to keep dialing them in
Chris.
if you want
I
to.
had a look around at some of the different branching
I'll add 30 to that one.
of black
060.
corals and their depths and so on, and lilipathes seem like a good possibility. Nice whip-like bamboo coral off behind it, too.
Seconds here and then keep going.
Bridge RV now.
Alright, quick partial video.
Go ahead now.
Ready for another ship move, same as last. 30
yeah, this is the
meters,
same one. You're just seeing the back side of it in this case.
0.2 knots.
No, it's okay. We had a pretty good image of it earlier.
So the chat room,
Range 30,
Michael
bearing
Vecchione is
060.
also agreeing that that blue shrimp is
0.2 knots.
probably not from the blood.
It's a good copy, Bridge.
Yeah, so Michael Vecchione is a specialist in a lot of midwater animals, including shrimp. So Michael, you actually think it is a pigment in the shell itself that's given it that beautiful color. That makes it even more intriguing in my mind.
Ship move initiated.
Move
So
initiated.
I'm glad
Thank
we
you,
grabbed
Bridge.
your rock when we did.
Yep.
This would have been a little difficult here.
Yeah. It's time to
Ship
bring a
starting
rock
over
drill.
Give it a good push. There's not much here.
So there's a good chance that sometime in the past, and correct me if I'm wrong about this, Jasper, that this used to be more of a talus slope, and the manganese has created this entirely uniform surface here.
Yeah, the surface is covered and sort of completely coated everything together. The lumpiness of it, though, makes me think that either we're looking at a talus slope of cobbles, or it could just be that these are broken up pieces of pillow lavas and the like that got covered in a nice thick layer of manganese. This is the tricky part about finding rocks here. Is you can have an inch or more even of this manganese coating on the outside, and so you're never quite sure what's on the inside.
Got a dead sponge. It's got a bunch of crinoids all over it. A little bit of it's still alive, but not much.
Any interest?
No, I don't think so. We're gonna. And there's a little stock sponge, which could be a Calophacus down there to the left of the fan. I'm gonna go ahead and mark that in.
There's a bigger one on the left.
Yeah, they are Calophacus.
Any interest on a zoom?
Maybe a snap zoom, just
you
for.
Want this bigger one on
Yeah, the bigger one would be good.
Video quick snap zoom.
This is a different Calophacus than we saw yesterday. And this is, I think, called Calophacus subgenus Oxydiscus, but I'll double check that.
So as you're pondering that, Scott Franz points out he did a little research that the coloration on the blue shrimp we see it might be a genetic rare defect. At least that's what it is in lobsters.
So we were imaging a mutant?
Yeah, apparently.
Oh, man.
Mutant shrimp.
Mutant shrimp.
That's cool.
This sponge is actually probably in the subgenus Calodiscus. I always get them mixed up. So this particular genus, it's in the family Rosellidae. It's being worked on right now by one of the world's authorities on deepwater sponges, Dr. Henry Ricewig up in Vancouver. And a lot of them don't just have a genus name, but they also have a subgenus name. And so it's a matter of, he's trying to straighten all these out. Because right now it's pretty confusing. You have a Calophacus, you have Calophacus calodiscus, you have Calophacus oxydiscus, you have a whole bunch of things in it. There's another one of the craliums that we just took a look at. Hemicralium, that is. All right, so from the sound of that, was that a screwdriver or a wrench that hit the deck? Or a hammer?
He's still hammering, so I don't think it was the hammer. Yeah. Although pretty much everything is a hammer, right?
Yeah.
But that sounded light enough. I'm guessing a light pair of channel locks.
Another one of the Norella that we were looking at earlier, off to the right. And I'm still waiting for the big community to start.
So compared to yesterday, we do have a little bit more sediment
it's
on
like
the
starting to veer a little
rocky
further
bottom
north at this point.
Even on the solid rocky bottom, it looks like there's a light dusting of sediment. I think that was missing more yesterday.
Next move we might do more like 050 or 055, 050.
Copy.
This is straight up for me.
Looking up slope.
Yeah, 5550.
Yeah. Even 045 might work.
Yeah, whatever.
50 is a little closer to Bathy.
You know, this is a fine
Five
substrate
zeros
for corals and
just to the
sponges,
right
so
of
I'm
that.
not getting it.
You're not
Generally
happy with
there
it.
are
If I was a
returns, but
sponge, I would certainly consider
fine.
settling here.
Yeah.
It's
Oh,
just-
there's one. Right in the
yeah,
middle.
there's one. There's a few of them, but you know,
Going up
it's-
on 4-5, you can see what it looks
I
like.
don't know. Maybe there's a.
They're just
Here's some passes,
protesting this
045.
particular hillside.
Yeah, that looks good.
So you might have something up ahead, actually, to the upper. Could be a sea star. It might be worth taking a look at. Chris Ma, by the way, emailed me this morning,
Lasers should
and
be
that
on.
white sea
Yeah,
star we
okay.
saw yesterday, he
Starfish
confirmed it
hanging
was
off
a
a coral
pythonaster,
tip.
and was in
yeah,
fact really interested in the animal.
He's hanging off of a stalk there, I think.
Yeah, he is.
So ship's in position. Let me
So
know when
that
you want me to call
stalk
it
is
in
completely
at
bent
045.
over, probably due to the weight of that sea star. I think its base is all the way up top.
Yeah.
And he's basically finished this coral. So what is he doing? He's eating it, the darn guy. And he's almost completely cleaned off the entire branch except for a few polyps that he hasn't quite got to yet right underneath him. So he's been just working his way down, or excuse me, up along this coral.
At this point, it's down.
Yeah. So this coral is not long for this.
Interesting, because it.
Go ahead, Scott.
This must be a primnoid whip, like
tilt
maybe like
down.
a Candidella or something. That's not a bamboo coral. So those plates, the scales that we referred to earlier as covering the entire polyp, certainly aren't doing any good protecting against this predator.
Cool view on Sirius.
They're trying to do a zoom on it right now.
Yeah, I can just tell from the skeleton. That's a primnoid skeleton for sure.
Yeah, I agree, Scott. And I am trying to remember, trying to take a look at the Sea Star guide here to refresh my memory of which are the different species that are predators on corals. The guide I'm referring to...
Hipposterius for sure.
Yeah. Why I keep passing the asteroids up, but here we go. I finally got it. So also a Calliaster is a predator on corals. Hippiaster is a candidate.
Looking at you, 72.
Maybe Chris Ma is staying. I think he was going to try to participate today. So if we give him a few minutes, he might call in and straighten us all out here. There's also something called Circeaster.
Video's clear with it.
I don't think it's that.
All right.
Washington, are you okay if we move on?
Yes, that's fine.
Copy that.
Thank you.
All right, video, come on out, please.
Come up so you can push.
Yeah,
I'm lined up at 045 at this point.
I'll just center nicely in front of you.
I think it's a little large. I'm not so sure it's Hipposteria, Scott. I think it could be either that Circeaster, just looking at the pictures from last year. But Goniasterid, maybe. That's the family.
Yeah, I wasn't trying to make an idea. I don't know them well enough. I was just answering your question about what
Alright, you got
asteroids feed on corals.
Yeah, sure. So when I said I'm looking at pictures from last year, I'm going to put a plug out here for the NOAA Office of Ocean Exploration and Research's Deepwater Animal Guide. It's online. It's got 1,700 photos of deepwater animals that we imaged last year, and we're going to update that guide with the animals from this year and keep growing it. And if you are interested, those of you who are watching are interested in seeing some really beautiful imagery
move 30 meters, bearing
animals, you've got to check it
045
out.
degrees
You've
at a
got to
speed
go
of
to the
0.2 knots.
NOAA Okeanos Explorer website, and you will find that
Roger,
guide.
copy.
And then
Range
you
30,
can right-click
bearing
and
045,
grab them, any of the ones
speed,
that you want.
.2.
And they make marvelous desktops for computers, those images.
Or they can fill your iPad like it does on yours.
That's true.
In the background, they've definitely changed their tools. hopefully that doesn't come through too much.
Ship move initiated.
All right, video.
Move initiated. Thank you, Bridge.
A or B?
A or B? What?
Option A or option B?
Ah, that one.
Didn't know we had a optometrist.
What light did you shut off?
Oh, there's
Upper
some
swing
corals
arms.
over there to the right.
Oh, there's
We
a
got a
bunch of
little
them,
bunch of them here.
Is that the direction we want to go, Washington?
Let's see where we're at.
The direction.
Try
Sure.
to be aware. I'm
Maybe
going
let's
to push
go
a router change. It'll make your panels
cruise through
blink,
these.
but it should not change any
Hey,
sources.
I can even see some beyond that as well. So
Yeah, I can go that direction if you like.
yeah, let's go ahead and follow the corals because I can see some even in the background behind them. So
Yeah.
Copy
Yeah,
that.
all the way up on top here.
right, yeah, those are the ones I was talking about. These may be all corallium, which is pretty interesting. I'm not sure until we get up there. I think there's one sponge in there as well. Nope, they're not all corallium at all.
What was the last move put in that?
045. It's
045.
generally this direction, yeah.
Oh, we might be able to reach out.
This is good. You'll get there soon if you can't reach out now.
So corallium is, or hemicorallium is the one that's
Ship's
going off
underway
screen
and
to
in
the
that
left.
direction.
And then, yeah, we've got a bunch of
here.
other things coming up.
What was
Some more of these
fish?
primnoids.
Pretty good.
There's a, oh yeah, let's, if you could go ahead and zoom on the fish. And make some folks that know a fishery very happy.
Zoom on fish. It's still
There was
far
another one
away.
too, I think.
Okay, and we'll give Bruce Mundy at the NOAA Fishery Science Center a chance to get caught up. He's got a little bit of a delay.
You can continue to get close to it to give it more light.
And he should be seeing this fish any moment.
They really just swim around until they run into a rock, don't they?
Tell me if I pull you at all.
I think they're sort of blinded right now from the lights.
You're okay in a second, but yeah, I'll let you know.
Bruce is taking a long look at this one. He's not going to fire off a quick ID because I know he's on the telecom.
Yeah.
Bruce, the suspense is killing me.
Let's let him swim too so we can see
Got
the
to
dorsal
commit.
fin.
It's time to commit.
Oh, his fin starts nicely in the back.
Yeah.
Okay, now go back to the head.
Oh, he's not going to commit. All right, I'm going to throw out an ID. I'm going to say it's Synaptobranchus in that genus, and probably in Synaptobranchus oregoni or brevidorsalis. So a bunch of these are the
Yeah, hi,
hemicorallium.
Chris. This is Bruce.
Yeah.
Go ahead.
Almost gave up on you, Bruce. Go ahead.
Yeah, so I don't know if they were able to zoom in to see if they could see the scales on this thing. That's a way to tell those two species apart. But you're right, it's Synaptobranchus and either brevidorsalis or
Yeah,
oregoni.
I think we're good.
I think I need to leave the room more often so we see more fish.
So I see. But you came back in right in the nick of time, though, didn't you?
Yeah, I did. Yeah, and thanks to Heidi Hirsch for kind of filling in here when I stepped out.
So thank you, Heidi, for not inhibiting the fish occurrences like Bruce apparently does.
Awesome.
Just a little bit of trivia about the names. Brevidorsalis means short dorsal fin, which is a characteristic of that species. Oregoni was named after what's now the state of Oregon. But the species Synaptobranchus oregoni actually doesn't occur off of
Yeah, I'm
Oregon.
starting to catch up,
The
so
fish was named
gigantic
after
figure
a
out
research
what's
vessel,
important and get
the research vessel Oregon, named after Oregon Inlet in the Carolinas. which in turn was named after a
Ship
ship
just has a couple more
that
meters
was
left
named
to
after the
move.
territory of Oregon.
You
So
can
it's
see
a
a really
roundabout
large one upper left
origin
off
for that
screen.
species name.
Left. Is it up there?
That's interesting.
Sorry, that's in
Wow,
Oregon.
we've got some really nice corals. Thank you, Bruce. We've got the corals are definitely picking up, which is why I always like to get upslope a bit on these dives, because we often see this where it's
The lasers still
very
on.
quickly we can get a complete change in the community.
Yes.
So this is a beautiful big example of that I-4 clad icidid, and this time hopefully Scott won't correct me on this, which he did earlier because I misidentified it. I think I've got
Actually, I was correcting myself, Chris. I wasn't correcting you, so you have ID'd it
If
correctly.
we do image
Oh,
it, the
this one,
best view
yeah,
would
but
be
that other one
on
I
the
totally
opposite
blew.
side,
Wasn't even the right
down,
family,
looking
and
up
you were.
into the background.
Yeah,
Oh, I see.
yeah, that earlier one, earlier in the dive.
If they want to image it.
Copy that.
Yep.
Yeah, Washington. Any of these?
Yeah, this is a really big one, and we can surmise from that it is really quite old, many decades.
Let's mow right through these. We've imaged all of these things so far already, and see if we can find some new things here.
You know, we've been referring to these whips as bamboo whips, but the one we zoomed in on with the Sea Star was a primnoid, so I wonder if they're all primnoid whips that we've been seeing.
Yeah, we could.
Certainly the smooth curve of the one down there to
Ship
the
is in
right makes
position,
it look more
pilots.
like a primnoid than an icidid.
Yeah, I think that one was too, Scott. Maybe it could have been a Candidella, but that one we saw earlier was not Candidella. I think is probably more likely to be a Norella. I think you may have mentioned that as well. So you could have some of these unbranched Norellas here that we saw.
I didn't know there were unbranched fellows.
Yeah, the Norella hawaiiensis, I think the consensus was that, I think that was the final ID for that unbranched one we saw last year in the Northwest Hawaiian Islands. Steve took a look at the specimens that we collected, but let me check the gallery again to make sure that was the final say on that one.
Yeah, I seem to remember there was some controversy about the specimen that was collected and whether it was a fragment or not, etc.
It's another tunicate center screen right, going off edge of the screen. If they will. We saw one before. I just want to point it out.
Yeah,
I
so
think on the right side we're passing one of the baby versions of that candelabra I-4 clade. It only has 3 or 5
Just lost.
branches.
Oh, Pilot,
minute.
there's a bright red one in the center there. We have not documented this family today.
Right,
and that would be great.
Is it,
it's dead center right now, right above the lasers.
Above? Copy that. I'm going to get closer.
So Scott, yeah, we this is with Steve Cairns helping us out. Steve Cairns is at the Smithsonian, and he's quite an expert on primnoid corals and other things. Anyway, he took a look at the specimens and concluded it was most likely Norella hawaiiensis, that unbranched one that we saw last year.
You would know.
Another one of those carnivorous
starboard
tunicates
camera
that we
just came
imaged
back.
earlier
It
in the
was
dive.
very dark.
The octonemid.
What?
This might be what we were calling also
Your
Lillipad.
starboard
Here in the
nip, which is
foreground,
in your
and
left
Tina is,
monitor.
in the event
gone.
log now, so
Right,
she
I
might
called
be
it
able
up
to
on
give
my
us a better
router
idea.
here, and I saw it come
terrific.
into view.
Oh, I was turning it on and off.
Welcome,
Okay.
To see if I can get the monitor
emailed
back
me and
there.
said she was
I think.
hoping
So
to
you're saying subsea it's
participate
okay,
today.
but on that monitor
So
it's not.
Tina Molodtsova is who we're
So,
referring to.
oh,
She's
there it goes.
with the Shirshov Institute
I'm gonna write it down
of
anyways.
Oceanology in Moscow, and she's traveling right now. So she's, challenged to
Tuning in from away.
Tuning in, but she is one of the experts on a lot of corals and cereanthids and mushroom corals and black corals and all types of things. So we're always delighted to see that she's participating on the dive. So Scott has identified the one we're looking at right now as a Paragorgia or bubblegum coral, and that is the first one that we've seen. And first one in the monument, actually, too, because we didn't see any of these yesterday. So it's always great.
Anything new we see today is going to be new in the monument.
Date?
Yeah.
The second.
It's got a brittle star on it. I think it's in the family Astroschiumatidae, but we tend to default to that type when we don't see the spines or other features of some of the other brittle stars.
Look out for full specimen.
Copy that.
Tilt down.
Tilting down.
Those little red dots that you're seeing between the larger polyps, which are the much larger warts, are a different kind of polyp called a siphonozoid. Much smaller, specialized. They don't have any tentacles.
And as we pull out here, there was a black coral to the left that Scott had pointed out, and we've got another black coral expert on right now. It might be good to do another zoom of this branched one that we saw earlier. And
when you're ready.
And it's going to be sort of a brownish orange one. And let's see, where did it go? Oh, no. Is
it not in this area?
Let's see. Maybe pan just a little bit more to the left. I don't see it. So it could even be behind the corallium that's right in the center right now.
Yeah, I think it's behind the hemicorallium.
Oh yeah, there it is. It was hiding. It'd be useful to get a zoom at this thing now that we've got another specialist in this group. See if we can narrow down the ID. While I zoom in, Scott, this whip that we're seeing right now is definitely a icidid, an unbranched icidid. So you were talking about the other whips, I think, that will.
No, that's not a whip, Chris. You're seeing the lyrate on edge.
Oh, okay. Got it. That's right. There was that lyrate.
Much smaller lyre.
Yeah.
You guys ready for a move?
So Tina is going Trissopathes. Excellent. Thank you very much. We had 3 candidates for that one, I think, and Trissopathes was one of the ones that we considered. So
there's a Polychaete worm on one of the branches as well. You can see the little short white thing on the branch.
Full specimen?
Probably a little scale worm. You know, Chris, you noted that you thought you were looking at a bamboo coral whip there.
Video is clear.
And one of the things that's interesting is if you look as we're passing by it again, at the way that branches at the top, before it starts branching, it does look like a whip. So one could easily misidentify the type of bamboo coral that has that branching at the top if you encounter it when it's younger. It's one of the dangers of these identifying from video and photos when you don't know the age of what it is you're looking at.
Yeah, that's a really good point. That we don't know when that branching occurs, but presumably it's going to be unbranched for a while before it starts producing those upper branches right at the end there. So you're right. It's definitely a danger to go too far on your IDs.
Another thing is, the orientation of the camera. So that certainly looked like a whip from the orientation we were in, but now you can see that it's not. And so that's another difficulty if you're looking at static images, and it's one of the reasons why these videos are so nice, and we can sort of move around something and interpret it in a different way.
So we've got this white colony that's right in front, slightly to the left. This looks like it's a bit different than anything we've seen. Maybe we could do a zoom on this thing. I don't know what it is right now, but it's waving around, so it's not stiff, and that's not a hemicorallium then.
Copy that, Washington. Yeah, I'm just gonna.
I think it's going to be a bamboo coral, but I'm not positive.
Alright. Video, quick zoom.
Yeah. I don't know if that's a bamboo. I don't think so.
Yeah, I think I can already see nodes at the base.
You can? So it was right? I was starting
I thought I could. before we had zoomed in this close, I thought I could.
It's now looking to me like a planar chrysogorgid, Scott, but
Yeah, you could be right there. Yeah, I think you're right.
because I thought there was a node 2 at the base, but the polyps look wrong here.
Tilt all the way down.
More like a chrysogorgid.
Yep. Agreed.
So the only planar, sort of larger planar chrysogorgid that I'm aware of is this Chrysogorgia crises, but probably isn't that. It doesn't. Well, it may be that. I'm not sure.
Washley, do you want any more zoom on that?
No, I think we're good on that one.
Copy that. Copilot, I'm just going to get back in the center of your view.
Copy that. Yeah, I'm at 045. And
This is a nice little community down here. You got a lot of different things. And we can see stuff upslope too. Plenty of stuff. So I think we've gotten into the good part of this part of this ridge.
We're not moving, right?
ship stopped.
Ship stopped. You want to put a move on?
Yeah, ship stopped.
What's Bathy say?
Bathy says
070 to waypoint, final waypoint. But
yeah, but
uphill
is also 070 here. But I think you should just consider his screen and look uphill and see. Using your sonar, find uphill.
That's pretty good. 4-5. Follows the corals.
Chris, can we get a snap zoom on the?
I would think
the large branching spindly ones lower right.
More
okay.
like,
no, we haven't passed it.
north
It's
or
right in
something
front
in
of
a
us.
second. Maybe we fall off the ridge.
It's just to the right of the lasers. I don't know if you copied that, pilot.
Lower right.
Yeah, just keep. It's about right of the laser. We're about to go over it. Yeah, straight up. That one right there. The lasers are
behind.
It's got these little long branches.
I see behind another whip being devoured by a sea star.
Yeah.
Yeah, Copilot. I think
this is going to be one of those very long Morella, possibly
edge of the ridge.
Okay, so we will say.
Trying to think of the one that's got the really long branches. The
Get
Norella.
back onto it.
Okay.
If that's all right.
Yeah, sounds great.
So we'll do like a 035 or a 030 or something to try to get you lined up.
Sounds good.
Okay.
Yeah, Bauer's Eye. Norella Bauer's Eye has. It's the one to the right, not the pink one.
Oh, it's not this one. Oh,
yeah,
sorry.
this one right here.
I will rotate.
It definitely looks like a Norella to me, Scott. The polyps look like they're facing down.
You want another 30 meters in that direction?
That'd be good for me, yeah.
Yeah, kind of works.
I'll turn to 035. Is that what you're going to call in?
030, I got it, if that's all right.
030, yes.
035 if you prefer.
030 is good.
Okay.
Yeah, I would agree. I'm not sure about Bauer's Eye. I don't remember
Bridge,
what
ROV
that
nav.
species looks like.
Well, Bauer's Eye has got these really long
ship
branches
move,
because they grow quite a bit before they split and branch.
30
so
meters.
That's just one of the characteristics
Bearing
of Bauer's Eye. That's why I was
030
thinking
degrees.
it could be that.
At 2/10
I'm
of a
going to
knot.
step out for a moment here. Jasper is going to
Roger that.
take over,
Copy, range
so I'll grab a
30,
little bit of
bearing
lunch and I'll be back in a moment.
030, speed decimal 2.
As we're looking in on this zoom
Good
on
copy,
this
Bridge.
Norella, I'll point out that we've been saying the polyps pointing downward are one clue.
Pilot,
but a
I'm
better
lined up
one
on
is
030.
the way that the scale-like sclerites are
You were
molded
nicely in
together,
the center.
such that it looks like
All
you're
is well.
seeing almost two shelves on the back of each of these polyps. And the number and fusion and shape of these scale-like sclerites are what are used to describe and identify the various genera and species of these primnoid octocorals.
I'm not sure how long the science wants to stay with it.
Copy that. Washington, are you good on this?
I think so. Let me make sure. Scott, you've seen enough too?
Nav, ship move initiated.
Yes, that's good.
Yeah.
Take a bridge.
Baby sea star there.
Oh, just a little sea star at the base, yeah.
Sorry,
All right.
All good?
Not hearing a lot of enthusiasm about that. I think
All
we'll
right.
start zooming out.
Let's get out of here, video. All right, so the move was 4-5, is that correct?
3-0.
3-0. All right. Just gonna hop over these fellas.
Trigger another router change.
Copy that video.
Trying to fix a salvo that I have over here.
All right, I'm going to need a little help from the chat room or those on the conference line on which animals you'd like to focus on.
Ship's underway.
I'll do my best to guide you, Jasper.
Underway eye.
Thanks. Yeah, we're pointing out in the chat room there's a fair number of dead sponges on the right of the screen. They're sort of filling up the little pocket between the rocks. And then
It's interesting, Jasper, that we were just in an area that had lots of these different kinds of corals, and now
yeah, now it's almost barren.
like more dead sponge and not much else.
Yeah, it looks like towards the left of the screen I saw a few. Oh, there's another one of those. Is that one of those pink hemicoralliums? It's coming into view on the from the right.
Yep, now I see it.
About halfway
that
through.
Might be another one of the bubblegum corals, the Paragorgia. It looks like it's.
How's
I
that
see.
looking
And
for
there's the
getting us up on the
hemicorallium.
ridge?
So the hemicorallium is
Seem to
more
be
pink
working
one,
out.
and the
Yeah,
Paragorgia is the
looks good.
deeper
It's
red
following
one.
the corals
Right.
too.
All right, cool.
And then that bottle brush-like thing that's whitish, that's a Chrysogorgia.
Mr. Nav.
What's that?
What direction?
We are currently doing 030.
Looks like some tall sponges
Hoping
in the
to
back.
Yeah.
reacquire the top of the ridge and then
Yeah.
turn back to 045 or something similar to that.
Yeah,
I think we're trying to catch up with
I
there.
think we've sort of found it
dive
here.
track
so
a little
Sears
bit.
sort of slides in as
Been
well.
wandering off a little.
Yeah. Cool.
Looks like the
Any
corals
luck?
may be aligned along maybe the local high point on
Yeah.
the little feature we're on, and then as you drop off to either side, there's fewer of them
It's a
maybe.
big guy, isn't it?
I think looking at the little particles in the water, it looks like most of it's coming from the left of our screen, which is the northwest. Let me ask the pilot.
Yes, Washley. What was the question?
So the current seems to be coming from the port side.
Yeah, that's correct.
Yeah.
It's picked up in the last 30 minutes.
I turned my head to 4-5.
Nice.
So yes, I think maybe that's what it is. They're all sort of towards the left of our screen because they're trying to catch what's coming from the left over the side of this ridge.
Recenter. That's over here. Yes, Washley current's almost coming from due north.
Yeah. From the north, yeah. Maybe we can sort of hug the edge here a little bit more, maybe.
Yeah, I just want to.
That's right. Yeah, this path looks like there's a fair number of things in our near future.
Yeah.
looks like
Yeah, what do you got?
our
6-0 almost?
Let's say
just about done with that move, so we can start lining up for the next. See where we are.
Probably thinking 6-0, 6-5?
Let's see what we look up. Ooh, I want to say up is almost 6-0. Yeah. I think it's 6-0.
Is there any interest on the conference line to zoom in on any of these branched corals?
So far, I think most
think
of
I
these
fixed
are
the
what
side
we've been
town on this
seeing.
one.
Yeah.
Maybe on the far left,
Yeah, it was running a
that
little low.
would be a new
At negative 20.
primnode that we can see
It's all
from
the way
here. It's a
down.
primnode coral.
So I
but,
put it at 0 so we can
it
hear
might
again.
be different for
How's
the
that
day. It
happen?
looks like possibly a
don't know.
Calyptophora. Got that sort of swooping lower branch. It's off the screen now. But
Yeah, when it
My
was
second-
off my screen already when you started talking.
I'm right at like 6-0 there.
Sure,
Something.
I understand. Yeah, it's hard with the
5,
delay. Isn't
That's
it?
right.
7, and that's pretty much up. Yeah, okay. So 0. What do you want?
6-0 is fine.
I think
So
we kind
6-0.
of.
Okay.
Washington, did we pass something up?
Maybe try and go towards the left a little bit and up.
Left and up.
Well, I think you have no other option than up.
Yeah. Very true.
Another 30 meters, good.
Yeah.
That's super.
So Scott, I think it's coming into view right now. On the edge.
Oh, to see the one
Bridge, ROV nav.
edge. Of the,
Yeah, that's the
Nav,
one
this
I
is
was
the bridge.
referring to.
Another ship move, please.
It's another one, I think.
Gonna do 30 meters,
Any
bearing
interest on these
060 at 2/10 of a knot.
Is this worth the zoom? The one that's sort of a little bit above the middle.
Roger that. Good copy. I got 030.
Yeah, I think
range
just
30.
a quick zoom. We don't have to
Bearing
spend a lot of time,
060.
but,
is it,
Speed,
a quick look at the
decimal 2.
bulbs would help.
is it this
Good
one?
copy, Bridge. Thank you.
It's to the top right of your where your lasers would be. Yeah,
Copy that.
that's right.
Alright, video quick zoom.
So this
Nav,
has the polyps
ship
and the
move,
little rows
initiated.
again, sort of around.
Thank you, Bridge.
Those rings
Yeah.
called whorls. Yep. So that's great. That's a good view. Thank you.
Video
Give it
on
full
out.
screen.
Yeah. Full screen on the center.
And lose the lasers.
Copilot, please.
Lasers
lose
coming off.
lasers.
Lasers off.
That's good for us when it's good for you, Jasper.
All right. Yeah, they're just taking a
Yeah, we're just
image
getting
of the entire animal without the lasers.
Great.
Alright, video on you.
Go ahead.
Oh, good?
Yep.
Alright.
Move underway.
Thank you, Nav.
Top right corner is a different coral.
Yeah. Is that a coral or is that an anemone?
Oh, yeah. I will go explore.
Mushroom coral.
It's also one of the diagonal up left of that similar species, but different.
Any interest, Washwood?
I'm hearing nothing from the folks on the chat right now.
Good enough for
Sorry. Sorry, Jasper. I was typing. This is the first we've seen. I see two of them. This is a coral. This is an Octa Coral.
Would you like to look at it a little bit more?
Tina will give us a better identification. She's an expert in this group. And I think we may have two different species here. This light orange one that's on the lower left, and then there's a deeper red one. Yeah, so Tina's saying Anthomastus, genus, possibly species,
quick zoom.
Tinodes.
Any interest on good footage of this, Washwood?
Is this good for everyone, or does anybody want anything? more to-
Yeah,
Might
she-
want to take a shot.
This is good. She's got an identification there,
Okay.
so that's great.
So you want to look at the other one too?
A few of the polyps are retracted, which suggests they may have
You want some
eaten
lasers
something.
to get the size of the next one?
Sure.
They have
Turn on.
captured something and eaten it.
Right.
Lasers on.
This is on. And then next one is where, Wash?
Darker red up left.
To your left.
The red
and
one
up
that
a little.
to the,
It's the red,
yeah,
more
that
red
one
one.
may be different.
It's also a different anemone in the background, just diagonally upright from this red coral.
Copy that. It's
Yeah, that's the one he's talking about right there.
right there.
Yep.
Alright. Alright, video, come on in. Let's see the video.
So this is another Anthomastus. It's a kind of Octa Coral, but it doesn't have a skeleton like all these other ones that we've been looking at that are more fan-shaped or whip-shaped. This one's much softer.
All right, so we've probably seen this one.
Oh, good, Washley?
Yeah, I think so. Does anybody want to, since you're still zoomed in, do you think you can look up a little at that anemone?
Look up.
Yeah, I think it was up and to your right a little bit.
In the sand, too.
In the sand.
Just to note, Tina's identified this one as Pseudoanthomastus, so this is a different genus from
right
the
there, yeah.
orange one we were just looking at earlier. So
So it's got a barnacle next
good
to it
record of the diversity there.
Yeah, currents.
with
Load
its
real quick.
with its
Center.
This is
barnacle currently has its feelers out.
Alright, quick zoom video. Actually, rotate.
we just zoomed over to this guy.
Yeah, the anemone, that may be like the one that we saw earlier that we thought might be a tube anemone, but it turned out it wasn't. And then just to the left of the anemone is bamboo coral whip, and there's a little barnacle on the rock, stucked barnacle on the rock to the right of the anemone.
Actually, video, come on
That
out.
white thing.
I need to get better footing for that. Well, actually, I need to get better footing to get a better zoom. Do you want
Barnacle's
to do
actually
that, or
interesting.
move
It's a sessile
Do
crustacean.
you want any more footing? All right.
Did you get a close up of the red mushroom?
yeah.
Oh, okay,
Yeah,
then
that
that's
one,
probably good enough.
on? Okay.
Unless you want to, Scott, did you want to zoom on that bamboo whip?
Just very quickly, if we're in position, because this is something we have not
It's
seen
going to
yet.
be pretty quick, pilot. I got you
Alright,
at,
It looked like it
Okay.
had
5.5.
at least 2 associates on it.
you got a minute, but
But if we've already moved, Chris, that's okay.
No, they're doing it right now.
Just
Okay.
a few seconds while she...
Yeah, they're just going to give you a few seconds of it here, but polyps look like they're pretty much coming off of one side, Scott. And
It's a good current.
It's interesting that it had a Uropticus on it. That's what I noticed. There was something on there.
Alright, video.
Oh, there we
Yeah.
go. Yep. Great. And it's got an uneven distribution of intertentacular needles, so that will also be informative. Great.
you want to hazard a guess at this point, or, I mean, it's not going to be something like Bathagorgia, would it, or?
Yeah, I not right this second. I don't want to hazard a guess, but I can do a little look. But I am going to note that there were 2 chirostylid crabs on that skeleton. On that colony.
Okay.
It's just about done. So if you guys want to figure out the next general direction we want to go, see some blips off to
Right,
the
we have a
north
yellow
there.
colony coming up here on the right, and 2 of them. And I wonder if we
Copy
could
that,
do a
Max.
quick zoom on those, unless you took pictures of those while I was eating. These are different.
we'll let him push out.
I
then
have an
we'll
idea
see
of what
we
I
got.
think they are, but
Film these.
I also see another type of Anthomastus up above it. One of the very long
Yeah, we had
stem
seen
ones.
2 there just before, Chris. I don't know if Jasper relayed that information, but we saw a Pseudoanthomastus and an Anthomastus tahanodes.
Okay.
Do you still want zooms on these?
Yeah, just the
pinkish one
closest
and then the
one.
red one.
The one in the back is an overgrown Paragorgia with zoanthids, but this one in front is a little curious.
Alright, video on you. Come on in.
You're thinking probably a Paramerciid?
Yes, that's right, Scott. That's exactly what I was thinking. Or at the very least, you know, in the...
I'm thinking too.
Okay, and that's the first one of
With
those.
characteristic ophiuroid there.
Got you at 82 degrees.
82, copy that.
Almost time to push that.
Copy that.
So a lot of variety. I mean, we just keep seeing different things.
Yes.
It's a pretty neat spot here.
Video.
Yeah, beautiful. Thank you.
Chris, nobody can accuse you of lingering over lunch.
I eat pretty fast. That's true. It's a pretty nice spot we're in right now. I did not want to. Oh, look at that! We got a bunch of stuff in the background. I guess we've seen these already. These are the Slyrate Norella or whatever it is. And yes, we've seen this one before. Zoomed it. There's an anemone. I'm not sure if we've documented these anemones today, this larger one.
Just looking at one next to the Anthomastus, it looked a lot like the one that we had seen earlier in the dive that we thought at first might be a tube anemone.
Right, this one we're coming up on now is...
Well, not this morphology.
Yeah, this is going to be a different one. Yeah, let's get a quick zoom on this one. Gets... So good size. It's over 10 centimeters in diameter, probably about 15. So I think this could be in the family Exosalactidae. I think that's how you pronounce it. These are really, really difficult to identify from imagery. A lot of anemones are identified to genus and to species by collecting them and opening them up, doing some histology on the mesenteries, the little divisions inside of their bodies, the septa, and so
for full specimen?
Trying to go too far on some of these things is not easy at all. I know that Daphne Fountain for the University of Kansas was one of the world's authorities on anemones for quite a few years. She recently retired, if I'm not mistaken, and I showed her a lot of photographs that we had of deep water anemones there, and some she was actually able to go to family and even to genus, and then there are other ones she could only go to family, and she's the authority. So it's a difficult group, and they're hard to collect in any good shape as well.
Yep. All right.
There's a long-legged shrimp, the Metocarcinus.
Most of the identification of the anemones comes from a microscopic examination of the internal walls of the gut and histology, and so it's complicated and not easy to identify from a gross image, even if the whole animal's in your hands until you get it sliced up and put under the microscope.
Yeah, I agree. One of the things that helps us here in deep water, there are not tons of known deep water anemones, so we don't have quite a pool of candidates as some of the shallow water researchers do if they're in some different location that hasn't been well studied.
Sure, and the other benefit we'll have is with more imaging like this, and in future as we do get collections, then we can cross-reference back to these images and hopefully be able to make better identifications. We'll always have, of course, this imagery data available, so we can, as we learn more and are better able to identify things after we've made the collections, we can come back and look at the imagery and learn more about the diversity and distribution, biogeography, etc.
Yeah, absolutely.
Next ship move.
So I've noticed that Nicole Morgan has joined us in the chat room. Hello, Nicole. I'm glad that you're joining us today.
What do you see?
I don't know if we've mentioned who's in there for a while, but we've got Asako Matsumoto from Chiba Institute in Japan, Bruce Mundy at the NOAA Fisheries Science Center on Ford Island in Honolulu, Charles Wall with the Marine Protected Areas Division.
Copy that, 4-5.
We've got Michael Vecchione of the Smithsonian, Nicole Morgan. I'm trying to remember.
Got it, 045.
I'm trying to remember where you're from, Nicole. I apologize at the moment. Scott France from the University of Louisiana, Lafayette, and Tino Molodtsova from the Shirshov Institute of Oceanology in Moscow. So we've got a really nice group of experts
yeah, we are
helping us out with this dive today, because we're clearly seeing a lot of animals and a lot of different animals.
Go ahead, Nav.
So it's
another ship move, please.
It's very nice to have
30 meters.
Quite a bunch of people in chat.
Bearing 045
and also,
degrees,
after the dive is completed, then I'll typically get some emails the next morning where some of these photographs get passed around, and people who aren't watching live will then weigh in on some images that are getting passed around between scientists, or they're monitoring the Facebook site.
Nav bridge move initiated.
And they'll come up with a next day identification, which is super helpful.
That's just me pointing it out. I'm not calling the shot.
Yeah, Washid, any interest on anything here?
No, not yet. I think that's an ophiuroid, but everything else I think we've got an image of right at the moment.
Copy that.
There's a bunch of stuff in the distance that's coming up. So I don't know if you folks can see this yet, those of you who are watching, but if you'll notice, all of the corals are oriented in more or less the same direction. We're all sort of looking at them perpendicular to their main planes, and we see this a lot. And it turns out that these corals, well, they're going to orient to expose as much surface to the current as possible. And they're actually facing off the side of the ridge. And this makes sense because we believe that the current is perhaps flowing over from one side of the ridge to the other side of the ridge. And that's why these corals are here. They're getting some current acceleration. And they're going to go ahead and presumably orient in a manner that allows them to filter the water optimally.
You should have enough leash to get there.
Get to this lump
bridge, ROV nav.
Go ahead, Nav.
Just want to discuss our bailout here. I have
almost boulder-like
currently set at
formation.
315, so what you have.
Yeah, that's good copy.
I think I want to adjust that to
like it's more sediment.
southwest. So what's that, 225?
Turned out to be sediment.
Yeah, let's adjust to 225, please.
Good copy, 225.
Was he an evil starfish or a good starfish? Was he eating a
Turned
coral?
out to be sediment.
Oh, a sediment. Oh, okay. A sediment shape, okay. I don't know if you could get to it, but behind the pink one on the upper left, there's a wavering white thick branched one, and I think that could be different. I don't know if we could.
Is it way back there?
It's just behind that pink one that's coming above the lasers right now.
Behind the one you're
just on the other side of it. It's hidden by the pink colony.
If I can't get it now, that's the direction we're moving, so I can get it in a little
That's what that
immediately
planar Chrysogorgia looked like earlier from a distance. So this looks more fuzzy, doesn't it?
Yeah, it looks more fuzzy. That's why I'm wondering if it could be some species of white paragorgia.
This one lasers around right now?
That's it, correct. That's the one. I don't think we've imaged this before.
Could be, but I don't think paragorgias move quite that much.
Yeah, well then maybe it is this Chrysogorgia, this planar one.
Yeah, it's too fuzzy. Too fuzzy. Maybe it's a white black coral.
Try to get stable.
Come on, let somebody else guess.
Yeah, well the fun part is guessing from a distance, and then sometimes you're right and sometimes you're wrong, but you get better. I know what it is. I think I got it, Scott. I'm going to let you take your guess.
You got to give me 12 seconds.
Okay, I will.
Partial video. I drifted off that rock.
Partial.
Just want to make sure I don't hit anything.
Oh, it's another. Oh, it's an Acanthogorgia.
Acanthogorgia, yep. I was saying that's the first one we documented.
Very, very tall polyps. Yeah, that's good. So this is in the classic sea fan group. This is a Gorgonian sea fan family, Acanthogorgiidae. It's a very complicated family to identify. There are lots and lots of species in the genus Acanthogorgia, but there are a few other genera as well. But probably right now, given our understanding of the taxonomy, we'd call this Acanthogorgia.
Washington, do you want better imaging of that? I might have to get behind it.
Maybe a little primnoid whip drifting behind it.
Yeah, there was
corals, Tristopathes.
Video switching out.
Perhaps we could get a little closer look, but there's something behind it that is really interesting. It's that single stalk that's wavering right now, directly behind it, and this is really different unless the focus was off. I was trying to figure out what it was.
I thought it was a primnoid, Chris. I thought it was a primnoid, but there was very wide spacing between the whorls.
Yeah, and it's wavering an awful lot. Maybe it was a primnoid. I'm not sure.
Maybe it's coming
it's
back.
It's a pretty scene here, just looking at these boulders with the corals on them.
There must be a good current coming from the back side of that rock to be moving this stuff so much.
Yeah. So, Pilot, I think Scott's correct. I think it was a primnoid, and I just didn't really notice it.
Copy that. Do you still want footage?
Let's see. I think we're good. I think we could go ahead and move on.
Okay.
Thank you.
Yeah, now I think next move is going to be something a little more east. What do you think, Pilot? What's the next move looking like to you?
Turn around.
Chris, any idea what all the white spots are on the rock ahead?
No, but maybe we could take a peek at that. It could be barnacles. Pilot, if we could.
All over on the left as well.
Yeah, or the left. I meant on the left. Yeah, maybe we could take a zoom on these things.
There may have been a Metallogorgia off to the right as we started to go forward, so that might signal we're coming into a Metallogorgia zone.
And this yellow crinoid, by the way, it looks to me like it's in a group called Glyptometra. And the one that we've got in Hawaii is Glyptometra lateralis. It's sort of a pretty large yellow
white items.
One that sort of likes rocks, sometimes sponges.
Pilot, I'm going to turn my head to find the local hive if it's okay with you.
So that's
yeah, if you feel safe.
That's a partial zoom.
Yeah, boy. Okay.
Let's try to center out
a little sponge critters or something.
All right, video, do you want to come in a little more?
All right.
I'll try to center as you come in.
I think they're little sponges, Scott.
I think so too, Chris. Of course, now we have that old problem. Are these baby sponges, or is this just a species of very small sponge?
Yeah, no idea.
Yeah, come on out, video.
And if they are very young sponges, it means there's been a pretty widespread recent recruitment here.
Thank you.
There's lots of them.
All right, pilot, I adjusted my head to 080.
That was definitely worthwhile taking a look there, because I would have just wrote them off as barnacles. That's typically what we find when we zoom in on these
uphill
white spots on rocks, or sometimes tunicates too, I guess. But
Let's see.
To stay on this ridge, maybe.
so I think it's the same wavy branch Chrysogorgia, Scott, that instead of we got a fish here. I wonder if we could. Nah, he bolted.
Yeah, sorry, Washley. He's gone, I think.
Yeah, he's one of these little speedsters, tiny little speedsters.
I think next move could be.
I agree, Chris.
6-0?
Chrysogorgia, not
What do you
Metallogorgia.
guys think?
I think we're lined up more for 8-0, but if you think 6, we can go that way.
Cut the middle.
That's just what I'm seeing.
Go to 7.
Yeah, we could do 7-5. How about that?
Okay.
Line it up.
Yeah, Chris, this is Bruce. That fish went by really fast, but it looked like something mesopelagic, like maybe a myctophid from the way it was swimming, but it was really impossible to tell.
Yeah, thanks, Bruce. Yeah, that's what I was sort of thinking. But do myctophids get down to 2,000 meters? That's just a thing I don't know.
I'll call it in when you guys are ready.
I'm ready.
Yeah.
Yeah, that's rather deep.
Bridge,
but let me
ROV
check
nav.
on that. I'll get back to you in a moment.
Okay, thank you, Bruce. Wait, look at this. Just the numbers just
Go
keep
ahead, Nav.
increasing. The
Another
number of
ship
corals
move,
is what
please.
I'm
Sure.
referring to.
We'll do 30 meters.
A really pretty spot.
Bearing
Lots
075.
of, lots of variety here, and the density is getting higher.
Okay, 30 meters, 075. Stand by.
More corals than
Moves
sponges
initiated.
today, which is kind of interesting. Yesterday we
Rich.
saw a fair number of sponges. And
Well, it's also interesting, Chris, that yesterday I think the corals that we saw for most of the dive were all quite smaller than this. Towards the end, we got to that high point where we saw some very large bamboo coral fans, but we're seeing definitely a different community at this somewhat shallower depth.
yeah, I agree. This is sort
Saw
of.
that very tall.
Go ahead.
Sorry, I was going to say we just saw that very tall, long, bottle brush-like Chrysogorgia. We've seen those now 3 or 4 times, and they're different from these other ones that you now see that are kind of bent over and have more of a tuft on the end of a stalk. So there's several species of Chrysogorgia up here.
This is kind of this branching one that's here in center, just to the left, up above the lasers. And I guess it's a primnoid. I'm not really sure. We could
Yep, moving towards it.
take a quick look at that. Oh, you know what? Let's belay that. You see this long stem thing that's down below, right, going off screen, lower right slightly? This is what we saw earlier. This looks weird, and Scott thought this was a primnoid. It's kind of wavering right there, and lasers are coming down on it.
Yep. 1 second video. Let me try to.
It's really strange. I'm just not so positive it's a primnoid. I almost thought it was a sponge or something. Very odd.
Chris, the lengths you will go to prove me wrong.
I know. I've been looking for this thing ever since we left there. And we've got to find one of these. And I actually agreed with you
It
back
is
there,
very
Scott.
flexible, I agree.
Dina says it's a primnoid.
Oh, I don't think so, guys. I don't think so at all.
That looks about the length of the lasers apart, so maybe 10 centimeters long.
So,
Oh, good call, Chris. That is the sponge, isn't it?
yeah, and it's extremely
It's a funky
unusual.
little sponge.
I have never seen anything like this.
Yeah.
I don't know. Is
It's
this
different.
is this a possible collection? I don't even know how big it is or how difficult it would be to. Well, there it is. It's about 10 centimeters. This is a coral place, so we got to come back with a coral. here. But what do you guys think about this thing? Has anyone seen this before?
Yeah. From my opinion is that this is novel and certainly worthy of collection if it's in a place and
Pilot,
is
whenever
appropriate
you're
size and
ready,
it
I
can be
have more
collected.
zoom.
It's very unusual.
If you want.
Yeah, Tina suggested possible
All right.
collecting too. Pilot, is this a possibility, or is this going to be difficult?
It's not going to be easy, but I think we might be able to do it. I'll just have to look at the surroundings a little bit. But yeah.
Okay.
I'm going to stop the ship.
Yeah, stop ship.
Yeah, nobody's seen this before.
Bridge, ROV nav.
So I think if you could do it,
Hydraulics
this would be worthwhile.
Absolutely. I will try to set up for it.
Go ahead.
I don't even think I've seen
just
a
take an
sponge
easy
with
stop
structure
right here,
quite
please?
like this. I mean, the closest thing I can think of those, the ones that kind of like
Ship's
look
coming to a
like
stop.
are
Alright. Full
right
wind.
on the end of stalks.
I'm going to adjust your
not
tight.
a
If it's
line
okay with
in
you.
like a shish kebab like this.
Yeah, and there's
Like
another
two
one right below it
subjects,
too.
yeah.
I think could be the same thing. Yeah, they both. There's two of
They
them
look
there.
like it, yeah.
So whichever one might be easier.
And behind it, I'm not sure if it's an anemone on the rock or a small cup coral, but it was transparent to white just behind it.
So looks like the chat room is weighing in also. They think it would be a good idea to collect this thing. I don't think anyone recognizes it, has ever seen it before.
Copy that, watch.
Beautiful mushroom coral over there, the Anthomastus.
I think autos would hold here.
He's got a good toe hold. He's just just working it.
Toe is better. Okay. Just curious.
Yeah, autos might work, but certainly just good bottom lock.
Just need to adjust my Z bias real quick.
So we were actually going for that fan that's on top of the rock when we spotted this thing, but that fan is, I'm still not sure what that fan is either that's on top. It's got some ophiuroids, whether it's a primnoid or whether it could be a bamboo or something. It's pretty interesting. When we finish the collection, maybe we'll have a chance to buzz over it and take a look.
Since I know you like me to guess early, I'll guess primnoid.
Okay. All right, just to be contrarian, even though I think you're right, I'm going to go Isidid. We're going to the bet's on.
Just so that you don't agree, huh?
So are we going to go for a ship dollar again, Scott, or something else?
Yeah, something like that, I guess. But I think we can agree, both agree it's not going to be a sponge.
Staying on this.
You're right.
Are you going for the upper or the lower one?
Yeah, the upper looks slightly bigger.
Okay.
I think I might have
easier.
one ship
Yeah.
buck left in my office, so we're both in range of a ship buck.
Well, what we're talking about is here on the Okeanos Explorer, they sometimes have some entertainment in the evenings. And last year they had a bingo game. They had one this year too.
But that looks fine.
If you win at bingo, then you get these little plastic, looks like little plastic cards that are like money, and then they'll have different crew members' faces on the ship money thing, and they're really great. They're collector's items, almost like Okeanos baseball cards or something.
If it's possible to get a full zoom of it before
That's
we
what we're
collect
talking
it,
about.
the
They're all worth
scientists
$1
might
in the
appreciate
ship
it.
store, which they
copy
open
that video.
every day for a few minutes.
If you want to come in.
Okay.
Yeah, watch this. We're just getting a full zoom before we collect.
Sure, excellent. Thank you.
Taking down.
So is that another sponge in the background?
Yeah, and we don't know what that one.
Just it's sitting there.
Let's call it a rattlesnake sponge.
Decided it doesn't want to be on a
Too close
stalk. It's
now.
one that's sitting on a rock. Good. Some similarities, but
Like a lupin sponge. Yeah. Or the
very weird
shark
animal.
sponge.
Never seen it. Sponge on a string. Is that what you call it, or sponge necklace? I don't know.
It does look like a little necklace on one.
Pearl
doesn't it?
sponge.
Pearl sponge, yeah.
I might zoom out just a
If you're
little
just
bit.
joining us now, this is a live broadcast from the NOAA ship Okeanos Explorer and its Deep Discoverer ROV. We are currently in the northwest side of the Wake unit, or the Wake Island unit, of the Pacific Remote Islands Marine National Monument.
Yeah,
and we're
I'm
finding
very happy over
lots
here.
of cool
All
animals
bioboxes
down
are
on
empty,
a
if I'm correct.
Seamount, which is called
There you
Delilah.
go.
I don't think that's the official
Right.
name, but we were on
Right. You want
Samson
it.
yesterday, and
Video
Delilah
whenever
today.
you're
And believe it or
ready.
not, we
Okay,
did not
clear.
make up
Alright.
these names.
Come on
Somebody else did that for us.
Yes.
Yeah,
All right,
about
full
20
wide.
years ago.
Let's do this.
And Jasper actually did the research on these names and figured that out. And we are currently at a depth of 1,893 meters. And we have uncovered a really wonderful bed of deep water corals and some other interesting creatures that we're trying to document here today. It's
Relatively small subject, eh?
So I'm thinking about just coming behinding it. Coming from behind.
Yeah.
What do you think, Nav? Sound good?
I'm trying to help you out with this Titan here.
Yeah, I'd get an upper view, see if you can see what the slope is like. As long as you can get your fingers without clamping the wall.
Yeah. Maybe come from the side.
Not Titan over here.
You can try from above. Do it.
So I see we've got some additional members in the chat room here while we're waiting for the collection. Tara Luke has joined us, Nolan Barrett, and also Deborah Glickson. So welcome you folks.
That's gonna be good. It's very reasonable. It's hard to
Some of
tell
you
with the
may
wall.
remember that Deb
Yeah.
Glickson was
Where the rocks
a
are, but
science lead
gotta
on
be able to
the
get it
first
just
leg
fine
of the
like that.
Marianas cruise
I think
a
the
couple
hard part's
cruises
going to
ago.
be not hitting your fingers
Yeah.
on the wall when you close. But yeah, it's really long enough to grab. Nice. Just wait till it blows just right.
We did not give them the easiest task here.
No.
This one's,
You picked a tough one.
yeah, this is a challenge, but they're lining it up pretty nicely right now.
It's a sneak attack from behind.
So what's going to happen here is they have mounted actually cutting blades on the inside of the manipulator arm. That's a kind of shiny part right above the sponge. It's also got some
just a little wrist
what do
rotate
they call
to
it?
the right. You'll be lined up
Polytubing.
nicely.
What are they? What's the name of it?
Yeah, it's a tubing kind,
Polytygon.
Tygon probably,
Yeah.
which is just a sort of a rubbery type of tube.
Yeah. So what
They're
what they can do is they can cut it,
Yeah,
and then
wrist
the
left a
tube
little
will
bit,
grab
maybe.
it after they cut it. So it's a really remarkable little modification to the manipulator arm here.
There we go.
And there we go.
Nailed it.
You can actually see how the
perfect.
pieces of tube are
Yeah.
smushed together holding it.
That was beautiful. And I'll bet this is going to be a floater, guys.
Yeah, copy that. All right, video, do you want any footage?
Maybe just a second.
This will be specimen 1 bio, yeah.
Video always wants footage. It's a silly question.
Should we center it? There you go.
All right, I think that's probably good with what we got before.
All right.
And do you want mini Zeus or do you like take
All right, we're full wide.
I'll just follow you.
Yeah. You follow me.
So you want to pull off then?
So I'm going to set up, probably get it close to the bio box, and we'll pull up.
Okay.
And go from there.
Cool. You think it's starboard or port?
Port, probably. So you can set that up if you want.
Boy, what are we going to call this thing?
There was a kebab sponge as one of the suggestions for the.
You know what? I
the
like
chat.
that.
Sponge
Why don't you
kebab.
put the field
That's a
ID
good
as kebab sponge?
Okay.
That's
No, no,
very
sorry.
descriptive.
We're
Thanks,
listening to
Tina,
the
for that
conversation, sorry.
suggestion.
All right.
I am ready to draw out.
Draw out.
On your mark.
All right, so I'm going to lift up.
Port bio.
You have your Z bias up real high too.
Did you say it's up high?
It's at 23.
So I noting Sacco and Scott talking about the sound volume. So I'll try to speak up a little bit, Scott. I don't know if that'll help.
Rising.
15.
There you go. That looks good.
So get over these things.
So cruising over there on the right, there is a dead sponge that's absolutely covered with yellow crinoids or feather stars all over it. That's what you were seeing going off screen to the right.
An auto depth. How's that holding me?
Put auto head into it if you want it, I guess.
Still drifting very good.
It's okay.
Look at it for a second.
It'll wobble a bit.
You want me to extend the drawer?
Yeah.
Oh, didn't open. Stand by. Try that again.
Yep.
Port side, retract. Port side, extend.
Headed for port inner or outer?
Probably inner. It's kind of small.
There she is.
Nice.
She is all the way open.
So what I might do
If you'd rather
this
have a
evening
manip cam
after
up here,
this
just let me
one
know.
comes up,
But
assuming they can get it into the box
Can you put,
successfully,
actually,
is I might take a tiny little piece of it and take a quick look under the scope and can obviously confirm it is indeed a sponge, and secondly whether it's in a group of sponges called the demo sponge,
Thank you.
demo spongia, or whether it's a hexactinellid or glass sponge. And then if it turns out to be a glass sponge, then I could probably go further, since I'm a little bit familiar with those, and I'm not very familiar with the demo sponges.
yeah, try to get it in there. It might be a floating object.
Yeah, so
And it
now
might have to give it the shake too. It might get stuck.
let's see.
If you adjust your auto depth up pretty high from where you are, right
Yeah.
before you let go.
Are you in a safe spot, Copilot? So what I'm gonna do
Yeah,
is
no, we're
come
happy.
up.
Big delta.
All right, let's do this quick and see what happens.
I'm ready to close the drawer on your mark.
All right. Close. Thank you.
Perfect. Thank
great.
you very much, guys. That was a
Package
great
is
collection.
Excellent, Copilot.
Sorry, that's my glasses hitting the microphone. I need a little adjust.
Titan has a good view too, if you need it.
All right, so while Chris is adjusting his microphone, I'm taking some notes here. We'll be getting ready to get underway here in a minute. They're going to put the
nice
grabber
collection,
arm back.
pilot.
And I think we're also getting
that was a little bad, too.
A little change here of
great job.
The ROV pilot's navigator and co-pilot.
Coming off. Hey, Washley, we're just going to do a quick watch change.
Sure, thank you. I'm trying to find pictures on the web that look anything like that sponge. So far I haven't succeeded.
Hey, Watchley, pilot back with you.
Right, so pilot, maybe this is a good time for the new front row to introduce themselves.
Okay. Stand by one. All right, well, piloting is me, Chris Ritter, working for Global Foundation for Ocean Exploration. My navigator is Andy Lister, working for GFOE as well, and my co-pilot is Dave Casagrande, who is also working for GFOE.
Give it to video.
And video, would you like to introduce yourself, Annie?
Yep, my name is Annie White, and I also work for GFOE.
You've got Art Howard back here clipping.
Can't forget about Art.
Doesn't have his headset on, so he can't. So he can't say hello.
So he can wave if he wants.
So Les Watling just got on. Les Watling's at the University of Hawaii Zoology. Oh, excuse me. The lasers are right on a coral. If we could get a zoom on that, that's a new one.
Sure. Spiral?
Yeah, the spiral one. I mean, we've seen it before on other dives, but it's a new one for this site.
Is that a Rudogorgia?
That's correct, yeah. Excellent. They're one of the most spectacular deepwater corals down here, so it's kind of nice to see them show up. So while he's getting ready to zoom in on this, I was just pointing out that Les Watling, who's with the University of Hawaii, is just logged into the chat room.
Copy that.
He's also has a lab up in Maine, and he's a bamboo coral specialist and chrysogorgia coral and all types of other animals. But he noted that the sponge we just collected looked just like a type of sponge that was collected off the Galapagos Islands by the Nautilus.
Okay,
so they described as a bunch of balls on a stick. So
that's pretty similar to our kebab sponge analogy. Yeah.
Well, I don't know if balls on a stick is a better name for that. I like the kebab sponge, frankly. So this beautiful coral we're looking at right now is Iridogorgia, and it could be something called Iridogorgia magnispiralis. It's in the family Chrysogorgidae. And yes, Les, who knows these animals really well, definitely concurred. And I did it as magnispiralis.
That's full zoom.
Copy that.
And he's watching and then typing right in the middle of dinner. So, thank you, Les, for this.
Lasers off.
Scott says he's already trademarked kebab sponge.
I don't know. I mean, if there's any royalties there, I think Tina's going to probably, since she's the one who recommended this.
That's right. I wonder whether this could be a synthetic product too.
Okay, why don't you come wide video? Are we happy with that, Watchley?
Yes, we are.
Okay.
Thank you.
Let's keep going then.
All right,
Yeah,
pull
so
wide.
we have got a lot of corals in the background
Copy,
there.
thank you.
This is turning out to be a really pretty sight.
Well picked.
Yeah, these ridges, I'm telling you, these ridges really are really nice habitats for deep water corals. But being a guy who really likes sponges, come on, where are the sponges for goodness sakes? What's the matter? What's up with that?
We had them yesterday.
Yeah, we did. Well, we already used up our quota for several days?
Maybe.
Now, was everything about that sample recorded correctly during the watch change?
Yes.
Great.
So while the ridge is remaining fairly narrow here, we are coming up onto a little bit of a flatter part of the ridge line.
Have you guys seen this anemone yet today?
Yes, we have, thank you. Pretty much we've seen everything, I think, in front of us so far.
Okay.
So Scott's pointing out on the chat that it's not just the fact that we're on a ridge, but also the depth range seems to be important.
Yeah, I totally agree.
2,200 to 1,700 meters.
To the end, we only have to average
And we're
.09.
homing in on this depth range, but we don't understand what's so magical about it. It could be the oxygen levels.
The last one were about 30 meters at a time.
I don't know why the temperature, it's not going to change that much down here. You know,
probably copy that.
You know, a few hundred meters on either side of it, but oxygen could be a possibility here.
Direction.
Yeah,
I don't know.
I think he's turning right now. Co-pilot, what do you think about your heading?
I think it looks more to starboard.
80 degrees?
Oh, that's right, my set point. I mean, the ridge looks maybe more like 80 or 85 to me.
That's fine. Let's just do 80.
Okay. Bridge, Ravi Nav.
Nav, this is Bridge.
Hey, we're going to go ahead and put a ship move in.
Roger.
Okay, we're looking at 30 meters at 080 degrees, 0.2 knots.
Well, looks like it could even be the
Alright, copy, range 30, bearing.
Looks like it's got pretty tight spirals on it,
yes.
but maybe
Speed,
it's just the angle.
.2.
Good copy.
Roger.
Try to come
not.
broadside to it.
Yeah, it's starting to look more like Magnus Borealis. It's just an end-on view.
Running out of tether, I'm giving you a little more.
Yeah, copy that.
No.
Want to do a snap zoom here video?
Yeah, let's do a snap zoom.
All right.
We've got a few folks on right now who really know these animals. That's got a pretty tight spiral to it. All right, Les, you're on, at least on the chat.
There's a tether.
Nav, ship move initiated.
Scott's, while we're waiting for Les to come up with an idea on this thing, Scott was sort of pointing out that, yeah, as we're getting into this flat area, we're sort of losing our corals, but it may be a very...
Or full wide.
Copy.
Very little bare patch and a momentary bare patch before we get back into
Hey,
them.
co-pilot.
Yes.
You still got him on screen?
He's off to my left a little bit. We're sort of tugging each other with the tether.
Yeah. Do we want another close-up of this, Chris?
No, I think we got it, thanks.
Sure. We're just at the end here. Gonna come back home.
All right, so Les did not weigh in on that Iridogorgia, so I think we'll just call it Iridogorgia for now. Oh, the sponge, please. Yeah, we haven't seen one of these so far on the dive, so this is a good one. And we didn't see one on...
not alive.
Well, we haven't seen one on this cruise, I don't think, alive, one of these things. This is a pretty common deep water sponge elsewhere as well.
See if we can get a black background video.
Sounds good.
Yeah, we already have a patent war on the chat about who should own the rights to the kebab sponge name.
So Scott contends he mentioned it first, and he may have, because I was all excited right when we were zooming in on it. So yeah, we're going to have to review the video and determine who really owns it.
I'm going to agree with you. I don't think we've seen this before. At least I don't remember seeing this kind of thing.
All right.
Yeah, so Tina has already identified it as Walteria, which I agree with, and I'm going to go a little bit further and call it Walteria CF Lucardi, which is what we're calling sponges with this particular type of structure. So what I'm looking at is these round holes in the center column of the sponge itself. And that's distinctive. There's another species of Walteria that has a different arrangement. And last cruise, somebody was talking about these little spiny things that are coming off of it, and they actually thought they might be hydroids or some weird black coral or something. And
We should have plenty.
thought they saw tentacles coming off, but
Alright, can you take my camera?
I don't know.
Sorry, watch they were fighting
Yeah,
each other.
almost looks like they are tiny little polyps, but I don't know.
What do you want me to do?
Yeah, just come up.
Yeah, you can see it here and there and against the blue background.
Yeah, but they could also be the actual spicules sticking out as well.
Slowly.
Coming up slow.
But anyway, it's definitely Walteria.
Yeah, hold there. See if that helps.
Yeah, against the dark background, you can really see the...
Yeah, but they're not waving around like tentacles. That's why I think they're spicules. They look like they're quite firm. That's a beautiful shot of those. We've been sort of pondering whether there could be something there. Charles Wall thinks it looks like a hydroid, and that's what was being bantered around, Charles. I think it was last cruise or the cruise before. If it is an idarian, it's probably going to be a hydroid. I don't know what else would be that small. But
maybe keep them up just a little bit.
I think they are spicules from the sponge, and I know that the species has been examined microscopically, and no one's ever described finding any hydroids on it.
Okay, we can hold there. Holding.
So again, maybe some of these Glyptometra feather stars.
Anything else we want to take a look at here, Washley?
I think we're good with this. Thank you.
Sure.
Maybe go up to the top.
Yeah, go ahead.
That's it.
Video, did you want the whole organism?
Yeah, if I could get a full specimen, that would be great.
Go ahead.
And would you like lasers for that?
I don't know. Video, you want lasers or?
Yeah, Scott's saying that he couldn't find any of the hydroid polyps on
Okay.
ones that he's collected.
Bring the camera down a
I
little.
may just shoot off an email to
Holding there.
Henry Reiswig in Vancouver. He actually has got some specimens of Walteria. In fact, I was up visiting him, and we did some electron microscopy on a species of Walteria up there. And
Right, I think video's clear.
Neither one of us, I don't remember seeing any cnidarians on them.
You want your camera back?
Yeah, sure. Thanks.
It was a while ago.
You have it.
So the animals are diminishing. We're getting a little more sediment.
Oh no, I thought for a second I saw a fish, but it's not.
Yeah, just the stalk of something here. Yeah, the corals are still here. That was a pretty brief moment. Maybe they've decreased slightly, the number.
Is that ship move done? Let's get another one in.
Copy, same everything?
I think so, yeah.
Okay. Bridge, RV Nav.
Go ahead, Nav.
Hey, can we get another ship move of 30 meters, 080 degrees, 0.2 knots?
So, Pilot, I don't know if we have a chance to take a quick look at that colony.
Roger, ship
bottom
move 30
center right
meters,
now
bearing
that's got the
080
little
at
stars
2
on it.
tenths of a knot.
Good copy.
Yeah, sure. Which one, left or right here?
There'd be the one on the right.
Okay.
Nav, bridge, move initiated.
Okay, video. Come in.
All right. That's a partial.
Copy. And you can keep coming.
All right.
So it's a primnoid, I guess. I guess it's possibly in the same genus we've seen before, Narella. For some reason, I thought it looked a little differently, but yeah, I think that's good enough, Pilot. I think we've got this one. I thought it could be something else, but
Okay, let me know when you're clear, video.
All right.
these brittle stars are a little different than some of the ones we saw on one of the other colonies.
All right, we're full wide.
They've got
Copy.
spines on their arms. They may be something in the family Okeanos, but I'm not really sure. So Jasper, these little holes in the substrate, they could be just where there were boulders or talus or something like that, and they just didn't get filled in by manganese, or would they be something else?
That's a good question. I think most of the time this is just growing over existing rock, and as the thickness of the layer increases, you start creating pockets between things that were sitting next to each other, between the rocks, the cobbles, and such that were sitting next to each other. And so, I mean, some things come out or fall and roll down, but I mean, once it's this thick in coating, most of this stuff isn't moving anywhere. Not by itself. We even saw them pushing earlier, right? It takes effort to make this stuff move. Once it's coated in, that's an inch of glue.
Yeah, it's sort of interesting that down there, this manganese coating is really hard and strong, but when you bring it up to the surface and it dries out, kind of all gets...
Yeah, it's a mineralization that has water in its structure, and so once you let it dry out, it sort of breaks down and becomes more dusty and greasy feeling, and it'll sort of slowly crumble and fall apart.
We're 10 meters. 10 meters left.
Okay.
So on the chat, Deb Glixon is pointing out that
Sierra should
you
have about
can
a 4
see
minute delay
that
by the way, too.
the actual surface of the manganese has this sort of bumpy look. People call it a cauliflower texture. And the sediment's really highlighting this. You sort of see these little darker and lighter parts to the surface of the manganese crust. Most manganese crust that we've seen today has been a little bumpy, and you can really thank the sediment in this case for highlighting that for us.
Okay, that I see.
Well, there's a little stand of corals there on the right coming up.
That might be
I guess it's just more of...
I think at the top there.
Yeah, oh yeah, there's a
whole
bunch up there in the top. That's right. So you're just in a little bit of a barren patch. These corals we've been seeing today, it's the hemicorallium. So a fair number of these corallid in the family Corallidae at this site.
We're headed up that way, so we'll run right by this.
Okay. Oh, there's a big bamboo whip, or unbranched bamboo.
We're right near the end of that move. I can actually call another one in if you're comfortable with it.
I'd go ahead.
Okay.
As long as co-pilot's alright with that heading still.
Yeah, that still looks good
good
to me, and you're well out ahead of me.
Yeah.
Bridge, RV Nav.
Go ahead, Nav.
Hey, same move. We're going to go to 30 meters, 080, 0.2 knots.
Good copy.
That's kind of interesting
30,
that the
080,
ROV has sort of traversed the top of the ridge and is coming over to the southeastern side of the ridge, and you're seeing an increase in the corals in the center of the ridge. We weren't seeing quite as many, and perhaps we were over on the left side earlier.
Yeah, we were.
So maybe
they're right on the edges where the water seems to sort of flow over the top.
Yeah.
I can get a view out that way. Looks like there are more. That's the way the current's heading too.
Yeah. It's just coming up and over the ridge.
So you think this is tidal-driven current?
I think so.
Swirling around it, essentially.
Yeah, I think that's the dominant current.
Ship move initiated.
Oh, it's Ophiure. They just bailed off his coral. They do that, believe it or not. Who knows why they do that? You just see them. Had enough of that coral, so on to another one.
So,
yeah,
dead sponges again.
Yeah, get them accumulating, but there's not a whole lot of live ones.
Interesting. Yesterday we still had some live ones and a lot of dead ones. Here we just see the dead ones.
That's possible that they're, as we go upslope, we'll start to see sponges. The way these communities change down here,
didn't we start a little shallower today already?
No, we're moving up. Well, we started shallow, but what I'm saying is that, but I'm saying that as we move up slope, there might be a stand of these sponges that...
I see that.
Fell downhill.
Yeah.
Little pink holothorian down there.
Oh, yeah, there we are. Okay.
Saw it.
I haven't seen a whole lot of these today, so...
Gonna get a quick ID, or...?
Yeah, sure. Let's go ahead and...
Okay, go ahead, video.
Take a look at this guy.
I'll center it up.
It's a really pretty shot to show the manganese crusting, too.
Okay, keep coming. Hold there.
Hey, watch lead. We want to come up at 3:32 UTC time. About 3 hours from now.
So, yeah, what is this guy? Could be something like Hansenothuria, but I always default to that for pink sea cucumbers with the tiny little, I guess they're called pseudopodia or whatever they're called, coming off of their backs. Let me see if I can look in our gallery again.
I guess you can zoom a little more video if you want.
Alright.
And whenever you're done with this guy, we can move on, I think.
I'm catching up with you.
Copy that.
We have another 15 meters in the ship move, too.
Copy.
All right.
Alright, I think we're good.
That looks similar.
Okay. This is similar, yeah, so it could be a Hansenothuria. That's in the family Synallactidae.
Or full wide.
Copy that, thank you.
Yeah, so it's a reasonable guess, I think. A little stock sponge. I think that could have been a Bolosomini.
That thing looks rather giant.
Yeah, dead sponge covered with these Glyptometra crinoids. Very common crinoid down at these depths. Assuming it's the same thing. This looks like a Narella musicae I think we just went over.
Might be some loose rocks there. Look pretty big. We only have one rock, right?
Yep.
Yeah, we've got one more rock and one more biologic to collect.
That's right.
We don't see anything different. I may propose a collection of the Corallium just because it's been so dominant here, and who knows if it's the same species, but then other people will probably weigh in on what they think might be a better
Is
collection.
that a live sponge under that guy?
Yeah, it is. Take a
There you go, Chris.
A live one.
Spot to get a good look at it. Okay, video start to come in.
All right. There's a partial.
Copy. And you can keep coming.
Okay. I've still got a little bit more.
Okay. Just hold there for now.
Well,
doesn't that look kind of the same as the ones that are dead? Just
keep coming.
Live?
Okay.
I guess so. I don't know.
That's full zoom.
It's a glass
it's all
sponge.
covered up, right?
It's all covered up, yeah.
Looking straight down at you.
Copy that.
Darn crinoids.
Yeah. Get in the way every time.
That could be it. There's the pretty shot of the stalk of one of those hemicoralliums and the sponge itself. Yeah, I think this is that Spidoscopulia, which I think we had documented. Yeah, we got it in on the list.
Okay. All right, come live video. Push out ahead. I'm right under Sirius.
Right, full wide.
Copy. Where are we in that ship move?
Ship move stopped.
In the
It
hillside
stopped
ahead, you
2 minutes
really
ago.
see a lot of
Another
little
2
lumps
minutes
there.
of,
This could very well be a pillow mound, a little small hillside made out of pillow lavas and then covered in a thick layer of manganese. If only we could just grab one out of the stack.
Yeah.
If you find one, Jasper, you think we can get?
Nah, those look pretty solidly in there.
So I was curious, why haven't you mounted a jackhammer on the ROV
Actually,
so far?
But more interesting, we have like a paleo mag core driller.
Yeah,
You just drill it straight into the
maybe
wall.
a rock saw. Get nice cubes.
You're going to need something like one of those to get a rock sample.
Exactly.
Explosives.
That'd be a little messy.
Make for great video though if they could back off and
Yeah,
they had a
you'd
little
have to
plunger that you could push down with a manipulator.
Yeah. Just for the effect.
Yeah.
And you back up far enough that you can't really see it anymore. It'll be too dark, and then all of a sudden it's light again.
There's a Chrysogorgia in front of us and another Primnoid. Some more Hemicorallium. As we already noted earlier, we haven't seen the really big bamboo bushes or trees that we saw yesterday at this site. But then we're seeing a lot of stuff at this site that we didn't see yesterday.
Yeah, and we're at almost the same orientation of a rift zone. Almost the same depth range.
Yeah. So a lot of similarities in the topography and in the orientation off the seamount, yet there's completely different communities.
Yep.
We can get another ship move in too if you want.
Stand by on that.
Copy.
I think I've about settled out.
Okay. There's a little sponge dead ahead if you want to look at it.
Oh, there it is. Okay, we'll take a look at it.
Bad video.
All right.
He said little for a reason.
Yeah. Well, you spotted it immediately. I hadn't.
So I think that's full zoom.
Okay, we haven't documented this genus, and I think it's Polychaete. Looks like it's got that type of attachment. It's got a scooped out side. It's called the atrial surface.
Stand by one video. We'll try to settle out here.
I don't know. I guess it's Polychaete. Hard to.
Come a little wide video.
We got a nice little view
You
of
want
the
full wide?
Right there.
cauliflower manganese crust there.
Yeah. That's a good point you made earlier, you and Deb, that the sediment in between really highlights the texture of the rock.
Okay, come back in video.
I should just take that whole rock with the sponge.
And that's full zoom.
Copy.
Yeah, so I think this is in that genus. It's in the family Pheronematidae, I believe. So if the idea is correct, which I'm fairly comfortable with, the Pheronematids are the really, really massive sponges that you can find down here. And some of them can be as big as, or bigger than 55-gallon drums. There was one a number of years ago that we saw in the Northwest Hawaiian Islands during a submersible dive, and we called it Jabba the Hutt because it was this immense, huge sponge. And then, of course, last year up in the Northwest Hawaiian Islands too, we basically documented a sponge the size of a minivan that wound up getting a lot of press, and there was a little paper that came out of it. That particular sponge, though, was not in this family. So that was a pretty interesting observation. We still don't know what it is, but typically when you see a very massive barrel-shaped or scooped sponge down here, it's most likely going to be in the family Pheronematidae.
Okay, Watch Lead, I'm going to switch with my nav here. Andy can pilot a little bit.
Okay. Going to do a little bit of musical chairs in the front, and then we're going to proceed on. Tina saw Cletterized. I'm not sure I saw that yet. I hadn't been checking the chat for a few minutes here. Let me see what's going on.
Okay, Watch Leads. I think we're going to get the ship underway again here.
Alrighty, thank you.
Co-pilot, how do you feel? Same distance and bearing?
That looks good to me.
Copy.
We might be getting a bit off to the south side of this ridge.
Okay. I think that might even be better for us. I think that's where more biology is, but
easier with the things right up above the D2. Oh, just kind of floated away.
Oh, is it a fish or something?
I couldn't tell. It's just a darker shadow, but it's sort of behind over the line.
Bridge now.
Go ahead now.
It's not really. Hey, can we do a ship move, please?
Roger.
Range 30 meters. Yeah. Bearing 080 degrees, 0.2 knots.
Roger that. Copy. range 30 meters, bearing 080, speed .2.
Good copy. Thank you, Bridge.
Ship move initiated.
Copy. Thank you, Bridge.
Does it look like it has a couple of those little white?
Copy that.
Oh, the rocks on the bottom?
No,
yeah.
the little white.
Yeah, the little white things that we saw yesterday.
We're out there too.
It's sort of strange.
Is that the same sponge we already looked at, or a different?
Yeah, that's the Aspidoscopulia.
Yeah,
just a smaller version of it.
I turned back as well,
but
so
it's
this
alive.
is kind of where we were.
Yeah, well, the other one was too.
Yeah,
it just had a case of crinoids.
No, but I meant this as opposed to the complaint we had earlier.
Oh, that they were all dead.
They were all dead.
Yeah, I got a lot of colonies in the distance there. Keep thinking we're going to start to see the really big bamboos show up sooner or later. I'm not sure those are them, but nice little stand in the distance. Maybe more of these corallium.
Let me give you a little more tether. Check that out.
Or corallids, that is, because they're probably in the genus Hemicorallium. Are we in a position to make a quick snap zoom to the lower left right now?
Sure.
I think this is going to be an Acanthogorgia. It looks like it's got balls on the end, almost yellow, white stem. Yeah, pretty strange looking, but
A little bit of a Dr. Seuss looking tree.
Yeah, it's...
Yeah, you want to come in real quick, video?
Alright.
Maybe zoom over here.
So that's a partial zoom.
Copy that.
Yeah, it's going to be Acanthogorgia again, similar to that other one. Yeah, that should do it. Thank you very much. That's seen two of these now. They look different than the Acanthogorgia that we've seen elsewhere, but maybe it's my imagination. Problem with this group is no one's really working on them, or very few people are working on them, so they don't know that much about them.
I don't know what that is, but
that's a sea cucumber.
Oh,
yeah,
right there on the right,
there.
towards the center.
Yeah, I think we can pass that one by. I think it's the same as the other one.
And I must have missed that one.
And then I wonder if we can get into the stand up straight ahead. I guess these are all corallium, but there could be some other things in there as well.
Yeah, let me go that way.
Pretty good colonies.
Pretty far north of our track, but you could try it.
Yeah, I'm just going to try going directly over there.
It's a little forest.
Yeah. Yeah, look at that. That's a lot of the Hemicorallium. There's another live sponge down there as well.
Got a lot more current up here.
Yeah. So Tara Luke has noted too what we're seeing. We're beautiful little grouping here.
So this is again looking off the north western side of this ridge line.
Pretty good current right here.
Yeah.
Could be my imagination, but they do seem to be concentrated on the edges.
Right on the edges,
right
yeah.
on the edges.
Not even on the very ridge, but more on the edge of it.
Yeah.
Yeah, when you get up on top of the ridge, you can really feel it.
Definitely. Come back. Get back in view here. Sorry, watchers, I'm just trying to saddle over back to my co-pilot here. But we can, if you want to check that out, we can move that direction.
Yeah, maybe we could run along that edge a little bit as we're going up slope.
A couple right after this move, we might want to just turn that way and go a little bit in that way.
I think we can maybe shy on that at ridge. I don't think we're going to go that way necessarily, but like saddle up like Chris was saying, yeah. Yeah, cheat more to that side.
Yeah, we could. Our next move may be 060.
Yeah,
like that.
sure.
Let's start looking more in that direction.
Anyway? We're about done.
Okay. Yeah, if you want to turn that way, I'll get over there.
Well,
once we're done.
Yeah. I mean, we'll have to wait for it to stop swinging.
Right. I'm looking at 060 now.
Okay.
That looks fine, but yes, still moving.
Like sort of a big boulder there sitting on the bottom.
Yep. There's a number of them. Chances are that actually has a rock inside.
Yeah, that or it's the biggest mud ball that anyone's ever seen.
Well, sometimes you get pieces of the old coral reef that have rolled down. You'll find a chunk of an old coral inside as well.
Got a couple of those lyrate isitids that we imaged earlier on in the dive. It's the, I think Scott was saying it's the I4 clad.
Pilot, do you want me to follow you over there?
I'll come back a little bit. I know you're not done moving yet, are you?
No, I'm not. I'm watching you on the Titan. I just don't have you in my main.
Okay. Yeah, I'll come back a little bit.
Bridge nav.
Go ahead.
Can we do a ship move, please? Range 30 meters, bearing 060 degrees, speed 0.2 knots.
That's a nice colony.
Range 30 meters,
right
bearing
there,
060 at 0.2 knot.
Because
good copy.
That looks like it's more or less the direction we're going.
Yeah, that should be like right in front of us, watch lead.
Yeah, no, that's terrific.
Okay.
That was a nice concentration right here.
I'll head over there.
Yeah. That's it's not where we put.
Ship's moving now.
60.
Copy, thank you, Bridge. Yeah, but you're facing 030.
Yeah, hi, this is Bruce. I have a question for Jason. He mentioned coral reef potentially coming down slope, but I'm wondering, is this geote a shallow enough one that would have been at depths where coral reefs could have formed in the distant past?
Well, yeah, that's I was just answering you in the chat to that same effect. That's the idea. Is likely the way you make such a flat top, some sort of tabletop here, is some combination of erosion of the volcano near the sort of sea level surface, where wave action can help you break down the original volcanic shape, and then if you're in the tropics, just like in the current tropical islands, you'll have a reef starting to build on it. So some combination of erosion and reef building will end you with a flat top coral sort of platform that existed somewhere in the distant past, and is essentially as the volcano moves further down into its line towards subduction, the volcano cools and sinks, and the plate sinks, and the reef at some point doesn't keep up anymore with staying near the surface, and the whole thing drowns, and you end up with a flat top underwater volcano.
Well, thank you very much. Is this geote of an age where it would have formed in the South Pacific superswell and then subsided?
That would be the idea. Given the amount of manganese crust, I would say this one is probably similar in age to the ones that have been sampled a little bit further to the west and northwest from here. But we have no actual ages for this particular volcano, so your guess is as good as ours. From the flat top seamount structure, similar to ones we have dated, I would say it's probably close to 100 million, but that's based on the fact that we know some of the other ones that look like this are that age.
If you wanted to cheat a little, Copilot could come 30 degrees over on his heading and follow you up there.
Let's. There's maybe 20 meters ahead at 060. There's some pretty good.
Oh, thank you, Jason. It sounds like we need to get another really good rock sample.
So if he comes over and you go the way that you were going to go.
Yeah, there's a few loose ones.
Continue. You just got to keep in mind we're not moving the heading.
Okay.
You should be okay though.
Okay. I'm at 030.
Oh, here's maybe something. This rock up here. Oh, there's the fish somewhere.
There's some to the right over there on that boulder.
Yeah, and there's a fish right here.
Where's the fish? I don't see that.
It's further
oh,
right.
it's serious?
I see it. And your manip. Keep going right. It's moving behind you. I don't think you're going to get it.
What?
Vert.
What about it?
This vert, do I need to reset very quick? Is it still to me?
Oh.
It's 90 degrees to your
bring
right.
Bring up your thruster page.
Okay. Thrusters. Starboard vert. Close contactor.
Yeah. Power it off. Wait a second. Okay. Okay.
I'm wondering if it's something in here. No, it's not that. Want to try again?
Yeah, close it.
Close contactor.
So we're waiting here. Those of you who just joined us, this is a live feed from the NOAA ship Okeanos Explorer. We are currently in the northwest section of the Wake unit of the Pacific Remote Islands Marine National Monument. So we're about close to 200 miles northwest of Wake.
Try it again.
And we are down at a depth of about 1,880 meters. We're on a ridge that's extending off from a seamount. That's we're calling Delilah Seamount.
Right
and
yeah, I'll move forward just a little bit.
Was it like that earlier?
No, it just.
We found ourselves in a really beautiful community of deep water corals, dominated a lot by corals and then a few sponges as well.
Yeah.
And then there's been a bunch of other animals that tend to live in these communities. There's been some fish and shrimp and brittle stars and a bunch of other things. We managed to collect right now a very, very unusual sponge. And we got a really nice rock. We're still going to try to collect another rock and a sponge as we move further upslope.
There's a fish in the lower left just going out of screen now.
Is it below me?
Yeah. He was swimming pretty fast.
I think he's gone.
We're at the end of that ship move.
Yeah, I can go check out these guys over here. There was some interest in this earlier.
Pilot, I'm trying an ID on this.
What are you trying?
Continue. I'm going to let the voltage drop to 0 before I bring it up again.
Okay.
Looks like we've got a nice little collection of the corallium and the Norella on this boulder. Oftentimes you'll see the corals kind of clustered on these little rises, boulders or ledges or things like that. So we're surveying this area. This is the first time the seamount has ever been explored. Also, this is the first time there's been any exploration in deep water in the Wake Monument itself. So yesterday we were on a seamount about 30 miles away to the southwest of where we are now and had a completely different community of deep water corals. They're very different. Also a lot more sponges. So it sort of just punctuates the point that
don't think so.
You can go down in very similar depths
come up a ways if
and
you
on
wanted to
similar
disable
types of
everything.
seamounts, and in this case, ridges that are in the same orientation, and find something that's completely different.
No,
So
keep piloting.
Yeah, that's what I mean. I can fly like this. Just gotta be cautious.
Yeah.
it's pretty exciting because we're starting to get a handle on what types of animals that we'll see down at these depths, but what combination of different animals that prefer this depth is always different from seamount to seamount.
Or nav.
It's going to be another pseudoanthomastus, I think.
Did you want to zoom in on that? Watch it. I didn't know if you had any interest.
Workshop
in that,
now.
or just in general.
Did you hear?
Excuse me? No, I'm sorry, I missed it. What was it?
I didn't know if you wanted to zoom in on that or had any interest in that, or just keep.
I think it's a dead sponge stock, actually, so we could probably pass on that one.
Okay, I'll head up to that community up ahead then.
Sure, that would be great.
Okay.
Thank you.
Right here?
Little Chrysogorgia. These pink corals are Hemicorallium. They're in the family Corallidae, which in shallow water they're harvested for jewelry in some places in the world. The dark red ones are a soft coral, mushroom coral.
Yeah.
We've seen a bunch of those
That's
today
the solution
already.
on deck. But
yeah, do we have any reason why that happened? Idea, I mean.
It's an electrical question.
So we've got sort of a big sponge coming up ahead, and that would be kind of nice to image.
Sure.
Yeah.
Absolutely.
Up and a little bit to the top left.
Yeah, this big white one right in the center.
Because I don't want to crush anything here. The one that
the big one that's got the crinoids on it. Yeah.
That is a very interesting looking one.
Yeah, I think
More
it's
of
just
a fan shape.
Yeah, I think it's just an older one of these Mosquitoscopulia, but again, we haven't seen very many live ones today.
Yeah.
So it's. Oh, excuse me. Can we look at the white sponge to the right?
Oh, sure.
And if you zoom in on it, I think you're going to see a whole bunch of little anemones all over its surface.
Okay. I'm just going to get a tow hold real quick.
What's your camera angle, co-pilot? Are you looking
I'm
down?
straight down.
Yeah, okay. I'll hold off on another
let's go
move.
ahead, zoom in.
Yes.
All right.
And co-pilot, you want to control my camera?
There they are.
Backing away really quickly.
Whoops.
Yes, you are.
So there is a genus of glass sponge down here. It's called Lithroella, and it seems to have a commensal anemone that lives throughout its tissue. And when you actually key out that particular genus, there's no mention of the anemones. In fact, it keys out very quickly from the family it's in, which I think is Eurydidae, and it keys out on the basis of its funnel shape. And we noted in the Marianas that we found one with these anemones that did not have a funnel shape, and this one really doesn't either. And so I think the genus has more members that we haven't discovered yet.
Do you want the middle or slowly pan?
I took a look. So happened that I was dealing with and maintaining the specimens from the Marianas as soon as I got on the ship, and I took a small piece of the one that was collected in the Marianas. It actually looked like a paddle. It wasn't anything near like a funnel, and it certainly looks like a Lithroella. So that key is probably a little outdated. I think we've got some different shapes for this genus.
Right there.
So we collected a number of the Lefroyella in the Northwest Hawaiian Islands last year, but they all look funnel and sort of classic shapes of what they're described as looking like in the literature.
Watching, was that, do you want to see something particular on it?
No, that's terrific. Thank you.
Video, if you want to get your shot, frame a shot there. You can zoom out.
Actually, we're all the way in.
No, you can zoom out.
Oh, okay.
If you want to get a frame a shot.
So a number of years ago, the suggestion was made that these anemones are something in the family Edwardsiidae, but that hasn't been confirmed.
If it could tilt down a little bit, that would be great.
Yeah, there's
the one in the background is bouncing.
Yeah, I think there's some current.
Must be.
If you just kind of pan
This
down
particular
towards the
sponge
base,
is
that
a
would be
rigid
wonderful.
sponge that we're looking at right now, so it's not moving. It's a hard sponge.
There's the base.
Yeah, that's great.
Okay. Yeah, whenever you're done then.
We could get a full specimen for just a second. That would be great.
Holding.
So we could probably skip the bigger one in the background. I've got a pretty solid ID on it, and we've imaged one earlier on. Just wanted to check it out when we were heading over there.
Alright, I think we're good.
You should come up and then push straight
And
ahead.
full wide.
Coming up.
Or to the right. Yeah, there you go.
Give me your camera back.
Thanks.
You have it.
Just so you don't hit any of those corals.
Yeah. Thank you.
Wow, there is a big colony over there to the left. Is that how we're headed, or are we headed a little bit different?
We're headed a little bit to starboard.
Oh, to starboard? Okay.
I think that's kind of the north ridge. North edge of the ridge there.
I see.
Going down. What were you going to say, Copat?
We should be able to move there if that's what they'd like to do.
Yeah, I mean, I think it's a trade-off of moving up slope or over there. I'll talk with them. Oops. Watch the nav.
Roger, Nav, go ahead.
Hey, so that's on the, like we said, that's on the north side of the slope here, kind of going downhill. We usually don't like to go downhill, but we can kind of saddle it where we are right now and get some views as we continue to the northeast. Does that sound like a good plan?
Yeah, that sounds fine.
Okay.
That'll work. Thank you.
Sounds good.
Okay, so just kind of continue what we're doing.
Yeah, let's maybe do another 030
Okay.
or a 0.
We actually moved 060, I believe.
Oh, okay.
Continues to be up the slope.
Yeah,
kind of sidestepping.
So let's do that then, if you guys are happy.
That sounds good.
Okay.
030.
060 for moving.
Yeah, I'm gonna turn to 03 just to
Copy.
continue what we're doing.
It's a little bearing.
It is bearing up there.
Are you out in front enough, or
Yeah,
do you
I'll
want a
get
little
out
bit of time?
So we've got a sea star on the seafloor there. I don't know if you see it.
I do. You want to?
Yeah, let's go ahead and take a look. We haven't seen a whole lot of stars today.
Watch where you set down.
Copy
Some
that.
stuff under your left toe.
Yep. I'm watching out for those guys.
Is Chris Ma East Coast?
He's East Coast, so he's been checking out the Facebook site after the next day, but he's been trying to get a little sleep. I don't know why, but seems to need sleep.
Yeah. sure he'd wake up for something like this.
Yeah, if he knew about it. This is a different star. Something weird is going on with a sea star, and I guess we'll get a look at it in a moment.
Yeah,
got some weird bumps on his back.
I'll hold off on the ship move.
Okay, thanks.
Is that like a little black coral sitting next to him here?
It's crinoid, I think.
Crinoid.
Yeah. And this over here on the left looks like it's a Crallimopharian. That would be.
Want me to take your camera here?
Not yet.
Okay.
So that's a partial.
Okay, now I'm going to have to hold forward.
Taking it. And do you want me to just hold it, please? Do you want me to track them?
Well, I'll guess the family, Goniosteridae. Beyond that, I have no idea. That's a pretty strange looking sea star. Now, if Chris hasn't gone to bed, he'll hopefully call in another 30 seconds or so. We can always hope.
Co-pilot, you want to tilt down on the tether cam, maybe? It's
this one.
Yeah, I think it's that one. Yeah, if it's.
Do we have any more zoom?
Yes, we do.
Acting properly.
Shot.
Is this something particular, or you want to just see behind it?
Yeah.
Alright, that's full zoom.
Okay.
Keep coming. Yeah, I just wanted to get eyes on that. That's fine. Thanks.
Yeah, that'll certainly help Chris with the ID, I think.
Okay.
Responding.
Of course.
So is there anything else you want me to focus on here?
No, I think we're good. I think that'll do it. Thank you.
Then video, it's whenever you get your shot, you can come out.
Alright, I think we're good.
Okay.
Can't quite tell the relief on.
Alright,
Thanks
roll
from your
wide.
view. Okay.
Bring your camera up, and then I'll give it back. You have it.
Thank you. Now we're about to hit this large bearing spot.
So the substrate's changed quite a bit here.
I'm ready for that shit.
Yeah, we're getting a lot smaller particles here with
tether cam back up.
sand and small pebble-sized pieces.
So then I have to ask you, is this a good spot to consider your second rock, or would you rather be closer to the rocks?
Yeah, this is all very small.
Okay.
Bridge now.
I'd be worried that we would be picking
Go ahead,
up
Nav.
just a piece of manganese crust, essentially.
Like a ship move, please. Range 30 meters, bearings 060
And
degrees,
we want to
speed
optimize and
0.2
get as much
knots.
as we can in one rock.
So if we get
Good
a
copy.
chance,
Range 30,
there's an interesting
bearing
star to the
060,
rock to the
speed
right,
decimal
but then
two.
there's it almost looks
Good
like a
copy.
tree branch in the upper left third of the
Are you
screen.
going to head to 060?
I can do
If
that.
there's any chance of getting over there and looking at that, that would be good. That's going to be different.
What was that watch lead?
Well, there's a sea star up ahead that we haven't seen, but also in the back, about the upper 1/3 to the left of the screen, there's looks like a big branch.
Okay.
And that's something new, I think.
Okay.
So maybe those two items would be.
I'm still looking for the sea star.
The sea star is straight ahead, unless it's a crinoid. Could be a crinoid. It's on the rock to the right.
Ship move initiated.
Kind of
Thank you, Bridge.
upper right, about. And he's up from the lasers about now. Yeah. I guess it's a crinoid, but not sure. There's another one of those weird kebab sponges. Yeah, it's a crinoid. Maybe we'll skip that.
Okay,
And then I
I'll head
wonder if
over
we.
to
Yeah, that.
This looks really busy up here.
Yeah, this is going to be a different type of bamboo coral. This thing here. That's why we gonna take a look at it. At least that's what I think it is from a distance.
What's this?
I don't know.
Is it a fish?
No, it's not a fish. I think it's a
No.
Walteria
Yeah,
that's
fell
fallen over.
Yeah.
You just got to pick the large enough rock if you want to get big, or you're going to flop over.
I'm not gonna. I don't have enough tether just yet.
Yeah, I don't really want to give you much more.
So I guess we have that in common with them. We should be getting the bigger rocks. They just pick the small ones now and then.
So what about this one right here? It's kind of a
I'd
nice.
probably rather go closer to the.
Oh, closer to the outcrop itself. Okay.
We're under way right now.
Yeah. Yes, we are.
Power cycle the aft
Wouldn't
pan
surprise me if that bamboo coral, the rock has fallen over to a certain extent as well, and that he may have started growing up a little bit. If that's what it is.
Yeah, looks like he's on a little more black piece of rock than.
Okay, Nolan Barrett is signing off. Thank you for watching for a while, Nolan. Appreciate it.
Here just so they can see what we're looking at.
All right.
He's another one of these guys who seems to need sleep for some reason.
You want me to go all the way in?
No, not right now.
Okay.
I'm still just trying to.
That overrated thing called sleep.
Yeah.
Yeah. There's two of those branches going up.
Yeah.
I'm looking at the end of this one, it looks like we know where the sea star came from.
Okay.
Yeah, so I think this rock did sort of fall over. So it's a very sort of leggy bamboo coral, all right. And I don't know if Scott's still on audio or he could talk about this a bit. I'm going to try to look it up in the gallery. And maybe Les is on still. I don't see me have signed off already.
He's still in the chat.
He's still in the chat? Okay.
I think so.
Well, some of these really sparse ones we were calling Keratoisis species sparse. We were calling Keratoisis species sparse. So, suppose we could stick with that for the moment. There's the knocked over Walteria sponge.
You still want to go to that bamboo next, though? Or do you want to take a look at this? Watch it.
No, we can skip this, I think. Looks like it actually came off the rock. Didn't look like it was attached any longer. And then, yeah, I guess we can go ahead and push on. I think we got a good enough view of the bamboo.
All right, come on out. Video.
All right, we're full wide.
Thank you.
So the current today is coming from the northwest or north. Is that correct, Pilot?
Yes. I'm seeing some pretty good current out of there.
Okay.
Now, if that tilt didn't come back up, any ideas?
Nope.
Okay.
Sorry.
Yep.
I see lots of rocks there. I don't know how big they are, though.
Well, some of them are big.
Most of them look pretty big.
They're on.
Yeah, I know. I mean, I can
Got it.
them on there.
Oh, some of these in the front here might, because there's your lasers there. These might be okay down here. The front left area.
Yeah, let's see where's the lasers. How big is that one to the left?
Down over here in the
That
front.
looks like a nice one there. That one just sitting there.
Or the one that's on that was just
It's
on
coming down. The lasers are about on it, just to the left a little bit of their lasers right now. Yeah, right in there.
Should give him a little zoom.
Okay, here, let's go ahead and zoom in.
The ship has stopped, so if you want to take it, you're all set.
Okay.
That's a partial zoom.
Thank you, that's perfect.
Oh, no, it looks like a pretty good rock.
It's got some stuff on it. Yeah.
Is that on it or on the side of
I
it?
can't tell, actually. Maybe it's behind it.
I think it's on it.
I think
It's behind it.
What do you think? Yes, no?
How big is this?
Yeah, 10 to 20 maybe?
Nah, I don't think it makes it to 20. Yeah.
15?
Can we,
Come on, zoom out.
how about that one? A little bit.
Oh, the one
next, what's next to that stock with the crinoid on it?
Oh, the one that's right.
Yeah, you got the right laser on it right now.
Okay.
It's about
Do you want
20.
to zoom in again?
Yeah.
Okay.
Yeah, right there, that one. You got your left laser on it now.
Sure.
Think that's free?
I don't know.
We can.
Could be.
check.
Yep.
Can you
Okay.
push it and squeeze it?
Yep, we can go do that. Let's zoom out real quick.
All right.
The contour test.
That's full wide.
Thank you.
Sounded like that Led Zeppelin song there. Forget how that went. Was it called the Lemon Song or something? I think it was something like that. That was back in, was that Led Zeppelin 2? I'm not sure. Hey.
Yeah.
Charlie Wall is signing off. Thanks, Charlie. And Deb, East Coasters just have no staying power whatsoever. Jeez. As though they have to work tomorrow or something, right?
Yeah. It's only the middle of the week.
Right.
A little closer. You think that's a good distance away now?
That's a little tight, actually.
A little
but
close, okay.
You're fine. I mean, that'll you can grab it from there. Sure.
Okay. So then, for setup, I can just put into.
Joy locks all the way to the right. And I would put in auto heading as well.
Auto heading?
Just know that if you lose a foothold, both those are in.
Okay. Okay.
Okay, I'll bring up hydraulics.
Copilot, get you spun up.
That should be plenty.
See how your right foot is not in? Yeah. So you might wobble a little, but that's okay.
Okay, looks like Sirius is stationary. I'll bring up some cameras. Let's put Zeus on the right, or Mini Zeus on the right, rather.
Okay. I will let you follow me over.
You want me to take both cameras?
Yes.
Great. Okay. There you are on Mini
Errors
Zeus.
have already been cleared, it looks like.
So just clear them now.
Right. Okay.
Test. Come.
Oh, just do it. Okay. Okay. Power is on. Copilot?
Hydraulics are on, yes.
Okay.
Turn on hydraulics first.
I haven't turned them on here yet.
Yeah. telling you to.
Oh, yeah. Oh,
Yeah.
before I even move it?
Yeah.
Okay.
Go ahead.
Twitch.
And then look at your grip force.
3.
Okay, you're going to want it to be more than
7
that.
or 8 for
7.
the contour
We're going to
test.
set it up
Yeah.
at 7 to see if this crushes. Okay.
Okay.
Okay, I'll follow you over.
Great. Looks pretty solid.
It's like a good one.
Yeah.
Nice thick one.
It's not squeezing. No.
Oh. So shall we do it
Yeah.
shall we do it like yesterday, where I carry up the biologics and you carry up the geologics?
Sure.
Yeah.
Yeah.
Okay.
You get that little white guy there.
Do we want to do any imaging or anything like that?
Maybe that white thing that's right on the rock. It's the, I don't know if it'll.
Keep it in the light.
Yeah, I'm trying to get good light here.
Looks like you got it there.
Okay, let's go ahead and zoom in.
All right.
I think it's a sponge.
Yep.
And it could be one of those
Any more zoom?
strange little
Let's go for it.
white sponges.
That's
Or just a little one.
cozy.
No, it is. It's a sponge, all right. Yeah, or it could be a little one, but. Oh, that's good. Still didn't count as a biologic, right?
No.
It's just
No.
commensal. I'm just
Yeesh.
making sure.
You're gonna have to start pulling
Yeah.
up some biologics with some rock
You
attached.
can do it. Have you
Yeah.
done continuous rest before?
Long time ago.
So,
First time we did
Put it in.
it. Okay.
so I guess the one thing is if you could put the rock in a manner where that didn't get mushed up too much, that would be good.
Toggle it.
Try.
Oh,
Might have to be indexed out
have
there.
to be just completely difficult about it.
Probably want to zoom out.
Okay, can you zoom out?
Yep.
Thank you. All right, there. We'll rotate it around for you.
Sounds good.
Hey, Chris.
Hey.
What speed do you
Oh,
normally
is that
like it at
Chris
like
Ma?
That's perfect, I think.
Okay.
It is. I can hear you guys, you know.
Hey.
So did you copy
Are
the
you guys still looking at the starfish?
watch lead?
No, we sit on.
No, we're not, but you can go ahead and ID it for us in any case, because it wasn't too long ago.
Put it in the rock box so that it doesn't squish the sponge.
Okay.
If you can.
Okay, well, it looks
So
to me like
getting with both of you guys, co-pilot and nav,
Evosoma,
where do I currently
which is
have
not really a surprise
samples?
given
Well,
all
you're
the
gonna put
coral
it in
that
the
is area.
starboard one.
Okay.
So
Starboard.
Do you want me to wing in?
albeit belatedly,
Starboard rock box.
Oh, starboard rock box.
a goniapterate starfish which feeds on
Wing in?
coral. Many
actually,
of
let's zoom
the
out first.
stars
Okay.
that you're going to see in this area
Careful.
tend to feed on a lot of these big
Okay. Bring the wing in.
metazoan
Okay,
colonies. So a lot of the coral, but also some of the sponge predators, you'll see around.
There it goes.
Interestingly, a lot of the coral predators have an interesting convergence that they all have spines on them.
Okay.
You
let's
very
roll
rarely
in.
see them
I need some
that don't
swimming.
have the large prominent spines around the edge.
You can
But,
extend too if it makes
but yeah,
life easier,
the
but
one that
just keep
you
in mind we
just
have
saw
bias
has all the
samples.
characteristic
Yes. Would you like me to
features
extend?
of
Yeah, just
one of the
go ahead and make sure
sort of the
this
best
retracted.
known of
But,
coral
Gotcha.
predators
Retracting
called
both.
Help me
Evoposoma,
watch my
which
port
is related
side, if you
to
would. Make sure we
Hypnosperia
don't push off
and
anything.
some of the other things you see closer to shore.
Oh, right.
So,
But there's another one in this area called
let's try
Polychaete, which
this.
you've seen as well.
Okay. Yeah, see
So,
if you can get it.
Anyway, good to be back in touch. Sorry there's such a lag. I'm trying to stay on top of it, but
Don't forget
believe
your
it or
wrist
not,
is
Twitter
still in
actually
continuous.
was sending me some of these
There you
things a few minutes ahead of time, and I'm sort of anticipating some of them now.
So, Chris, I think you're talking about the sea star that was on the very end of the unbranched coral, and it had eaten most of it already. Is that correct?
If you want to use your tight mic.
The one that I'm
That as
talking
well.
about, the one that I just identified, was the one that was actually on the salt.
Basalt. Okay, that
You
was
guys
it.
have seen one that was just on the
Hello?
Yeah, so it was the one that was on just the substrate a little while ago that kind of had a very bumpy back. Is that correct?
Yeah, so that one is Evoposoma.
Okay, and did you?
Be in the
Yeah, and did you happen to see the one that was it had munched its way all the way to the very tip of an unbranched primnoid, so that the whole coral was bending over right down to the substrate, and he could probably finish the whole thing off in another day or two. Did you see that one?
Yeah, I think that was the one earlier today, right?
That's correct.
I believe that one was Caliaster.
Caliaster, okay.
I'm not sure exactly which one. Because,
yes.
It's a very complicated species complex. But that's one of these other sort of spiny goniasterets that feed on coral. And then I think you guys even saw one earlier today, which was on the base of another coral, a little pentagonal one that had spines around the edge. And I think that's a juvenile Hyposteria or something close to it. For all I know, it could be something entirely new.
Okay.
Like,
is that good storage?
Maybe the juvenile we haven't seen.
Rotated in a little.
So there's a lot of unknowns in this area that better sampling will resolve.
Yep.
But so far there's such good imagery that it's at least nice to know occurrence of a lot of these in this area, which previously had been really poorly sampled. Well, not poorly sampled, there's just never been sampled. So.
Great. Well, thank you very much, Chris. And we haven't seen that many sea stars today, so if we see some more, we'll certainly try to stop and get some more close-ups.
I'll try to keep a heads up on what's coming up on when I see them showing up on Twitter and stuff. But
so I'm Max
no worries.
back.
It's kind of my blog Murphy's Law that the moment I step away from the computer, there are a million sea stars that will show up. So it's just part of the dynamic.
Get up there a little bit.
And lots of places to see them. So.
Sure. Well, thanks again, Chris. I appreciate it. It must be pretty late where you are right now. Is that right?
Yep.
9:30.
Well, that's not
so bad.
Not too bad. Okay. Good.
No. Well, I mean, yesterday, I think you guys saw that Pythonaster close to around, I forget exactly what was your time, but I think for us it was closer to 12:31 1 a.m.
Yeah.
So.
Yeah.
But, which was too bad because that was a nice one. But saw it on the rebound, so no problem.
Great.
Cool.
Thanks again, Chris.
Night. I'll be watching still.
Okay. Thanks, Chris.
Have a good night, Chris.
Yeah.
I'll talk to you again later, hopefully.
Yeah. Bye-bye.
Bye-bye.
So that was the voice of Dr. Chris Ma, who's working at the Smithsonian right now, and he is one of the world's foremost experts in sea stars as well as other echinoderms. So he has been a regular expert for Okeanos cruises and an extremely valuable person to have participating because he's so knowledgeable about these animals. So we always look forward to his calls. And then when he doesn't call, we feel abused now. We're so used to having him. Like it's not fair. Something's wrong. So we're so dependent upon him now to help us with those animals.
So if you want to poke off a little further north,
sure.
You have plenty of tether right now.
Okay. And you can probably get another ship move in after that, after I get out there.
Seems to be a lot of particulates coming up over the ridge.
You gonna follow me up?
Yes.
this is going to be my new route here, up to here.
Yeah, this is kind of almost like a cross section or a road cut here.
I'm going to make a new move, 055.
055.
Copy that. Does that sound good, Copilot?
That looks good.
Good current here.
Yeah,
Good,
another
is
one of
it?
those,
Strong?
I think, Arispidoscopula.
Yeah. Wow.
If you'd like, next dive we can send down a knotted line.
I think
that you
they're
can
battling
pay
the
out.
currents a little bit today. It looks like it's pretty good rate, the current.
It's yeah, watch it. I'm feeling it.
Yeah, but is that correct, or is
Go
it
ahead,
just a mirage here? It looks like you got a pretty strong current.
No,
today.
it's a good current.
I'm gonna read that back. I've got range 30 meters, bearing 055, speed 2. Is that correct?
See a little
Copy that. Stand by for
unbranched
input.
coral that was at the bottom there that was just waving in the current. Give you just sort of like little current indicators.
Here's a good one right here.
Yeah, yeah, little crinoids there are just blowing as well.
Let me get across this little.
Army Nav bridge move has been initiated. Range 30 meters, bearing 055, speed 2.
Good copy. Thank you, Bridge.
Well, that looks like it's a Paragorgia on that boulder there to the right.
You go take a look.
Seen a few of those already. Yeah, I think so. Looks a little redder than the other corallium that we've been seeing all over the bottom here.
You want to do a quick zoom?
Sure, yeah, quick zoom would be good. I'm pretty sure that's what it is.
Go ahead, video, whenever you're ready.
All right.
Thank you.
I think we've got a little
Yep,
bit more
it's
zoom.
one of these beautiful Paragorgia bubblegum corals. Tiny little sea star right over it, yeah. Little baby, I would assume.
Gonna drop the lasers.
Yeah.
It's got 5 arms, maybe a goniasterid, not really sure.
Might do another quick zoom on that star in the back there. See if I can get anything.
Pretty far away from it, and you're back.
I'm real far away from it.
Yeah. All right, thank you, pilot. That should work for science.
All right, video. Whenever you're done.
We can get lasers going back on.
All right, we're full wide.
Great, thank you. 055 was our last move.
Oh, Bruce called out a fish. I never even saw the fish. Did you see him?
I did not.
Yeah.
Yeah, Chris, the fish actually wasn't on the main screen when you were picking up the rock. It did swim past it. We caught it on one of the quad screens. But it was moving along at a fairly good rate, so it just swam
Looks
through
like
the
there's
view.
a
It was another
fish
Synaphobranchus.
Ah,
in
okay.
the center
All right, thanks,
at the bottom
Bruce.
of the screen right now. A little tiny thing.
You're
That
gonna
might
have
not
to,
be a fish.
You have to yell when you see it, because I definitely don't.
No, it's getting smaller. Never mind.
Did you adjust your Z bias?
I did for the rock. I was trying to.
It's probably a little bit too
kind
much.
of nice formations
So
there.
a little less.
Yeah. No, I mean that's too much of an adjustment. So like up to.
Bring
See
it
how
up.
you're drifting off of
Yeah.
everything. Yeah.
Yeah, I'll bring it up some. Right.
Yeah.
Maybe right there.
Yeah. Okay. You might have to play with it, but
Yeah,
that'll explain you kind of.
That's kind of what. Yeah, I was messing with it because I didn't get it perfectly right.
This is the manganese. I see. Yeah, so this would be your manganese over here on the left, right?
Oh,
That's
RV9 bridge.
Okay. Yeah. Yeah.
Okay, bridge.
Did you want to zoom, Jasper?
Yeah, be advised. I'm going to bring our head around.
Yeah,
the
right where you
forces
see
of-
that.
We got everything on the port side right now.
Right
I'd
here.
like to bring my head around to 070, and we finish this position move. We're about 4 meters away from your spot.
That's perfectly fine with us. Thank you, bridge.
Copy that. I'll let you know when I begin after we stop.
Roger.
Kind of sort of see how the manganese sort of forms a blanket over everything.
It looks like there's a sea star at the base of that whip on the left, on the right.
Up above the lasers a little bit?
No, down and to the right.
Yeah, Succo is pointing out a
there's a whip
branch
coral
coral on
still
top.
off to
I'm not
the
sure I
right.
see that one. I've got my eye on a
Of
big
the
one off to
lasers.
the right after we
right.
image
To the
this.
right again?
Yep.
Yep, I
Yeah.
see it. Go ahead and zoom.
Okay.
And you wanted them
No,
to go up a
no,
little ways,
it's
right?
just-
Come on out there.
RV9 bridge.
Yeah.
We look like we are finished with
Okay, watch
position
it. Should
move,
I
and
go
I'm
ahead
going
and
to
push
bring
it
our
out?
head around the port.
Sure.
Okay.
Copy that, bridge.
If you have something that you like.
So there's a really big colony over there to the, just coming on screen on the upper right. I wonder if you can make it over there. And I think there's a fish behind it too.
Yeah. There's a fish.
And
Just
the
up
fish
above
is going to
corals.
swim right down to the colony.
Zoom.
Very,
Go
very,
ahead and zoom.
Yep.
yeah, all in the same frame.
Let's get it all here.
So that's just a
So
really
that's a
big
partial zoom.
primnoid, I think.
Thank you.
I keep thinking we're going to see big bamboos. I suppose it. Yeah, I don't think it's a bamboo. So this one is either going to be, I think, a calyptrophora or a paracalyptrophora, this particular coral. Well, there. Yeah, no, that's fine. It was just.
He's getting off my camera here.
I can zoom out if you want.
I think I got him right here.
You're really far away.
Yeah, let me get it.
Bump up your Z bias some more.
Okay. Got it 8.5.
Donk.
Yeah, there you go.
Head first into the sediment.
Did you want to go see those
Synaptobranchus, okay.
corals again?
Sure, let's
Zoom on out. Where is it? There it is. Okay.
do a quick look at this. I think this could be a different species of the primnoid coral. Yeah, and it's going to be pretty much sideways to the ridge, so they're all lined up to take the current flow coming over the ridge, I think. Yeah, sort of looking this way. Yeah, there's sort of an east.
Okay, cool. I didn't know if you wanted that to go out to other geologists. Yeah.
You can see this coral sort of waving around in the current too. You see the ones that are not, they're rigid. So the corallium, these peak ones, or the corallids, they have a very stiff, brittle skeleton, so they're not going to wiggle around at all. You can see they're perfectly stuck. Whereas this primnoid, this larger fan that we're coming up to right now, you can see it moving all over the place. So it doesn't have nearly as rigid a skeleton. Yeah, and there's one of those anemones.
We're stationary now, guys. You want to get another move going?
Yes.
Sure, he's...
I'll go ahead and push out.
Still pretty close.
No, you're
to
pushed
me,
out. Aren't you?
I'm
looking
at
down
78
at
right now.
Okay.
Yeah, I can push out. Not a problem.
Oh, no rush. I thought you were out a little further.
Looks like the ridge continues up at 055060, something like that.
Copy that.
Bunch of stuff off in the distance. Look at that. On that big rock up ahead.
Yeah.
There's something really big again, almost slightly straight ahead to the right. Or what that big massive thing is.
That guy right up there?
Yeah.
Let me turn in that direction, see what it looks like.
He's looking pretty much your center line, isn't he? Or was?
It was.
Yeah. Wow, this is a pretty scene here. Corals all over these rocks.
045.
045.
Okay.
It doesn't look too bad in that direction. Do you think we should do that?
Sooner or later, we are bound to run into the big bamboos. That's why I keep focusing on these large. I'm convinced they're there.
Let's do 0.
We're at 1,850 meters.
Yeah, I'm pretty sure this is one of them. I think that we're finally going to get to. Yeah, I think so.
Or let's do 045. Yeah, that's
045.
So which one was that?
This big one that's straight ahead.
The one right here?
Yeah, we're sort of looking on it on end. But
okay.
I think this could be one of the things I've been looking for.
Ooh.
Go ahead, Nav.
Let's zoom in to start with. Just snap zoom real quick.
Yep, it's going to be one of these big bamboos. That's what I was looking for.
Wow.
All right, I copy that. I've got range 30
how high that
meters,
is.
bearing 045, speed decimal 2. Is that correct?
Here in a second.
Copy that. Stand by frame.
Lasers.
Zoom in or out?
Did you say kill lasers?
No.
Oh, you want
okay.
them. Bring them back on.
Nothing yet. Let's wait. Actually, would you like a zoom in?
Sure. Yeah. Well, let's go ahead and do a.
Let's go ahead and do that.
Zoom in.
And now should I lose them?
Harvey Nav bridge move has been initiated. 30 meters, bearing
Ours are so
045,
far
speed
out, we
decimal
can't
2.
get much light on it.
Okay, I'll try to get quite a bit closer then.
Okay.
Zoom out just a bit.
We're moving in that direction, so if you can't reach it.
Okay, I think I got an idea.
I'm going to get a little bit closer, watch Lee.
Okay. I'm going to look on the OER Animal Guide. Another plug for it. Shameless plug for it. To see if I can.
It looks like we were looking at 2 of them on the end.
Oh, yeah.
What we need to do is we need to see how it's branching and where the branches are coming out.
Okay.
One thing. So, and if it's branching the way I think it is, I think I have a shot at it. And maybe Sacco or Scott. Scott's still in the chat. So maybe they might have an idea about this too.
So then if we zoom in right here.
Yeah, so I'm seeing
take a look.
Yeah, this should be good. And then I'm seeing a bunch of these big bamboo corals.
Let's keep D2 in view there.
Yeah.
Sorry.
That's all right.
So Scott, I don't know if you're still on. Maybe I can spur you on by a questionable ID, but I would think this could be a Jason Isis. In the, I guess you call it the J-clad, but anyway, if you're still on, maybe you can blast that to heck and gone and come up with a good idea, or Who knows, you might agree with it too. That's terrific. And we've got another type of Chrysogorgia there to the right, a little different
Let's
one.
lose lasers.
Okay. Yeah, you can go ahead and do that. This one right here?
Yeah, that one there. It's a little bit different from some of the others we've seen today. It's safe to put it in the Chrysogorgia, the genus, but that's about it.
Zoom in a little bit? Yeah.
I think we've still got a little bit more zoom too.
See, it's got some anemones in there. I think that's what that is. Or it's a crab. Maybe it's a crab. No, anemone.
You're still fighting your Z-Bias. Yeah. Just try to throw out
So Scott has confirmed
the second one
that it's
Gnarly.
likely a gnarly old Jasonisis type for this big one we're looking at right now.
Okay.
So good.
Let's zoom out for just a second.
So it's branching right off its nose.
Yep. And then of course he goes, or possibly Orstomyces. Wait a second. Is that right? You go look.
Now you're just going to try and disprove them?
Yeah, exactly.
Uh-huh.
I obviously can't because he's one of the authorities, but Orstomyces, where's the Orstomyces on this thing?
Looks like there's a fish swimming behind the Chrysogorgia. Way in the distance,
way
off
back
to
there.
the right.
Okay. After he's finished with this guy, we can go. Oh,
There's something
there's
coming.
a shrimp.
There's a little shrimp guy.
And it's not blue either.
There he goes.
Maybe the color of the shrimp is reflecting their political parties, do you think? I don't know, it's
You're going to zoom out a little bit. Watch it. Did you want to get closer or see anything else?
I think we're good, thank you.
Okay.
Yeah, so I think we're getting a change in the community. That definitely looks like we're getting a stand of these big bamboos starting up.
Before you come out of Octa, look at what's keeping you down. Or was it 12 and 13 for that entire thing? Okay. So that's a pretty good idea of what your Z-Bias should be.
1871.
11.
Yeah.
No.
No, I know I'm off.
1852
You're right.
then.
Yes, you are.
Okay. I'll head.
And right now it's even more. Right. So when you adjust Z-Bias,
Oh, I was
do it
looking
very,
at the
very
CTD
small
pressure,
amounts.
right? Not
Okay.
depth. Okay.
Because you were at like 17 or something, right? You brought it all the way down to 5.
Oh, there's a fish swimming into view
Just keep that in mind.
on the right.
You're good though.
No.
Go get that fish.
Oh,
There's a
there's
fish
another
coming in.
one.
That's the wrong way. Okay.
Yeah, I wonder if we
Let's
could.
let's zoom in. There we go.
All right,
Got more
Bruce,
zoom.
you're going to see
What's that?
something very
Add
soon.
a little bit more zoom too.
Okay.
If he-
If it'll.
He's coming at you.
I don't want to hit that rock full of sponges. Actually, I want to get. Okay, go ahead, zoom out.
Yeah, why don't you?
Yeah, I wanted to
Oh,
get.
this looks like another
Yeah, I
synaptic
wanna get-
blanket
let me get
eel,
placed
but the
well.
head looks more pointed on this one than the others that we've seen. So this may
So
be
we're
a
zoomed out all
different
the way?
species.
Or
Again,
all the
if we
way
can zoom on the head, and then back on the body behind the head. That would be very helpful.
Yeah, so Bruce, I thought I saw white lateral line pores, so I was thinking it could be an Iliophis. Does that make sense to you, or is that off?
No, that makes sense because it looks like it's a different head shape than the Synaphobranchus.
You got a little bit more down,
Iliophis with the white lateral line pores seems like a good identification.
Yeah, right there.
Great.
Yeah, thank you, Bruce.
Okay, I'm going to rotate back to the heading we're moving.
Okay. Push out ahead.
I think that may be the first one of that genus that we've seen here at the Wake Atoll region. So that would be probably a new record for this location.
Looking back.
Yeah, Bruce, we saw one yesterday that I tentatively identified as Iliophis as well, and you'll have to take a look at it. But it also had those white lateral line pores, and it also had a dorsal fin that was very far forward, very close to the pectoral fins.
Pretty busy up there.
Yeah, thank you, Chris. I wasn't able to participate yesterday. So, definitely Iliophis haven't been recorded in the Central Pacific aside from the specimens that were observed, the individuals that were observed in the Okeanos Explorer surveys in the Hawaiian archipelago in the last couple of years. So it's a new record of the genus. Both of those, yesterday and today, are new records of that genus for the Wake Atoll area.
Oh, terrific. Yeah, so we've got a beautiful scene right now.
It seems to continue on into the background too.
Yeah, but we've definitely got a really high density bunch of big corals here right on this little rise.
Yeah.
Really beautiful. These are big primnoid corals. I think they're probably Paracalyptrophora.
I'm kind of losing you behind me to the right.
A stunning scene here.
Get out there.
And you can see the big colonies, as Jasper is pointing out, beyond there on the rock, so it isn't just this rise.
I think I'm over to your right still. Yeah, get out in view. Okay.
Just keep coming forward.
Over here. Get the skids out, yeah. Okay. Would you turn to look at him, co-pilot?
He's coming to view.
So when you look up here, it's clear that you don't see what's below you.
What's that? Yeah.
All right. So we need to adjust heading then.
Okay. Yeah.
You know, that
looks like
the edge.
Yeah, it looks like
the way the contours were, that we didn't see that whole little
in front of him.
pinnacle that was sticking up on top. It was
keep pushing out.
And then I think we're looking down,
and
either down slope or down into that flat area there.
Yeah, we were looking down in that direction
off of the
actual
the ridge. Okay.
Yeah.
And we're still facing.
Still facing to the north.
Yeah.
Okay, I'm looking at 090.
Push out in that direction.
This should bring us back to it.
So you want to get a ship move in, co-pilot 090?
Yes, I think that's about the right heading.
Okay.
So Nav, watch lead.
Go ahead, watch lead.
Roger, Nav. Just an update on our time here and how we're doing.
Yeah, sure. Stand by one. Let me get that for you.
No, it's a coral.
And just so you know, we came off of the north side, so we're going to saddle back here.
Sure. Sounds good. Thank you.
I'm sure you deduced that.
Yeah, I'm just trying to think when we're going to need to make a cutoff for collecting our final biologic, and I'm starting to look around already for candidates.
Copy. Okay.
About an hour. Okay. There's a beautiful big bamboo tree, I think, right on that little, I don't know if you call it a boulder or what. Little things, the thing sticking out of the side of the
thumb.
ridge. Yeah, the thumb.
Giving it some more tether.
Actually, we have about an hour and a half left.
Oh, okay.
An hour and a half. Okay,
bit
great.
more.
Yeah.
Great, thank you, Nav.
And 300 meters, 320 meters to the what we call the end of the dive.
Okay, 300, 320 meters, thank you.
Yep, sure.
Now I think we could start moving in that direction.
Copy that.
Whole cluster of these little mushroom corals down here on the end of that rock.
Bridge now.
Go ahead, Nav.
Hey, ship move, please. 40 meters, 090 degrees, 0.2 knots.
They definitely like it here.
All right, read that back. I've got 40 meters bearing 090, speed decimal 2. Is that correct?
That is. Good copy.
Copy that. Stand by for input.
Standing by.
Oh, we've got the yellow keratoisis showing up here, I think, on that rock dead ahead.
I am too far out, though.
Wonder if we could take a look at that if we're headed in that direction.
40 meters bearing 090, speed decimal 2.
We'll be there in just a second. We're getting a ship move underway.
Roger.
Sure,
It's
thank
a
you.
little bit out of reach right now.
Okay, thank you.
You can give me a little bit of tether.
Yes, I can give you a little more.
Thank you.
That's about all I can give you.
Okay. We'll wait till that ship moves.
There's a change in substrate type right there. What do you call those things? They're not.
Which
Well, right
things
here,
are
you
pointing
see
at?
this.
It's this hard surface of manganese, I think. You're pointing at.
You mean right here, this flatter area here on the.
I'm not sure. I think
sediment?
that might just be dusted with some sediment, making it look like it's something else.
Oh, okay.
Because it still shows some darker spots shining through, and it still has that little bit of a lumpy look to it.
This is a really nice community. I'm pretty darn pleased. It's a nice spot. I hope I'm not the one who's going to annotate this dive. We've only seen about 3 million corals today, but maybe not that many. But three of us that do the annotation later on, so you just pick whichever one is next in sequence. You never know who's going to be doing what. Right now, a lot of the annotation is being done by Miss Sarah Davis, who's in the lab, who's really, really good at spotting things. In just a few months, she became quite proficient at identifying many of the animals from last year. Another person in the lab is also Virginia Moriwaki, and she's our QA/QC person, who's a magician with Excel and databases, and takes our rough annotations and whips them all up in the shape for submission to the national database. So all this video we're collecting today, folks, it's all going to be examined very carefully, and we're going to extract a lot of information, particularly about the corals and sponges and fishes. We're going to extract all this using some software that was originally developed by the MBARI, the Monterey Bay Aquarium Research Institute, and they were kind enough to allow us to bring their software engineer out to Hawaii and do a custom install in our offices. So the software is called VARS, and it controls all the video, and it extracts records of all of the animals with the counts and substrates and all types of information. And then once we've completed a dive, we usually generate hundreds of records per dive. Then all of this information will be sent to NOAA Fisheries, and it'll also be sent to the Deep Sea Coral Research and Technology Program in Washington, D.C. that is has developed a national database for deep sea corals and sponges, and this will be incorporated into all of their records from the Atlantic and elsewhere in the Pacific. So,
hey, Watchley, we had a little shift change, but I'm back in the seat here.
Okay. And then we got Jeff is up there. I've got to get used to the back of your heads.
Yeah, yeah. We'll when I got everybody switched in for our watch, I'll announce us. But I haven't lost track of the boulder they told us we wanted to look at off in the distance there. But I'm still waiting for Sirius. And there's this boulder with things. Did you already look around here?
We could take a look at this large bamboo coral here while we're waiting. And
okay.
It could be a little different than the one we just imaged. I think it is.
All right, well, we'll take a little look here. And then,
I think it's different than what Scott called a gnarly old Jasonisis. Maybe this is a...
If we find it's more of the same, then we won't linger too long. But
it looks more, less flat, less planar.
Yeah.
It's a bit more stretched out in
leggy
directions.
I'm just assuming we're going to take a look at the polyps. Is that good?
Yeah, and then the branch points, so
okay.
To see how it's...
All right, we'll do it flying then. You can come in video.
Scott says if it's Ecnomyces, we'll see volcano-shaped polyps.
Thanks.
I don't know, Scott. Looks more like a seep rather than a volcano. These don't look volcano-shaped, do they look? That was a really stupid joke, wasn't it? Anyway, they don't look volcano-shaped to me, Scott. Some of them are closed up, and they look too long to be...
Bridge, this is Nav.
Volcano-shaped.
Go ahead, Nav. Can I ask how long do we have for this move to end?
I'm sorry, say.
He says it's a volcano when
how
the
long
polyps
do we
are
have
retracted.
for this move to
Which
be
a lot
over?
of them are.
We have 3 meters, about 9 feet to be done with.
Copy that, thank you.
They're half retracted, some of them, I think.
Yeah. It's a declayed.
Declayed, okay.
It's just a pretty, I think it's the nodes before the branching. I don't know if that has any, that's helpful.
Yeah, it is. I think that helps Scott. He's already said it's either a keratoisis or an ectomyces.
Okay, so he's already determined that.
Yeah.
I'll lift up, see if I can find any open polyps, if that's something we want to see. There's some there if you want to take a little more zoom video.
Yep. On the left, maybe.
So that's full zoom.
Yeah, so those are closed up, and I don't think they're volcanoes, Scott. I don't know if you can see this right now, but
there's a couple open ones, partially at least. Sirius is catching up, so that other boulder is in reach soon. Might already be, so we can jump over there when we're done.
yeah, so Sacco also noted they're not fully retracted, and then Scott doesn't, now that he's got a better look, doesn't think they're volcano.
I don't think they look like volcanoes either.
Yeah. You might have a different impression of what that means.
Yeah.
There's some open polyps on the bottom.
Starts with size, I think, too. This is
Co-pilot.
Pretty small for
This
a
is
volcano.
Nav. Go ahead.
All right.
The
when
ship
science or
just
video are
finished
clear,
the
I'll
last
take
move.
off to the other boulder.
We're holding position.
You guys all set?
Yep, we're all
Yeah,
set.
thank
Ship
you.
move's just completed.
Alright.
Okay.
Pilot.
Alright, video zooming out.
Thank
Sirius
you.
is still swinging.
Aye, aye.
Alright, that's full wide.
Thank you. I'll push out to the little boulder out here. Let's see what's going on there.
Once again, I have 2 full pages of animal names from this dive today that are unique animals I've been trying to keep track of. And so it's been a awfully productive dive.
That'll be your pre-breakfast treat again tomorrow morning and filling those out.
Yeah. So, Pilot, we're looking at that yellow thing that's sort of right in between the...
Yep, all the white?
Yeah, between the white.
Okay. I will get situated for that, and we'll take a good look. While we are getting situated, we had a little watch change in the front row. So piloting now is Jeff Lanning. The navigator is Fernando Aragon. My co-pilot is Nick Polenko. And if the video, I don't know if video wants to introduce themselves.
My name is Annie White.
And at clipping, I can't see. Sorry.
That's Art.
Yep.
And Art Howard. And we're all here as a part of the Global Foundation for Ocean Exploration.
And I don't think we've mentioned our names for a long time
it's
either.
Been
so
at least
it's
an hour.
No, it's been longer than that. Yeah. I have a habit of not doing it, or maybe once during the trip. But I'm Chris Kelly with the University of Hawaii Undersea Research Lab, and I'm the biologist lead. And to my right is
Jasper Conter, and I am also at the University of Hawaii in the Department of Geology and Geophysics. And I am the geologist. So you're interested in these yellow guys?
Yes, so
let's see if I can't get up a little higher. Get a little more better light on there for you, sorry. Blocking my lower swing arms light.
There's two possibilities for what's producing this yellow color. One is that there really are yellow isidids. Second possibility is this isidid is overgrown with zoanthid, another type of cnidarian that's very yellow. And we've seen both of those cases.
So that's partial.
So it looks like you got a little of both, maybe. We'll go a little further in and you'll see.
I definitely see zoanthids. So those are zoanthids.
That's zoanthids there,
and I think they all are.
Oh, okay. Just maybe two different kinds, or
yeah, maybe a couple different kinds, right?
I've got a little bit
I think
more
it's just
too.
completely overgrown, which is pretty amazing.
If there's anything, we got a little more zoom. If there's anything in particular you want to take a close look at,
maybe pan down a little bit just to see if everywhere is the same. Yeah, I think so. I think it's...
Can't catch a glimpse at all of any polyps from the original, huh?
No, I don't see it.
Wow, they're so well, like, it looks like... I thought zoanthids were each separate little polyps grabbing on us. Looks like...
No, they can be connected.
Oh, they can, okay.
Yeah, so this is so smooth, the what do you call it? I've forgotten the name of it. The tissue between the polyps, it's so perfectly covering the branches.
Yeah.
That it looks like it's a real, like the actual coral itself when it's not.
Wow.
So we could have definitely mistaken that one in the log if you hadn't zoomed in on it. I would have gone to one of these yellow species of bamboo when we were probably annotating it. So that's very helpful.
Awesome. Well, pilot's clear when everybody else is.
I think we're full.
Okay. Great. Thank you.
All right, we're full wide.
Thank you.
A pilot for our next move.
Yeah.
Chuckling over
like
the
050
message. I don't know how many people saw
possibly
that.
good heading or
heard that. Excuse me, but
060.
Is that the high? I actually don't. I didn't. What was the last moves they were doing?
We stopped at about 5 minutes ago, and it was 090.
090.
We're coming due east, get back on top of the ridge. Is that correct?
That is correct.
And then keep moving.
Yeah. Okay. But you're facing due east now, and I see the. Well, I guess there's some out here too. Yeah. Okay.
There's a ferry near Oka Erecta. That's the first time we've seen that sponge here. So I'll mark that in there.
Need a longer look or keep going?
No, keep going. I've pegged him already.
All right. We will continue on.
In a mental box.
Yeah.
Some tall stuff in the background there.
Yeah.
So still similar terrain, this really large, blocky, bouldery stuff.
And as a result, you see
you're looking down on me, so we got a little time before we can dial anything in. Let me just see what's around here. It's pretty dense, and they might want to look at something more. So.
Right. Well, we're also looking for our second biologic collection here. I've got an eye out, and my fallback, if we can't find anything interesting, well, maybe this bushy one straight ahead in the top.
Okay.
This could be this very large Chrysogorgia that we saw, I think, on the last leg. I think it is. I think it's this. This is great. This one here, and some big Polychaete sponge behind it. But yeah, let's get a close-up of this colony right here.
Okay. We can do that.
And I don't know if Les Watling is still on, but
I don't think so.
I don't think so either. He was, I think, working on this. I'm not sure it has a name yet. Some type of a branched Iridogorgia, if I'm not mistaken. This thing is beautiful.
Oh, yeah, all the spirally branches.
Yeah. Yeah, exactly. The normal Iridogorgia just has that one strand, as you know, that spiral strand, and this guy is just tons of branches.
Interesting. I guess we want to try to get in there and make sure we see branching. Is that what we want to try to find here?
No, because there's nothing else like it, I don't think. Just, we can do a zoom, and maybe Scott's on right now. Is that you, Scott?
Video. I'm going to back up a little here real quick, and then we can carefully not to blow it around too much. And yeah, you can start in. I'll go for these sort of outer branches with the dark behind them.
We've got Amy Bacco-Taylor, who just came in, and she actually is the one person that's collected a specimen of this in the Northwest Hawaiian Islands. That was perfect timing, Amy. Oh, this is the first one of these we've seen in the monument. Yesterday, they were only on dive 3 here, but
they're waving around a little on the current, so I'll try to keep that in view.
It's okay.
Oh, we have Tara on video
Amy
now.
is at Florida State University.
Yes. Good afternoon.
And she's another specialist and expert on deepwater corals.
The current's blowing me around. I'm gonna try to settle down a little bit here.
It's easier. We could go over closer to where the little
stars
are.
Cypasteria.
Sea bias seems to be a little off. Oh, I have the current on my port quarter, which makes this interesting for holding. Every time I try to settle down, I get blown into it. Look at this.
And Sako said she thinks we saw these on a previous dive in Marianas, and I think we did. Thought we could not collect it. So Sako was wondering whether this is a collectible.
Okay to start
And Amy or Scott, would you guys like to weigh in on whether or not... I mean, this is undoubtedly going to be a new record at the very, very least. And I don't know if this would help with the identification if we've got one in the Northwest Hawaiian Islands. But we do have one more biological collection. Amy, just to let you know, when you're thinking about this, there's an awful lot of corallium here on the bottom, hemicorallium.
Yeah, there's not a good spot to settle down here.
So it'd be kind of a hard one to sample. Is that right,
I'll
Pilot?
just take what zoom I can, and then
okay.
Come on in.
We can just move around the organism if we need to.
Okay.
This is a partial.
Yeah, so probably the dominant coral today has been hemicorallium at this location. There's been a lot of primnoids as well, Narella, and we just got into some really large isidids just recently.
Take a bit more zoom.
Okay.
Scott spotted possible Isidella. That's another type of bamboo in the background there.
I don't think either of them heard you ask whether you wanted to collect them.
Well, I think the pilot was saying that this is a rough
spot.
Yeah, rough spot.
Well, no, we have the video wouldn't be as nice if I went into Auto XY, but I think Auto XY will probably hold so we can take a sample.
Well, Amy's just come in, and she, in her opinion, the corallium might be a little more
We go to the brittle stars. I'll come out a little
to
bit.
grab over this. So I was sort of thinking the corallium would be a good possible collection too.
Okay, so something else around here that might be a better grab?
Yeah, so the corallium are all over the place. We can find a nice general place once we're finished with this boulder to grab one of those.
Okay, I'll go in again.
Yep.
If that's what everybody wants to grab. Let's see. Scott and Asako haven't weighed in yet. We're looking at some brittle stars here. These are typical commensals on different corals. These spiny brittle stars, maybe some species in the family Okeanos. The name basically referring to the spines.
Okay.
Watch it. This is Nav.
Video's clear.
Yes, Nav, go ahead.
All right.
After we're done with this, should we start a ship move to keep going, or do we want to stay in this area for a little while?
Well, it depends upon whether we want to grab one of these pink corals here, or whether we can find a better place to grab a pink coral. And this, again, we've been seeing them all day, so this is not the only.
Which coral are you looking for?
See these pink fan ones? They're on edge here on the upper left.
Okay, yeah.
But there's lots of them all over the bottom, and so I think we could probably find an easier collecting spot, because they're going to be really brittle and they might break up and fall down slope.
Oh, okay. Yeah, all right.
Hey,
well, then.
Chris and Jasper, apologize. I haven't been able to be completely tuned in the last couple of hours, and I overheard you asking about this collection. I'm not sure. I guess you weren't getting enough positive feedback anyway, but it's the only one we've seen, and I think, didn't we collect one on the Marianas leg, a piece of one, certainly a branching.
I don't know I don't know if we collected one. We saw one, as Asako had pointed out, but you know what I'm wondering is that the corallium has been so dominant on the site, and we got so few corallium in the Marianas. I'm not, maybe one. I'm not really sure. My vote would be for a corallium, because we have so many.
He starts to get away from you. I'll just put a
that's
ship move
fine with me.
whenever you want.
Copy that, Nav.
Why don't you mind restarting my sonar?
So
This has overall been a pretty spectacular ridge that you chose here. An amazing
close
spot.
and reopen my sonar?
We were joking earlier about the
See how it's not scanning
trademark
on the
the sponge name, and I'd say now that we should be filling out the paperwork to conserve this whole ridge.
Yeah, well, this ridge is in the monument, so it's protected.
Oh, good stuff.
Yeah, so it's absolutely marvelous. This, of course, it's the kind of thing we're hoping to see in the monument.
Yeah, it's fantastic. It's been really good diversity and high abundance.
Yeah, just the diversity just makes for beautiful scenes here with all different types and shapes of corals and all these rocks
something over here that's big.
Yeah,
rocks running wild.
So
rocks running wild.
I'd agree with that.
And you're facing that way already?
I'm facing a little bit.
Okay. Anything else you want?
I
Chris, is that funny-looking sort of lyre thing there that's on the right side? Is that another Norella or Cryptophora or something? It's off the screen now.
It's coming back on the screen. I think these could be Calyptrophora angularis, Scott. And
okay.
Yeah.
Might be three of them that just have slightly different shapes.
Yeah, so Pilot, can you come around on the left side of this boulder? Is that possible or not? Or is the current going to...
No, currents, it's not so much current. That's a factor. It's more likely, I don't know if I have enough tether, but I will come around this side and see what I've got for a room. And we can...
Just doing a swing right around the boulder, I was thinking this would be a really pretty seam. And I was also wondering what that white thing is, but I think it's now the dead base of a coral. Thought it could be a big white sea star, actually, when I first saw it. No, it's looking very dead base-like.
Yeah. At some point, I might tug, just so you know.
Right. Yep, there we go.
Really beautiful scene.
And I'm tugging on Stereos a little, so this is pretty much as far around here as I can go.
Okay, that's great. Thank you.
In the background,
we can push the ship this way, but it's not really a long track. Be a little out of the way, but we can go this way if you want.
No, I think just having a look here will work. I think what we'll want to do is find a little more level ground and then get that second biological collection in the bag.
Okay.
And,
well, I'll head back towards where I can safely, without tugging on Stereos, fly around. And maybe before we start ship moves, we'll find something of that nature. So I'll get sidled over in the direction of the next move. See what we see. Of course, as I go this way, it looks a little more barren, doesn't it? Should get better. Hope it gets better.
It'll get better. keep the faith.
Yeah. I'll drop down a little if you want to come with me, co-pilot.
Copy that.
All right, so I'm thinking this way is the ridge. And that sort of, everybody agree?
Yeah, keep coming east.
Yeah. I think this is the direction we kind of want to push.
Okay.
At least like a 30 or 40 meter hop in this direction will get us on top of the ridge again.
All right, I'll call that. So 40 meters at 090.
Yeah,
sounds good.
Sounds good to me. So we're gonna go sort of this way, Watchley.
Great. This is Nav.
That gets us back to the top of the ridge.
Go ahead, Nav.
Sure, that would be fine.
We're sort of on the north.
Get the
come
next
down the
move
northern
in.
edge
It's
a little
gonna
bit.
be
It looks like
40 meters at 090.
Okay,
at
and don't worry, Amy, we're not going to run out of this corallium, I think.
Copy that. I've got range 40 meters, bearing 090, speed decimal 2.
Got a.
They've been showing up in force
That is
all
correct.
day.
Thank you.
All right, stand by
Got
for
a little
input.
something over here. Just peek and see what it is.
That's a chrysogorgid.
Okay, and then a fan behind it of some sort. Or
yeah, that's
okay.
one of these bigger bamboos. So what we're looking for are these medium-sized pink fans.
Just making sure it's not something new.
RV Nav bridge.
Don't have as much detail in my other cameras as I do in this one, so I
bearing
couldn't tell what it
090,
was.
speed decimal 2.
Copy that. Thank you.
So,
just a big Polychaete sponge up on the upper side of the image.
Okay. I'm going to take a second to settle out and see if I like the Z-bias still. It seems kind of high.
And boulder on the right side, you can sort of see how there's a smooth top to it, and then there's sort of crumbled out manganese crust that sort of crumbled off of it. It looks like there's a good couple of inches of it on there again.
Kind of nice to see these kind of broken open where you can really
I think I see the
crust.
coral.
Yeah.
It's not coral or just a weird coloring in the bottom. That would be weird. We've got second while the ship settles up and comes our way, and I saw this little white flash. If you don't mind if we take a quick zoom just to see.
Yeah, no problem.
Come on in, video, real quick. I just want to see what this is.
See what you mean? Yeah, no.
I don't know if that's a little cup coral or something else right there. Oh no, it's some dead something. Okay.
It's a dead cup coral, though. That's a good
Thanks.
call.
I don't know.
Good eyes,
I was
pilot.
skeptical. I think it is. I think it really is a dead cup coral. It had kind of sort of long flanges coming off of it. But
Yeah. I was hoping it was a live one.
yeah. Oh, okay. All right. Well, then it was a bad ID, pilot. Bad ID. I was impressed there. I was giving you credit for getting that one.
Well, I didn't know if it was alive. I was hoping. Fingers crossed. Okay. Well, we're just... ship's underway, yeah?
Yeah, it looks like
so
we're
we've
starting
got
to get some movement.
what appear to be
Alright.
either little rubble or these could be nodules too, right?
Yeah.
Well, I got settled out this way before we started the ship moving in hopes that there'd be a big field of coral right here, and then there wasn't. So, sorry for the little delay, but we have the ship moving and it's starting to catch up, so we'll get to make forward progress in a second or two here.
Okay, thank you, pilot.
You could come down a little for me.
Sure.
Give me a little room.
So Sako, we're still talking about these collections, and I don't think we collected that that branch to Iridigorgia, Sako. The reason being is, as I remember, it was right on a really steep edge of a drop-off that we were sort of going along and surveying because there were so many animals on there, and it would have been, again, very, very difficult to collect that animal. And it was sort of toward the end of the dive, as I recall. But yeah, we may have opportunity. We sure have got a lot more dives here in Wake.
Yep.
And we might see some other colonies of those.
Seen these anemones before?
Yeah, this is the Exoselectus. We got a good picture of it.
Maybe another pearl sponge or two?
Yeah, kebab sponge.
Kebab sponge.
Please.
Kebab sponge.
We've already given it a name.
Get your barbecue food correctly. Or your barbecue sponges.
I think that's a really great name for it. I mean, if it ever gets described, it would be
kebabensis.
Yeah, or Regadrella, kebabensis.
There's 4 or 5 of them here.
Yeah, a bunch of them.
Another big sponge.
Very weird.
And things are getting a little denser just out of reach there, so should become interesting again soon.
This lyrate coral is, I think, Calyptrophora. In fact, I didn't write that one in. That's Calyptrophora angularis, I believe.
And that rock is pretty full of them again.
Oh, wow, look at that. So those are a lot of coralliums on there, too, I think. But, look at the size of this sponge. It's a beautiful big Polyopogon. Rock looks really black here on this side.
Yeah.
Is that just because it's not sediment?
I'm not sure.
Yeah, it's a little overhung, so maybe it just doesn't gust it up.
That might be what it is, yeah.
Or maybe all this rubble down here is just stuff broken off of it. No, not likely.
No,
Could just be the lighting,
the top
too.
of this thing has more of the dusting on it. I think it is just that steep.
Yeah.
There's a very leggy bamboo colony in the upper right there, too.
So if you've tried to sample one of these here, it would just fall in the sand.
I don't know. So Pseudoanthomastus, I think. Oh, there's tentatively calling them Acanella weberi. You see this? If we could zoom in on that coral that's right directly above the sponge, kind of spidery looking one.
Yep.
the first of those.
Okay. Maybe lift up for it here.
There is everything here.
Okay, video, come in.
And it may not be Weberite, it may be something else, but no, it's branching from the nodes, it looks like. So let's see if Scott and Tina or Asako will weigh in. And if you could get right at the center where it's branching from, that would be really good.
Working on it here.
That's full zoom.
There you go, there's a branch.
Yeah, it looks like it's from the nodes.
There's 3 branches coming off.
Yeah, and then a little further up too, if you can pan up just a little further right at the main branch point.
Okay, sorry.
Yeah.
I started to sink back down for the rest of it. Fight my last command and go up a little more. There you go.
Yeah, look
There's
at that.
The main branch. Oops.
So its real name is not going to be Acanella weberi. It's going to be revised, but I think that we're still using that for the time being for this thing, unless I... Yeah, so Scott's going Acanella as well.
Finished. Good copy. Thank you, Nev.
Alright, video. I'll hold here-ish. If I can hold it.
Sounds good.
There
There's a little worm crawling
on it, yeah.
Yeah, a little worm, some type of a polychaete worm that's...
It's done with that side.
Yeah, it's finished work. It's commuting home. Right now. They work out at the end of the branches.
There's some polyps right here on the foreground.
Yeah. Polyps are all coming off of one side. I don't know if that is I don't think that's escaped Scott's attention.
Okay, I'll come out for a establishing shot.
Okay. Do we have lasers on Copilot?
That is a very dark looking lyrate.
Just hold.
Coral there. I don't know what that is.
There it goes.
Okay.
I'll lift up and get the lasers across a couple branches there so that, yeah, there you go. Cool. All right.
Beautiful.
At about 80 degrees, pilot.
Okay.
Ship's been stopped for a few minutes. Cirrus is still swaying a little bit.
Okay. Yeah, if we were able to grab this entire rock, how many species would we have? Let's see. 1, 2, 3, 4, 5, probably 6, 7.
Maybe they could just hold it in the grabber arm.
Right.
No lock valve, sorry. Otherwise we would.
Scott says this looks like what is called Acanella weberi, but the one sequence available on GenBank with the name is not an Acanella. So, but yeah, so that's another thing that's going to get fixed, I think, when Scott and Les finish their work in this family, the bamboo family. Ken Sulak's on the chat now. Welcome, Ken. So we're let's see. He's talking about the eels that we've encountered.
Yeah.
Something weird. Oh, that's just maybe possibly no longer alive.
Oh, yeah. So we've got a different sponge here. This looks like it's a conolasma.
How are we doing, Copilot?
Looks like the swing settling out.
This is a glass sponge.
Do you want a longer look, or are you content?
I'm pretty content, but how about a snap zoom real quick? And all I'm checking for is whether there's any anemones on it.
Any what? Sorry.
Anemones. Commensal anemones, and it does not.
Oh, it does.
Oh, it does. You're right. It does.
Yeah.
So that was definitely worth a snap zoom. So then that's going to be one of these weird Lefroyella, I guess. So we actually collected.
I remember that, yeah.
Do you remember this?
Yeah.
It could be the same thing,
Take
but
a
I'm
slightly
not sure.
closer zoom on the edge there.
Okay. Like that?
Yeah, that's a beautiful, beautiful shot. Look at the channelization you've got going on.
Okay.
We're likely to discover that Lifroyella is not the only genus that has these commensal
lasers
anemones.
on it real quick for the sake of scale.
Awfully different colony morphologies.
So is this an old dead one up here?
Yeah.
Serious, stop swinging.
Yeah, it could be.
Thank you.
And Opie's bailing.
Yeah.
So,
Nice sound effects. He's going to roll, too. Oh, no, he grabbed on. I was going to say, he's going to take the roll. Roll approach. This is pretty dense rock here. Still on that one boulder a little bit. Or it's more of a nose, I guess. It's not so much a lonely boulder.
It's beautiful. Spectacular. And these are, I think these are huge corallium here that are in the back there.
At some point, we're just going to have to bite the bullet and go for one. Just
well,
being,
if
being
that's really
picky since so many.
If that's a corallium on the right there, that is humongous. It
Yeah.
certainly looks like one.
Do you want a quick, tight
Yeah, just to
look?
confirm it really is that, rather than
Come on in,
these
video.
pinky bamboos. I think it's...
We're snapping on that pink one in the background there.
And the one to the upper right, actually, too.
Is this it here?
No, it was the, even the bigger one.
Oh,
yeah,
come
this
back
is
out one?
corallium
Yep.
for sure. And then
Oh,
the
those
one,
are two separate. I thought it was the same piece.
What's that? No, I was.
Did you say stinking bamboo?
No, I was saying that there's some. No, not stinking bamboo. No, pink. There's some pink bamboos.
Sure.
Yeah, I just wanted to.
Sure, that's what you said.
All right, you can come in tight again here. I think this
I
is
would
what they wanna look
never
at.
blaspheme your coral, Scott. Never in a million years. This is an enormous corallium. I can't even imagine how old this thing is.
As promised, more corallium.
Yep. So I don't know. Whenever.
Spectacular rock, this place.
Yeah, it's just extraordinary. I don't know, wherever you want to try to grab a piece of one of these things.
Yeah.
What are you going to sit on top of if you get it here?
This one is going to be interesting, but there's a couple along the edge, I
So,
think.
Chris, a
that we might be
question.
able to
I don't know that anyone has aged corallium. Do you know? I don't know what their growth rate is. I don't even know how to judge.
So they
only that this is older than most of the ones we see, but not how old.
Bright.
Yeah, so they have aged smaller colonies that are that have been brought up from several hundred meters, maybe 300 to 500 meters. And this is Rob Dunbar, Brendan Rourke, and Tom Gildeson, who've been doing that type of work. Particularly Brendan, I think, has done a lot of it. And these smaller colonies of corallium have come out to be several hundred years old. They are significantly smaller than this one.
Sure. So, yeah, this is the biggest one I've ever seen.
Yeah. So. And these things are, we keep calling them corallium because all of these sort of pink and red ones used to be considered the same genus. And they have now been split out to 3 different genera. And this is hemicorallium, I'm pretty sure.
Okay, I'm sneaking in so I don't come out so I don't bonk into anything here.
Coming wide.
Thanks.
Watch lead, this is Nav. Just to let you know, we have 54 minutes of the dive.
Another hour. Okay. So we're
Thank you
gonna
very
find
much,
a corallium
Nav.
in the next hour. A corallium we can pluck.
Yeah.
That guy, I
Get a
did
nice
scoot in a little close there on that zoom, and I was already getting nervous. So I think that guy is amongst too many other old coral. But there are some down on the edge below my skids here, and I might be able to
Yeah, I think we could go for a smaller one. It'd be a shame to do anything to a colony that size and that age.
Yeah, so there's 1 or 2 or 3 right down here that are kind of baby.
Sure, yeah, these will work.
The whole thing.
Yeah. In fact, if you can grab it as close to the base as possible, I think you have a better chance of it not coming apart in pieces.
Okay. So we know for a fact that these are very brittle.
Yes, we do. Good luck.
Oh, yeah. We like it when they're brittle and hard to hold. But we will do what we can.
They're important species, but it's one of the reasons I don't like to collect corallium, because they're just so hard. They just come apart.
Yeah, and actually these, although they are on this nice sheer edge, it's not a good toe hold. Let me see here. What I got? So the tricky part is not only do I have to be able to get in and grab it, but we have to be able to open the boxes to get them into the sampling boxes. And
how about if you wait and when we're not on quite such steep terrain?
Yeah.
Again, we've been seeing them all day.
Okay. Because right now I'd have to grab it and then fly away and put it in the box like we did with that sponge. And if it's as brittle as you're saying it is, when we go to fly away, if I can't just get a good grip on it while we're flying, we'll lose it.
Right.
So
yeah, I think we could.
We'll look for a flat spot that's got one so I can open the drawers, or at least have one out on an edge where I can get to the drawers. Boxes there. But have no fear, there's more to come. Still have an hour left.
Yeah, no.
And we're still on sort of the side a little bit here, so when we get towards the
It does seem to be the happy side.
Yeah.
Well, This could be one of the more spectacular places I've ever seen.
If you say stuff like that, you have to say it with some enthusiasm.
No, I am. I'm just staring at it. I'm just stunned. And actually, I was looking at that jellyfish that went right by. That
we've got footage of those guys before. I think they're odd little critters. Some more corallium.
Yeah.
But on steep ledges again. Do they prefer a steep ledge?
No, they can. Well, a lot of corals prefer steep, but these guys can also do okay on just smaller boulders sticking up off the bottom.
Yeah, that would be ideal.
I think you can find some of those.
Okay.
Once we get out of the bouldery part. But it's like I don't want to leave the bouldery part. It's so beautiful.
Yeah. Well, by putting off till the last minute to grab a corallium, since there's so many, we might end up finding something very unique that we don't want to miss out on. So, it's really good.
True. Wow.
Gonna do just a little look at the other side of this boulder, just compare, and then I'll continue on. More fun stuff here. You've got a sea star down bottom. Looks like something we might have gotten eating a coral last, but we can take a longer look if you need.
Yeah, I'm looking at it. I'm trying to see if it's what we imaged before or not. Because could be this evoplosoma, which is normally on corals. I think that's what it is. So maybe we can just keep going on and seeing if we can find something else that would be a little bit.
We'll continue onward. Ooh. be a good opportunity if this is one of those. Take a peek.
Yep, those are them.
Yeah?
Yeah, that's
is that one right there?
Yep, that's one. Couple behind it too.
This, if I can get down here, which looks like I can, then this might be our opportunity to snag it.
I can come down a little bit, pilot, as well.
Yes, please, if you've got it. What's the ship move look like?
We've been stopped.
Okay, good. You're looking at me at 58. Okay, so I got room to come down and settle, right? Come down and settle.
So you got to make sure you pick a rock for us with your sample.
Oh,
so
what?
You got to find something that you can pick up so that it comes with a rock. All right. Yeah.
Actually, if we could find one of these on a loose rock, I bet we'd have better luck, because then it would be harder to break it.
Well, I think if you can grab it low enough, this one's kind of elongate.
Yeah.
Then I think you got a good chance as long as you hold on to the, as long as you can grab it as low as you can and hold on tight, then I think the whole top part will come off pretty good.
Okay.
It's when you try to sort of grab in amongst the branches there.
Oh,
then
they sort of...
Yeah, they all just crumble.
I see. Okay. Well, we will... If this is right, then I think this is a good opportunity.
Yeah, this is right, and we can get some brittle stars to boot with this guy, it looks like.
Okay, co-pilot, you have the starboard bio box empty.
Copy that. Thank you, Nev.
Make sure that we're settled here, which it looks like we are. And brittle, huh? Okay. Video, I know we're going to want to take a little bit of a tight zoom before we actually snag it, so why don't you do that?
Roger.
And we'll get hydraulics spun up.
Is it the one in front here?
Yeah, the one right here in front of that guy.
Gotcha.
Perfect. Yep. Watch, it'll confirm that's the right thing.
That's the guy.
Okay,
that's
good.
Gonna be
awesome.
Okay, getting hydraulics spun up.
Thank you.
Yep, it's going to be...
What RPM do you want, pilot?
What's that?
Yeah, it's
what
going to
RPM?
be... Oh, sorry.
You get almost all the way up, 3/4 or more. Yeah, that's fine.
Okay.
Okay, thank you. So I'm going to grab near where it's nice and thick down at the bottom, and hopefully the top stays intact.
Co-pilot, make sure the port bio box is closed when you pull out.
Good. Perfect.
Copy that. Thank you.
So it's correct with double L.
Where's the brittle star?
Yeah,
okay.
Yeah, double L. Yeah, you spelled it exactly right.
I actually don't see the brittle stars that you're talking of.
It's in amongst the branches there.
Oh, okay.
So you'll get them when you grab it.
You know I'm getting the one in the foreground, not in the back, right?
That's correct.
Okay, good. All right.
Coming up just a little bit, pilot, that boulder in front of me.
Okay. Just make sure you don't come up so much that you tug me at all.
Copy that.
I don't want to get yanked around or tugged off.
So that main stem will be pretty durable. That's why if you go for the main stem, it should be okay.
Okay. Well, before I get in there and grab, I'll get lined up close, and you can tell me if I'm doing it right. Oh, yeah, see, I think you're tugging me now.
Yeah, just let me come
so
down
why
a
don't
little
you
bit
come
more.
back down?
Yep.
We don't want to be, if this is brittle, we don't want to be bouncing around.
Coming down.
Thanks. If the ship's stable, then you don't have to worry too much. That, yeah, there you go.
So this main stem is going to snap sort of like a stick hard candy or something like that.
Oh, good.
So it's going to be like, it's just the finer branches are the ones that are really brittle.
Okay. I'll try to grab as low as I can here.
Okay.
So if I grab here, it's going to be awfully tall.
Yeah.
I don't know if it'll all fit in the box, but whatever is on top might break into the box, or we can try to get into the middle and maybe lose a few branches as I reach in.
I think that's okay.
Okay.
You're thinking about going in right where you are now, right?
Yeah, right in. Trying to get in right in this area. I mean, it's a big claw and a little guy, so we'll see how it goes. And if you don't like what's happening, say so, I'll back out.
No, I think that's a good spot where you
Okay.
lined up right now.
Okay. Let's just check here. Let's do 5. Yeah, that's perfect, Copilot. Thanks. All right, we're going to come in. Yeah, see that branch I'm bumping right there is going to break off, almost guaranteed.
Yeah, that's okay, though. I mean, there's going to be some you're going to lose for sure. It's unavoidable.
Okay. But I'm going to try to reach through and get that back piece, that thicker back piece, right?
Right.
Okay. Just going to go nice and slow and easy here. Yeah, I'm going to... Yeah, this might knock it over. I'm getting fearful here.
Well,
you call if you're on the same boat.
No, I think you're okay.
Okay. All right. I'm going to go for the grab here. Slow.
Moment of truth.
See how this goes. Slow.
Snap.
This is like one of the final manipulator tests that if you can actually get this in good shape, then
you're good.
You get your...
Ooh, it broke all the way down at the base.
Holy cow, that's a big piece.
That's a really big piece.
Well, we will aim as much of it in the box as we can, and then what won't fit, maybe won't fit, but I don't know if that's...
You can just crumble it in the box.
Now we're going starboard.
Yeah, we need to go starboard, yeah.
Copy that.
And I know if I wave this through the water much, it's going to break, right?
Yeah, well, you're going to lose a lot of your smaller
polyp-laden
branches.
Yeah, okay. Well, we will do what we can to...
Even if, again, some of it breaks in the box, it's no problem at all. There'll be enough material to work with.
Okay, well, we'll just carefully get it up. And I'm sorry this is taking a while. I just want to be very, very careful. Small movements.
Right.
Pilot, let me know when you want me to start opening the drawers.
Okay. I'm clear of the drawer, so you can very carefully start opening it, making sure that...
So first I'll retract the port bio so we make sure that lid stays closed.
Yes.
Okay. And then I want to extend the starboard.
Yep.
Okay. Starboard's extending. And then I want to extend the drawers.
Yeah. Sorry, I'm not really listening that well, but I am trying to comprehend it.
Copy that.
And listen, but I am also trying to focus on getting the majority of this into the box.
So is this going to be port outer, or what do you think?
Port side.
Port side, okay. both inner and outer, maybe.
I don't know if we want to try to sneak anything else in this. Once if it crumbles, it will crumble where it crumbles, and just try to get it.
Put the
starboard
in the port.
Starboard? You mean starboard side is where you're putting it, right?
Yeah, starboard
side.
Co-pilot, would you like to put any cam on that monitor, the right monitor?
Sure. Monitor right. Minute cam.
Either manip cam or mini SUS, yeah.
I don't know if you're looking at the feed there on the manip cam, but this is humongo.
Yeah, I am. I'm entranced with how you're going to do this.
I am not sure how I'm going to do this. This isn't.
Maybe it'll wind up in both boxes if you kind of.
Okay, I can sort of aim to spread it over both boxes like this.
Yeah, it just didn't seem that big when you were actually grabbing it.
Yeah. Okay, so option A is we do this. Option B is I drop the stem in first and then try to grab the top and put some in, or
I would say put the top in first so you secure the top.
All right, the top's the most important portion.
Right, yeah.
Okay. All right, in that case we will put, and we're drawn all the way out, or mostly at least.
I believe so.
That's the pin, yeah. Okay, yeah, perfect. Good job. Looks like drawers all the way out. Okay. All right, into the box it goes. I don't know if I have the geometry for this quite right.
Again, if it starts breaking into pieces, I wouldn't worry about it. They just never come up intact, these things.
Well, we're going to get
you can
at least one big chunk in there.
Yeah, and that'll be good.
With its commensals, anyhow.
Can I do anything with your views, pilot?
No, that's okay. You've got the right views up.
You know, the stars on it are kind of brittle, too.
Thanks.
Couldn't resist. Sorry.
I'll be here all week, folks.
Okay. Break now. You said it was brittle.
Well, the main stem's not as brittle, but maybe you can drop it already. It won't float, and then like kind of push it.
If I drop it now, I'm afraid the majority of the weight is outside the box, depending on how heavy the stem is compared to the rest.
Oh, I see.
There we go. I think a piece just broke loose. Try that. See if I wiggle a little here. Oh, of course, the ophiurite is trying to hold on to it. The one piece I have, we have an ophiurite hanging on. It's like, no, I want that piece of coral that you have in your hand. Please let me keep that piece. Let go?
Yeah, I think I would try and let go at this point.
Yeah, I'm going to get it over the box here.
You got the major heavy piece in the claw there.
And I'll just drop from here this piece. And maybe give this piece a convincing little nudge. waving around here. Hold on, let me let this roll go by. There we go.
Maybe push the top into the corner.
Trying, yeah. Sometimes these things are tricky. Trying to keep that nice intact piece intact while we push this little bit in. We might just close it at this point, so as not to do any damage to what's in there. But I'll try to.
Probably okay to push it down a bit. Like I said, I've never seen one of these come up intact.
How's that?
It looks pretty good.
That's in the seal.
Yeah,
we will collect it.
Okay.
Excellent.
I think I got majority of it in there even. There's one piece hanging on the drawer. But otherwise, we're in. Okay.
Okay, Pilot, you ready for me to bring in the drawer?
Terrific. Thank you very much.
You may close.
There's also a little piece sitting on the side of the box.
Yeah, don't ask for the rest of it.
Yeah.
That's all you get.
No, I meant that it'll be interesting to see if that falls out on the way up.
All right, it's in. Off we go.
Oops.
Where is Sirius? You lost me in your view there, didn't you?
Yeah.
Let's get all right. Slide HD. Yeah. Okay.
Thanks.
You're facing off that way. So why don't you face north real quick, just to see if.
You want me to come left?
Yeah.
Okay.
Oh wait. Look at the fisheye. I'm off on the right.
Yeah, you're off to the right.
Yeah. Sorry. Come right. You were at the light. You knew where I was. See him?
Oh yeah. You're just off to the right.
That's fun.
So we do have 30 minutes.
30 minutes left.
That is correct.
So maybe what we can do is just try to make a run along the summit here and only stop if we see something really unique.
Okay.
And see if we can get as close to the end point as we can. And we'll call it a wrap.
All right. I'll lift off. And which way do we want to head generally now? Back east?
Yeah, I'm going to put in a move towards the middle of the ridge right there.
Okay. So I'll head north a little to get towards Sirius and then head east.
Okay.
There you are off to the right, Pilot.
Perfect, thanks. I'll come your way. If you wouldn't mind rising up just a touch.
Copy that.
Give me a little space. And I will. All right. Hope that went all right.
Yeah.
Sample.
Got all of our samples.
Good.
All right, pilot and co-pilot. When you're ready, I'm going to put in a 50 meter at 090.
Okay. Just looking for. Sorry, I gave you a little tug there, didn't I? Backing up, just looking for the tether. Making sure I didn't leave it behind anywhere there. Yeah, okay, we got full tether view. All right, good.
Looks good.
And we're lined up for that, basically now.
Okay, so
090 is the way you want to head?
I'd be going over the ridge more.
That would put us
might do a little
somewhere over here.
I would do a little south of that, it looks like. This is all downhill for me. So I think going 090 would be awkward.
Okay, so
why don't we head over back in this direction? Yeah.
Would that be 110?
The waypoint is at about a 0.5 or 50 or 0.60 though. That's the only thing. We're just kind of going backwards if we if we go 0.90 or further south.
Well, we're not. Okay. Well, we can. Okay. Yeah. Okay. So you'd rather mostly make way to the dive.
Yeah, the dive endpoint just to survey along this ridge crest here.
Well,
that would be good.
I don't know that we're current. I was trying to get us back on top of the ridge crest because currently we are not.
Oh, we're not?
No, I don't think so.
We're on the
unless it's very flat on top
side.
and hard to tell. But which it might be. So you can. Yeah. Why don't you stick to 090 or that will head us towards the top of the ridge and towards the dive rather than sending us backwards.
Yeah.
Okay. We can do that.
Okay, so putting them up right now.
Perfect, thanks.
Bridge to snap.
Yeah, you're really tall too. You're really high up.
Let's put in a move right now.
There you go.
Range
That was the piece that fell in the drawer.
Bearing 090 at 0.2 knots.
Amazed that it floated like that.
That's a good copy already.
Yeah, it's coming down.
It's coming down.
Got a little push from the thrusters, I guess.
Yeah.
Copy that.
Quick open the bow box. I'll try to catch it.
You got an unbranched ice it looks like over on the right.
Yeah. Power down HPU.
Thanks.
Assume you're not just joining us since we got about 20 minutes or so at this dive, but this is the NOAA ship Okeanos Explorer.
Copy that, thank you.
You're seeing a live video feed from the Deep Discoverer ROV. We're on a seamount that we're calling Delilah Seamount. It's inside the wake unit of the Pacific Remote Islands Marine National Monument. You're currently at a depth of 1,847 meters. And
come down a little more even.
We're on a bit of a ridge that's extending off the seamount where we've discovered quite a beautiful community of deep water corals and sponges. So it's been a very successful dive.
We've already got our samples in the bag.
Got our samples in the bag, some rocks and a coral and a very bizarre sponge we call the kebab sponge. And we've come up and got some beautiful community shots, close-ups of some really unique animals. So this is an area of the monument that was expanded by President Obama a few years ago. So the monument boundaries originally were 50 miles out from Wake Island, and then President Obama expanded them out to the U.S. Exclusive Economic Zone, which is a 200-mile boundary around Wake Island. And we're just inside of that boundary right now. We're in sort of the northwest side of the monument. There's another eel coming down. It's at the upper part of the image.
Yep.
I don't know if it's a ways away or whether we could...
We'll try to get it here. Go ahead on in video. Just bringing it around into view. Okay, dropping a bit. Hey, co-pilot, would you mind trying to turn on our upper lights?
Copy that.
Thanks. Upper swing arm lights. Come on out.
Coming out, yeah.
Thanks.
There's a stock sponge in the background there. It looks like it's in the genus Caulophacus.
Full way.
Thanks.
The lights are under.
Under the ROV. Hit the ROV tab on the left, and then bank 2.
Copy that.
Nope, bank 4. It's one of those. And then upper swing arm. Bottom left.
Good copy.
Yeah, there we go. Light it up a little better out there, since we're in this kind of terrain. You can, what's your altitude? 20. Just going downhill is kind of awkward.
We're still in that same terrain of these larger little hillsides covered in manganese with some cobbles fallen out, probably little pillow mounds, something like that. Thickly coated in manganese, and it appears the animals really like hanging out, particularly on the rocks that stick out a little bit higher.
Yeah, most animals, I think, that are filter feeding like to get up a little ways.
They like the view too, probably.
Yeah, that's true. I guess what it means is that there are quite a lot of differences between ocean views, right?
Yeah.
They all have an ocean view, but some of them are better than others. These long stalked orange things are a type of soft coral, Pseudanthomastus. It's actually a type of a mushroom coral. Now, watch lead.
Go ahead, watch lead.
Yeah, so I'm just not understanding the HIPAC screen there. It seems like the ROV is pointed backwards. Is it just I'm not reading it right, or is I'm trying to understand what's going on? And
So I can face the way of track, but there, I'll just show you. It's all blue water over here. The way that we're heading to the dive right now, the HIPAC's off by a little bit. So if we come out this way, and I get out in front of Sirius, and I face towards the dive, and my sonar returns were very small. There wasn't very many of them. So I was trying to follow the uphill so that we're not going downhill with ROV while we make progress with Sirius towards the dive.
But the contours in HIPAC are actually pinching together right where the ship is showing, so probably this is the local high, and in order to get to the end point, it's probably going to go down a little bit before it goes up again.
Okay.
It's my guess.
Yeah. Well, I'll get D2 lined up. Wait for a ping to come, and then you'll see us.
Okay.
Come around. So this was just, I thought, kind of a boring view. And so I was just trying to keep interesting things in view until the last minute. Just because this is...
Yeah, it's the cover the area, actually. Even though we go out of animals, that's actually important for the understanding, even if we go to a more barren area.
Oh, I actually meant that this is downhill, so you're not going to see a lot of anything.
Oh, I see what you're saying.
It's, it's deeper here, and I think
yeah,
it's
I think it'll level off, though, right?
Yeah, it should.
Level off at some point.
Okay.
Well, we're going through a little valley.
Dive end is
or a little low point along the ridge.
Okay. Well, we can fly like this and just head towards dive end, and then
I see.
Yeah, let's go ahead and do that. Let's head toward dive end.
Yeah. Ship's on its way that way. We were just trying to keep the bottom in view, per se. You can see the bottom here.
Yeah.
But it's not. It's going to be very blue and kind of awkward for me to fly. So I was trying to make it less blue and a little easier, and then make our way. But here we are, coming around, and I'm just trying to keep bottom in view as I swing so that we don't lose track of it.
Pilot, I can zoom how this gets out of the frame if that would be better.
Sure. And, yeah, so why don't you come left a little, co-pilot?
Copy that.
Thanks.
Nav, how many meters do we have left in this one, do you think?
Dry left, time of bottom is 3:33.
Okay, what's your altitude? About 20, okay. Okay, so since Cyrus is up, he's at his comfortable altitude, and I'm 20 meters below him. So this is... I can go a little farther down. It's going to start tugging soon. This is about
3:33.
where we can hang out.
Is that ship move completed, or?
That is correct.
It is. Ship move is completed.
Yeah.
Copy
It
that.
finished,
This is almost
about a
sheets
minute
of
ago.
manganese here, huh?
Copy that.
It's all covered.
Yeah.
But you can still sort of see the rounder shape underneath it that's being covered.
All right. So,
Right.
see how I'm at Delta 20?
Right.
We're going to have to hang out like this, and you just make sure your altitude. You can come down as your altitude allows. So when you start to get to 22, you can come down to 20, 22, 20, 20.
Copy that.
Just don't come down too far.
Do you want to start making a final push towards the dive site?
Yeah.
So we can put another ship move, maybe 070.
Yeah. Which way are you facing? 090. Okay. So yeah. Okay. Final push towards the dive site.
Copy that.
Sounds
So
perfect.
070.
Got
So
sort
this
of
is a little
mix of
downhill right here,
nodules.
and then it's going to come up again there. Is that
There's
what I
some
understand?
smaller pieces. Yeah, they
'Cause I
might
see
be nodules in the
something
sand
in
there.
sonar
Yeah.
out this way.
And then the bigger surfaces.
Something over here. Push towards
Manganese
this
with
sonar
some
high.
light sprinkling still of
That looks
sediment
like
like we've been seeing.
Okay, so that's going to be
Yeah.
50 meters at 070. Bridge, this is Nav.
Go ahead, Nav.
Okay, that's all I've got.
I have another move for you. It's going to be 50 meters at 070 bearing and 0.2 knots.
All right,
Here
I've
it looks
got
more like
50 meters
yesterday again
bearing
with a lot of dead
070.2.
sponges.
Yeah.
Is that correct?
That is correct.
All right, stand by.
So this is sort of like a little catchment basin or something right here.
Yeah.
There's a low part of the ridge.
Yeah, we just
We're
went through the lowest part. We're starting to go uphill, so this is where most of
Army
the
Nav
sediment
Bridge,
will end
move
up.
has been initiated 50 meters
At least
bearing
partially
070
collecting,
decimal
and
two.
part of it will find a
Copy that,
steep
thank
way
you.
down from this area.
Pilot and co-pilot, the move has been initiated.
Thank you.
So just that quickly, the corals disappear as soon as we go into this type of terrain. They're kind of, I mean, there's plenty of hard terrain for them to attach to. They just, it's not providing them what they need, I gather, this little...
We might be out of the current a little bit here, too.
Yeah.
There's a sponge to the right. Seems to still be alive.
Yeah, let's go ahead and skip that and let's see if we can...
Yeah, just
make
didn't know
some-
if you saw it, so I was naming it. Not looking to zoom. Just trying to mosey along and see what we can find.
Interesting to see if, as we get closer and it starts to come up again, whether the corals reappear.
Yep.
Whether it's just this little dip here that is nobody likes.
Cheap land.
Yeah. So are these just pieces of cobbles, or do you think they're actually nodules? The way nodules form on the abyssal seafloor.
The little bits, yeah, they could be nodules.
Okay, pilot, I'm starting to get some movement. And you're drifting off to the left side of my screen.
Yeah, I'm going to do a little bit of mowing the lawn.
You can also get, if you have small little pebbles, small little rocks, or even pieces of sediment that end up sitting around, they end up becoming a good surface for the manganese to start growing on. On previous cruises, we've dredged up nodules from sides of seamounts that had anything from little pieces of volcanic rock to shells of animals inside of them. Manganese isn't very picky about what it's going to precipitate on.
Just needs a nucleus of some kind.
It needs
if
something
it's
solid.
Yeah, something, no matter how small.
There's
another anemone.
Two of them even.
Oh yeah, here's another one sitting, looking the other way.
These are the ones that we saw right off the bat, I think, because it's got that sort of those larger tentacles on it. Treatopleura sponge sticking up. But yeah, pretty barren area compared to where we just came from.
Yeah. There's our sponge. On the right soon. Oh, it's the type of that all these dead sponges are, I think. Like, I'm gonna see it soon, right? Like now-ish.
Yeah, so there's a live one. And I think this is probably the Feria near Oka.
And I'm just going to keep zooming along unless you say otherwise on any of these.
Okay, thank you, pilot.
Yep. If I catch something in my cameras, I'll bring it into view and then you can tell us whether it's interesting.
There's a little Bolosoma sponge. Make a note of that one.
Somebody's having a little party right up top there.
Yeah, the crinoids like to jump on anything. The question is whether or not they're just climbing up on dead sponges or whether they're the cause of the sponge dying, just because we saw them completely covering some of the sponges earlier on.
Yeah, how many meters do we have left in this move?
And an enemy on that guy, it looks like.
Yeah, a little anemone.
Oh, there's some bigger stuff up ahead.
Yeah.
A bit further uphill. It's interesting, it's totally changed all of a sudden from all these different corals to...
Now we're getting some more sponges.
Stand by, co-pilot.
It's the one in front of us is just the Walteria that we've seen, that sort of elongate translucent one.
What is that? Oh, it's another.
It's got a crinoid on top of it.
It's kind of weird looking thing on the bottom.
And another one of the anemones, and then...
Is it folded up?
Should be about...
I guess it's just tentacles.
Should be about 10 meters.
Another of these Spidoscopula sponges on the right that we've been seeing today.
Pilot, we're coming to about Less than 10 meters left in this moon. Do you want to tack on another 50?
I was going to ask that.
Yeah,
let's dial this in right to the end.
Do you want to bump up the speed to 0.3 since it's been pretty barren?
If we want to make it there, I'm happy to do that. How long do we have left? It's 5 minutes?
We only have less than 10 minutes, about 7 minutes left.
And how long would a 50-meter move at 310 take?
That's a pretty distorted one of those sponges there on the left. That's an interesting one.
That would take...
I think it's the same thing as the one on the right, just they
about 5
have
minutes.
different type of morphologies.
Yeah, sure. Can do
Copy
it.
that.
And when it comes near the end, then we can just call it.
Copy that.
Call it off if we don't make the whole way.
Okay.
Sako Matsumoto just commented on the sponge. It was a gorgeous specimen of that sponge.
Bridge, this is nav.
And this is again the same thing, just smaller version of it. So this one's a bit younger than the one we just passed over.
50 meters, bearing 070, and then the speed is going to be 0.3 knots.
Down there, maybe.
Well, maybe we could take a it's the first urchin we've seen. Maybe we could take a peek at this guy.
That is correct.
Copy that. Stand by for input.
I didn't think we were going to see anything different toward the end here, but
come in video.
Well, we are sort of seeing something different in that the community has totally changed.
Yeah.
Harvey Nav Bridge, moves input 50 meters, bearing 070, decimal 3 knots.
Copy that.
And take a little more zoom.
Yep. Pilot, the move has been initiated.
Well.
Copy Nav.
What have we got here?
Let me repeat. Well.
Currently not getting any hints from the chat either.
No.
So
gonna see if we can coerce Rich Bowie over in the Cal Academy of Sciences to watch a little more often. So it's not this guy. I think it's in the family Podinidae, but I'm not absolutely positive. Yeah, this could be Cynopodina. It's my best guess, and I'm sticking with it. So,
whoops. So far, nobody's disagreeing with you. So
yeah. So there's a
few more seconds.
It's in Hawaii we've got something called Cynopodina hawaiiensis, and this looks somewhat similar to it.
Yeah.
But again, we're pretty far away from Hawaii, so it's
going to be Wakensis.
Yeah, Wakensis. So maybe Cynopodina spa is the best way to mark that one in.
Interesting change here in the seabed.
Yeah, all consolidated, switching to sediment and broken up rubble.
Yeah, these are smaller pieces. The manganese crust doesn't look as thick. This may have been stuff that's been rolling more recently than the more massive things that we've seen to the side.
So this couldn't have been like a taller peak or something here that crumbled down. Sort of seems
yeah,
like,
it
seems like
it's probably
crater
crumbling
we're
from
in.
the thing you're looking at above it.
Yeah.
And it's just sort of breaking off and sliding.
So it makes you wonder whether the easiest way, if they were going to be mining the manganese crust in the future, is to try to find these sort of rubble areas and almost like the nodules try to harvest kind of the loose rubble
pieces first, yeah.
loose pieces first rather than trying to rip off all of the surface.
It's going to be much harder to, well, even if you do the rubble, you'll still have to remove the manganese from the rubble, and it doesn't necessarily have as thick of a layer on it. I mean, this would be more of a, anything sort of eroded off the surface here would be manganese crust. So if you could just sort of scrape even lightly, you would get a fair bit.
Mm-hmm.
Did we really change a lot in depth? I don't think we have.
We're at 1860, yeah.
This is possibly a coral over here.
Yeah, this is the Trissopathes that we've imaged. They were pretty abundant at the beginning of the dive. So yeah, we got, maybe it's going to pick up now.
Okay, pilot, we have reached the end of the
You never
dive
know.
right now.
It's starting off with a few individuals, and
this one has a feather in its cap too.
yeah, this is the Cerametra, the purple crinoid.
Watch lead. This is Nav. We have reached the end of the dive. It's time to come up to the surface right now.
Okay, thank you, Nav.
If you want to spin up hydraulics, skip pilot, and get everything stowed away. I'm just going to bring my tether out to this side.
We were just informed that we have gotten to the end of the dive, and we are shortly going to leave the bottom.
There was a single polyhydrozoan that we just went.
Yeah, go ahead, RV9.
It's kind of interesting.
Let's go ahead and stop the ship right here.
Yeah, and look at this. We are starting to pick up with some things.
Yeah. Copy that.
It looks like Paragorgia there, that red one.
Couple of sponge.
There's some of that guys in here.
Yeah. So, I mean, it hasn't picked up a whole lot, but it's picked up a little bit.
Notice this is more out of the sand again.
Right. And also, this is kind of a little rise.
A little bit higher up again. They really seem to like this the best.
Glance out in the distance, and don't see anything sticking up out there. And time to go. Bye.
You have to say thank you to Delilah.
Yeah, for some arms.
So for those of you who are still on the conference line, I think we'll give, I guess, Lindsay 5 minutes to prepare the Fleeter Mouse display, and then we'll have our dive planning call. You know, some of you, it's pretty late. So this will be 5 minutes after they pull off the bottom here. And there she is. You ready?
Alright, I'm at
Okay,
090.
well, Lindsay's
Stay
here, and
locked in
she's
for
going to
now.
go ahead
Copy
and
that.
And
That
you can start. Come up
port
as you
side
need, just watch your altitude.
swing
So-
arm.
What's wrong?
The port side swing arm did not come in when I went to stow.
Lower swing arm stow. Try again. Check your hydraulic pressure for me real quick. Pressure's up pretty high, huh?
Yeah.
Try it, I guess, one more time.
Let me see once.
Okay. We're up and basically lined up, and the lower still will not stow, huh?
Yeah.
Hydraulics, port. Oh, port lower. Stow. There it goes. Yeah, so you gotta go to the port J box for the lower port lower to stow. Sorry.
Nah, it's my
I
fault.
didn't look up and see which J box you're using.
Copy that.
Okay.
stand by shore. We're discussing tomorrow.
Ready to
Tomorrow's
start coming up? You can come out of
dive
heading.
positions.
So your left, all the way left button.
This one?
What's that? No, we
press
can
it
go ahead
once.
and give the summary. That's true. And so while we're getting set up and double checking our positions, just a wrap up. Today, I found today's dive to be really exciting. Not only did we get a spectacular coral community and sponge community, but also the blue shrimp and this incredible kebab sponge
I'm
that
assuming
we
you guys are off the
collected.
bottom end.
30 meters in there.
We were able to get a corallium as well, a very dominant coral
We are
here that
coming
we're
up,
seeing all
Bridge.
day. So it really went
All right,
well.
copy that.
I don't know if any of you would like to comment on it. I don't know who else is on the teleconference line. If you're still with us,
So,
maybe you could announce yourself.
Pull in the drawer and retract both pins. Keeping that guy.
I think we've got still people on the
Put
chat.
that guy behind the zip tie there, so that your knob
Oh.
recovery, get it right behind the zip tie, or bring
Anyway.
the zip tie so that you can get it behind there.
We are going to be diving on another geo.
Yeah, there you go. Just be careful it doesn't like snap back. Yeah, that's fine.
Could we see the positions again
Yeah,
first?
you're at 35. Okay.
Okay.
There's 30. Okay.